Hi, I'm Amy Proal, President of PolyBio Research Foundation, and I'm very excited to welcome you to the PolyBio Fall 2025 Symposium. PolyBio is a nonprofit organization accelerating research on how viral, bacterial, parasite, and other pathogens drive chronic disease and aging processes. As most of you know, a large portion of our research and treatment program focuses on long COVID. Starting in 2021, we connected researchers at different global institutions into a consortium to study key root cause drivers of the long COVID disease process. These are many of the people that you'll hear from today who will provide updates on their projects. But what I want to talk about now is the big picture of the long COVID field and how we want to continue maximizing progress over the next years. Overall, here's where we stand with long COVID. Tremendous progress has been made, and hundreds of papers have been published by our group or others showing clear abnormalities in patients with the condition. So, for example, we know SARS-CoV-2 can persist in the tissues or host cells of some long COVID patients. You'll hear researchers like Tim Henrich from UCSF and Morgan Baumsell from Institut Cochin present data with findings on that front today. We also know that long COVID can present with neuroinflammation and immune dysfunction. For example, Mike Van Elzakker from Harvard Medical School will show you imaging data today on neuroinflammation in the brains of patients with long COVID and also with ME/CFS. But unfortunately, despite all these findings, there is not a single validated long COVID diagnostic test and not a single FDA-approved long COVID treatment. And this lack of validated long COVID diagnostic tests means that doctors have a very hard time making long COVID medical decisions. And it's also a big problem for clinical trials. Now, we're running and supporting multiple long COVID clinical trials at the moment, and you'll hear updates on several of those trials today. We also wrote this group consortium paper. It delineates a roadmap for testing interventions aimed at clearing SARS-CoV-2 reservoirs in long COVID. But overall, despite these efforts, there's not as much engagement from pharmaceutical companies that could bring their drugs into long COVID as there should be when hundreds of millions of patients across the globe are sick. That's partly because, as you'll learn from today's talks, different long COVID patients can have different primary drivers of disease. One patient's main problem may be a failure to clear the SARS-CoV-2 virus. Another patient may suffer from reactivated Epstein-Barr virus. Another patient may have autoantibodies that are a major issue in their long COVID case. Thus, and I know this is somewhat obvious, if a company wants to run a clinical trial of their drug in long COVID, they want to be very confident that they can recruit patients into that trial with the issue that their drug treats. And that's where validated diagnostic tests become pivotal, so that you can measure a specific biological abnormality in long COVID patients in a trial and track how that marker changes over time. And without validated diagnostic tests for those markers, pharma companies are more hesitant to engage and pay for the cost of expensive long COVID clinical trials. So at PolyBio, we're working to solve for this problem by launching a new program called the Long COVID Cure Initiative, or LCCI. It's a program to directly accelerate and validate the top diagnostic test in long COVID, and then to work with pharma and our Mount Sinai Long COVID medical education program to get the diagnostics strategically positioned into clinical trials and medical guidelines. The first part of LCCI is VIPER, a program designed to validate the top diagnostic tests for persistent SARS-CoV-2 and other drivers of long COVID by running them head-to-head. And you'll hear more about VIPER today from Steve Deeks at UCSF, where the program's based. And it's important to know we're actively fundraising for VIPER. We've gotten over $1 million to kick off the effort, which is incredible, and we're using that momentum to find other donors. But here's the basis of VIPER and why we're working so hard to make it happen. As you'll learn from talks today, much of the long COVID disease process is happening deep in tissue. So, for example, we're finding SARS-CoV-2 often persists in tissue, which teams are collecting from body sites like the gut via biopsy procedures to find the virus. But these biopsy procedures, they're invasive, they're expensive, and they're not possible for the average doctor to perform. Meanwhile, teams in our network, including people you'll hear from today, have cutting-edge diagnostic test platforms that can identify tiny amounts of the virus, its proteins, or virus-directed immune responses in easily accessible samples like blood or saliva. For example, An Lee from Emory and Mark Painter from John Ware's lab at Penn will present today on diagnostic test platforms that can act as immune biosensors of SARS-CoV-2 persistence. Un's platform Mensa captures short-lived plasmablast immune cell responses to persistent viral antigen, and Mark's team uses tetramer assays to identify T cells responding to persistent viral antigen. And where Viper comes in is that the program will collect a central set of accessible samples and gut tissue from the same extremely well-characterized long COVID patients and healthy volunteers. Then groups like Un's and Mark's will receive identical sets of Viper blood or saliva samples. Viper will compare each team's diagnostic test measurement in the accessible samples against the tissue-based measures of persistence and to each other to determine the accuracy of each test for assessing viral persistence. And over time, this head-to-head infrastructure, this Viper comparison infrastructure, can be expanded to validate diagnostic tests that measure other biological drivers of long COVID, such as blood vessel dysfunction, immune dysregulation, and mitochondrial issues. So this is the overall long COVID cure initiative plan. Validating diagnostic tests via the Viper program gives us targeted diagnostics that can then be positioned into long COVID clinical trials, allowing the trials to more accurately recruit and track patients over time. That gives us smarter trials. Smarter trials leads to increased engagement from the pharmaceutical industry, who bring more of their drugs directly into the long COVID space. That gets us to effective treatments and eventually to cures. A huge strength of Viper is that diagnostic test platforms validated to identify persistent SARS-CoV-2 can very likely be pivoted into the much more accurate identification of other infections that also currently lack good diagnostic tests. You can already see this evolution happening. For example, An Lee and Mark Painter have pivoted their Mensa and T-cell immune biosensor platforms to identify Epstein-Barr virus and other herpes virus infections in addition to SARS-CoV-2. And this platform evolution can continue. For example, some platforms can be pivoted into better detection of Borrelia, the Lyme pathogen, or the enteroviruses that have been implicated in ME/CFS for decades. In fact, you'll hear Michael Peluso from UCSF talk today about a new program we're supporting to study enterovirus persistence, brain and spinal cord T-cell activation, and other understudied drivers of ME/CFS. The program is called CHIME, or Chronic Infection and Inflammation in ME/CFS, and it's one of the most exciting new efforts in the ME/CFS space. Really, the need for more accurate and validated pathogen persistence diagnostics is tremendous because persistent infection is increasingly being implicated in most chronic diseases, an active area of research for PolyBio. For example, just yesterday, this paper came out positioning Epstein-Barr virus at the heart of B-cell dysregulation in lupus. A growing body of research also places persistent infection at the heart of the Alzheimer's disease process. On that front, Ben Reedhead from Arizona State University and William Emer from Harvard Medical School will talk today about research connecting viral activity directly to Alzheimer's disease pathology, including the formation of amyloid plaque and tau tangles. Indeed, pathogens can drive many aging processes beyond cognitive decline. I recently wrote this paper with my PolyBio co-founder, Mike Van Elzakker. It provides examples of how pathogens can drive every feature of human aging, including mitochondrial dysfunction, microbiome dysbiosis, immunosenescence and T-cell exhaustion, epigenetic alterations, inflammaging, cellular senescence, and telomere attrition. Really then, the output of Viper will have flow-on effects to improve diagnostics for infection across the entire chronic disease and aging space. To maximize the diagnostic test platforms and teams included in Viper, our PolyBio team is holding a 3-day in-person meeting this December. We've invited 25 of the top teams in the world with novel, cutting-edge platforms capable of capturing components of infection in accessible fluids to the meeting. Here's a list of just some of the people coming. We'll be writing a white paper with key output from the meeting, and I look forward to sharing some of the insights with you at the next symposium. So with that, let me introduce the next speaker. It's Nicolas Huot. He's a postdoctoral researcher in Michela Muller-Trutwin's lab at the Institut Pasteur in France.
Thanks a lot for inviting me to present where we are and where we go regarding the characterization of SARS-CoV-2 infection in a non-human primate model. Just a quick introduction. As you know, it's well admitted nowadays that, in fact, SARS-CoV-2 could persist in different parts of the body. It includes the intestine, lung, brain, and different other tissues. It has been recently estimated that around 0.1 to 0.5% of infection may lead to a chronic infection in the general population. Nevertheless, as you mentioned previously, it's difficult to study due to the fact that access to deep tissue in human is difficult to perform. Hopefully, we have generated 2 animal models which allow to characterize and to study persistent viral SARS-CoV-2 persistent viral infection in different tissues. Indeed, the hamster model is used to study persistence of SARS-CoV-2 in neural tissues, whereas non-human primate model could allow to study SARS-CoV-2 persistence in different tissues. The main problem today is how and which mechanisms that sustain viral persistence in the nature of the host and the nature of the host immune response that accompany this is poorly understood. Indeed, we speculate that this is a dynamic interplay between the virus and the immune responses. In one hand, persistent infection might drive low level of replication, chronic immune activation, local inflammation, and tissue remodeling. In the other hand, the sustained immune response may select adaptive mutation or escape variant and lead to immune exhaustion, tolerance, and compartmentalized dysfunctions. So the aim of our study is to use animal model to answer some question regarding the capability of the virus to persist in the individuals. And we aim to answer different questions, which include which cellular or tissue reservoir allow SARS-CoV-2 to persist despite an active immune responses? How does persistent SARS-CoV-2 infection influence local tissue remodeling and inflammation? What are the consequences for long-term immunity, and which immune cell population contribute to recovery from long COVID? So to perform that, we use the macaque models, and we constitute 2 groups of animals. One group of animals were infected with the BQ1 strain and monitored during 23 weeks. Another group was infected with the BQ1 strain, vaccinated, then reinfected with the GN1 strain and monitored during 21 weeks. The idea of the second group was to mimic what does happen in the human population, in which there is a lot of people which are being infected, then vaccinated and reinfected. So we collected different samples during the follow-up, and we collected more specifically a bronchial lavage at day 8, 15, and 23. And as you can see, after 3 months of the acute phase, we are able to detect the viruses in all the cells isolated from the bronchial lavage. And at 15 weeks after the acute infection, we start to see a decrease of the virus quantity, RNA, in the bronchial lavage. And at day 23, some monkeys have completely resolved the infection, others still harbor a virus in their bronchial lavage. We next decided to collect bone marrow, and we do exactly the same approach. We perform qPCR to detect the virus, and we were able to detect the virus in the bone marrow in most of all the animals in which we detect the virus in the bronchial lavage. And at euthanasia, we collect all the tissue, and again, we perform different qPCR to detect the viruses, and we are able to detect the viruses in all the axillary lymph nodes, some cervical lymph nodes, and according to the monkey, we were able to detect the virus sometimes in the liver, other in the lung, sometimes in the spleen. Based on this, we decided to see if the virus was isolated from the tissue, were able to replicate. So to perform this, we co-cultivate 1 million of isolate cells from different tissue with virocell to amplify the virus. And as you can see, when cells were isolated from the bone marrow and from the axillary the virus could replicate and to amplify on virocells. Whereas when the virus is isolated from other tissues, the virus is not able to replicate and to be amplified. We do not perform yet the characterization of the virus in the gut. This is something which is still ongoing. We next perform some staining to see where the virus localized, especially in the axillary lymph node. And as you can see, we are able to detect replicating viruses in the section of axillary lymph node. So it's shown by the red dot plot on the picture. In blue and yellow, you have the B cells, and in red, in blue, sorry, you have the CD31 cells which stain the vessels. We are able to see that, in fact, when the virus is present, it's present in cells, and we can detect some replication in the cells, suggesting that the virus still replicates in the second lymphoid tissue, and it's not in latency as it could be observed for example, other tissue in other viruses. Interestingly, we also observe that in the lymph node from monkey with persistent viruses, there is a huge proportion of second lymphoid follicles. They represent around around 80% of the total of B cell follicles, suggesting that there is an activation of the immune response, especially on the B cell responses in those tissues. This is something that, again, we are still investigating. Next, we decided to look if there is a specific inflammation in those tissue and in the monkey with a resolved inflammation. So to perform that, we perform a multiplex analysis of 37 cytokines, both in the plasma and in the lymph node homogenate at euthanasia. Unfortunately, we are not able to detect any variation regarding the 37 cytokine when we compare LC control with persistent or resolved monkey. At the opposite, we found a huge difference regarding the inflammation in the lymph node in the monkey with persisting viruses. There is a very high level of many cytokines, such as IP10, IL-1 beta, MCF1, IL-5, present in the axillary lymph node of the monkey in which the virus persists. To confirm that, we also perform the same approach with different peripheral lymph node, and we choose the cervical lymph node. And as you can see, there is also huge inflammation in the cervical lymph node. So it's the bottom part of the slide. And when we compare the inflammation between the axillary and cervical lymph node, we do not see exactly the same picture, suggesting that, in fact, the inflammation results of the region which is drained by the lymph node itself. Next, we also decided to characterize the immune response. So we perform a first approach characterizing the lymphocyte responses, which include both total CD3 T cells, and we can segregate between CD4 and CD8 T cells, but also NK cells based on the expression of NKG2A. We perform unsupervised clusterings, and we are able to define different cluster of NK cell, but also of CD4 T cell and CD8 T cell based on the expression of CD45RS, CCR7, and CD62 ligand to define naive, memory, and effector CD4 and CD8 T cells. And we also used some marker to define and to characterize the NK cell responses. Just to give you a simple picture of what we observed, we mainly found differences regarding NK cell population. Indeed, we observe a marked alteration of NK cell population across all tissue, especially in the blood, the bone marrow, and the spleen, and the lymph node. And this alteration is also often sometimes in monkey, which resolves the infection. This suggests, in fact, that NK cell may undergo recruitment exhaustion or functional impairment in the context of viral persistence, which potentially contribute to the inability to fully eliminate the viruses. So we are still investigating how NK cell and why NK cell are blocked to recognize or to kill the infected cells. And to understand why we found this systemic level of NK cell disruption in chronic infected monkeys, we decided to focus on the homeostasis of NK cells. So again, we perform a new approach. We design a new panel which allows us to define and to characterize the different stage of NK cell maturation from the NK cell precursor until the fully mature NK cells. So for those, we use different marker, CD34 to define the NK cell precursor, CD20, 127 to define the NK cell intermediate maturation stage, and NKG2 and CD16 to define the different fully mature NK cell stage. Based on this, we use again an unsupervised approach, and we were able to define 80 clusters, which correspond to the different stage of NK cell maturation across 3 different tissues. So we focus on the bone marrow, the peripheral lymph node, and the blood. I will not show you all the results that we observed, but just to give you an overview and a picture, we found a huge accumulation of NK cell precursor, which is represented by the cluster 18 in the axillary lymph node of monkey with persisting viruses. When we compare to healthy control or to a monkey which resolved the infection. In the other hand, we also found an accumulation of intermediate NK cell maturation stage in the bone marrow of monkey, which resolves the infection. Taken together, these results suggest, in fact, that there is a huge impact on the hematopoiesis of NK cells, which could lead to an impairment of their capacity to recognize and to kill the infected cells. So to evaluate this, in fact, we decided to define a trajectory of NK cell maturation. So from the bottom part is the cluster and indicated by the nodes. The size of the nodes represents the proportion of the frequency of the given cluster. You have the edge in red, which represents the main way used by NK cell to differentiate it, and more the way the edge is in red, more this pathway is privileged by NK cell to become fully mature. And just to give you an example, we compare in healthy animals axillary lymph node and blood, and you can see that in blood there is more fully mature NK cell in comparison to axillary lymph node when we found more undifferentiated NK cells. Now, when we do the same approach regarding monkey with persistent and resolved infection, we found huge difference regarding the different pathway used by NK cell to become fully mature. Indeed, in persistent monkey, we found an accumulation of intermediate NK cell maturation stage, which are not fully functional. At the opposite, in the resolved monkey, another pathway is used, a conventional pathway is used to generate fully mature NK cells. So we are still investigating this approach, and we are validating our model in other infections. So what we want to do and where we go now, we plan to go deeper on the characterization of NK cell hematopoiesis, especially in people living with long COVID. We also define different panels to perform a phenotyping of NK cells and functional analysis. So we will use flow cytometry to perform that and spectral flow cytometry. We have designed some functional assay, and we plan to perform a data integration to define a good marker to investigate the impact of long COVID on NK cell in people living with long COVID. We also start to perform a single-cell SnRNAseq to validate what we observed by flow cytometry. So the idea was to isolate NK cell from different tissue and to compare, in fact, the profile of NK cell with the eyes of hematopoiesis. And then we plan to extend our observation on NK cell, if it's possible, to other kind of cells such as CD4 and CD8 cell to understand how the interplay between those populations could be responsible for the impairment of NK cell maturation. I will stop here. I would like to thank all our founders, and especially PolyBio, and all the people which are involved in this project.
Great. Thank you so much, Nicholas. That's amazing data. All right, our next speaker is David Price. He's a professor of infection and immunity at Cardiff University in the United Kingdom, and he'll be talking about mapping lung tissue correlates of breathlessness in long COVID.
Thanks very much, Amy, for the kind introduction. It's a pleasure to join you all again today and to continue the tradition of Europeans speaking first due to the time difference. So I wanted to just divide this talk into two fairly short parts. The first, just to give you an overview of what we've done so far using peripheral tissue samples that led us to the lung study that we're now doing with PolyBio. And then to give you an update on the work we've been doing in lung tissue with the funding from Amy and PolyBio. So back in the day, during the pandemic, we like everyone else, were alarmed to see the emergence of long COVID very soon after the pandemic, with people remaining sick despite apparent initial recovery. And so what we did in collaboration with a very talented clinician here in Cardiff called Helen Davis is we set up a case control study to see if we could figure out what was going on. Now, back in those days, we had no idea. So our plan was simply to collect as many samples as we could and measure everything that we could to get some clues. Now, these samples that we collected were from two groups. One was healthy convalescent individuals who'd recovered fully after a confirmed episode of COVID-19, and people with long COVID, people who had residual or new symptoms after infection with SARS-CoV-2. All were tested positive and confirmed with a molecular diagnosis of infection with SARS-CoV-2. Importantly, what we did was to match all the cases and controls who all came from the same area for age, ethnicity, sex, infection wave, and vaccination status. And we collected easily accessible samples. So we collected peripheral blood mononuclear cells, plasma, platelet poor plasma, serum, saliva, stool, and nasopharyngeal swabs. And we've done a whole bunch of different studies looking at lots of different things. Many of those samples now are linked in with some amazing projects at the JCVI that you'll hear about later in this symposium. But I just wanted to run you through what we've done here. So we've done a few things, but the most important thing in terms of being informative for our studies has been to do a comprehensive profiling of the plasma proteome. So that is proteins that are circulating in the plasma. And for this purpose, we used OLIG Explore, which was then maxed out at just over 3,000 proteins in the blood. The panels, it's a proximity extension assay, it's targeted, so the panels are divided into two panels each, one for cardiorespiratory associated or cardiometabolic associated diseases, one for neurological diseases, one for cancer, and one for inflammatory profiles. The four panels are shown in the top left. And what you can see, this was our first clue that long COVID was a systemic disease, and there's clearly something quite serious going on, is that most of the proteins in people with long COVID, or a lot of proteins in people with long COVID, are upregulated relative to healthy convalescent individuals. And these samples were collected at least a year after infection. The concatenated data are shown at the bottom, so that's all proteins. And again, the same pattern emerged. But what we didn't find, just by comparing their binary diagnostic status, was we didn't find any proteins that were significantly upregulated in people with long COVID. And this relates most likely to the fact that we were measuring so many that after correction for false discovery, the significance was lost. The proteins we did see that were upregulated using various reactome and geoterms are shown on the top right. And these really were pathways spanning things like apoptosis, cell death, cell proliferation that were upregulated in people with long COVID. So this was a clue that something major was going on. But what we did then to try and refine our analysis was we stratified the analysis, not by a simple binary classification, but by symptoms. And you'll hear more about symptom mapping from Max later in the symposium, which is really informative stuff in terms of designing clinical trials. And what we found using that approach was there was a very specific plasma proteome signature of breathlessness in people with long COVID. So specific proteins that were dysregulated, that were upregulated in breathlessness, and listed there, IDI2, SPRR3, and interestingly including olfactory membrane protein, which is involved in the sense of smell. And when we did the network analysis on this, it's not shown in this slide, but I'll show it in the next slide. We found that there was a network of upregulated molecules centered on CD40, which is expressed on antigen-presenting cells. And that was central in a module that contained chemokine CC ligands like CCL4, CCL3, CCL4, IK-BKG, and IL-18, which incidentally is known to suppress adaptive immune responses. And when we did the gene set enrichment analysis, what we found was that particular proteins associated with cell cycle progression, like RhoA, inflammation, particularly TNF, and intriguingly platelet activation as well, such as platelet-derived growth factor B and thromboxane A2, were specifically upregulated in patients with breathlessness. One of the very important things that we do, in our view, is to validate our data in geographically independent cohorts. So we went on to, there we go, hold on. There we go, to do an identical study in a cohort from Sweden in collaboration with Marcus Buggert at the Karolinska Institute. This was requested by the reviewers, quite rightly, and what we did was essentially to repeat the analysis using the same platform with bridging samples. And as you can see in panel E, just to summarize the key feature of this slide, the network analysis, we discovered exactly the same signature in patients with long COVID in Sweden. So a network centered again around CD40. We also picked up CCL3 again and IK-BKG again and IL-18 again. And we also picked up IRAK1. Gene set enrichment, again, picked up similar pathways that we'd identified in the primary cohort, including MET and the AKT-mTOR pathway. So the other things we've done, just to summarize our view from the periphery, is that we've done a comprehensive analysis of SARS-CoV-2 specific CD4 and CD8 T cells, including using tetramers to phenotype the cells, and you'll hear more about that from Mark Painter later, essentially using T cells as biosensors. And what we found using those was that SARS-CoV-2 CD8 T cells specific for the nucleocapsid protein. So this is not contained in the vaccine, so it reflects viral exposure or exposure to that viral antigen. Those cells upregulated exhaustion markers and inhibitory receptors in people with long COVID. So evidence then of ongoing antigen recognition and persistent antigen. In parallel with that, we found that neutralizing antibodies were actually lower in people with long COVID, at least against the ancestral strain that we tested. And one of the key observations we made was that complement activation or activation of the complement system is very prevalent in people with long COVID. These are all shown in the thumbnails on the right. Complement activation, particularly of the lectin pathway and the alternative pathway, which indicates persistent pathogen activated by pathogens such as viruses and or tissue damage, ongoing tissue damage. So those papers are all published, but the key finding was this disturbance of the plasma proteome. So we figured that the signatures we'd seen in the proteome were reflective of ongoing tissue damage as a mechanistic driver, specifically of breathlessness in people with long COVID. So what we wanted to do then was to actually try and find out where these proteins were coming from and what the mechanistic basis of these peripheral dysregulations was. So we wanted to go in and take lung biopsies and bronchoalveolar lavage from healthy controls and patients, which led us into the lung study, which was only made possible by PolyBio. It would not have been funded otherwise. So to give you an update on sample collection, we have two groups. Again, it's a case control study. We have healthy convalescent individuals with no evidence of disease on bronchoscopic examination for hemoptysis. So these are people who had hemoptysis, they've been signed off healthy, and this is the final check to make sure there's nothing going on. We've currently recruited 20 of those patients, and we've recruited 20 patients with long COVID from our previous cohort who agreed heroically, I have to say, to have bronchoscopies. I'm not sure I'd have been brave enough to do that. So they really are the heroes of this study. And again, we collected all the peripheral samples again, but we also collected bronchoalveolar lavage, fluid and cells, and transbronchial biopsies where we took 4 to 6 cuts per patient. And these are biopsies that go straight through the bronchial wall into the lung tissue. What we're doing is we're doing immune cell lineage analysis and antiviral cellular immunity on the PBMCs as before, in parallel with the lung analyses. On the plasma serum saliva, we're doing full-scale proteomics with Marcelo Freire, again at the JCVI, antiviral humoral immunity with gene tan and Luminex analyses. On the bowel, in collaboration with Marcus Buggert, we're doing immune cell lineage analysis, microbiome analysis with Chris Dupont and single-cell RNA-seq. And on the transbronchial biopsies, this is what I want to update you on today. We're doing single nuclei RNA-seq in collaboration with Chris Dupont's group at the JCVI. We're also linking all these parameters with clinical metadata to really try and figure out which disease processes are affected by the findings that we uncover. This project has been long in the pipeline because we had to get ethical approval to take lung biopsies from patients, and the pipeline required optimization. Each tissue requires optimization. Lung in particular has a lot of RNAs. We had a few potential hiccups along the way, but they've all been ironed out and rescued, particular thanks to Mark Novotny for figuring out how to isolate the nuclei intact via flow cytometry, and Alan Phillips for some fantastic bioinformatics analysis that I'll show you in a minute. This whole project's a collaboration with Chris Dupont and Richard Scheuermann at the JCVI. And we're also doing other things with the samples, including looking at antibody hypervariable regions to address the mechanism of impaired neutralization in people with long COVID in collaboration with Gene Tan. Here's a summary of what we've found so far. The data analysis is ongoing. This is the first emergence of the data, again, prepared by Alan Phillips. So the first thing we did was to essentially map the cells in terms of what types of cells they were from their transcriptomes, and then to map them to the lung atlas. On the x-axis, you can see the cell types we identified, all exactly as you'd expect in a lung: alveolar type epithelial cells, airway epithelial cells, and so on. And the graph just shows the proportions of cells in each sample, in each set of samples, comparing control in gray with the COVID samples in red. And what you can see is that there are no significant differences in cell type proportions when comparing healthy convalescent individuals with long COVID individuals in the lung, which is perhaps not surprising because it's solid tissue, so it has, specific requirements for structure and function. But what you can then do, and again, this is from Alan a couple of days back, is you can then focus in on each cell type, and you can look at nuclear transcriptional differences within assigned cell types, which allows you to subcluster that particular cell type according to its transcriptomic identity. So these plots are UMAP plots. They're simple, well, not simple, they're very complicated, but they allow you to visualize complex high-dimensional data in two dimensions to make it accessible to the human brain. And on the left, you can see this map clearly identifies 11 different clusters based on the transcriptomes of alveolar type epithelial cells. These are clusters that are related transcriptomically. So there are clear subclusters. Just want you to keep an eye on the purple one there on the left, subcluster 4. And then look at the left plot here, which is the UMAP overlaid by disease status. So blue is again the controls, and red is people with long COVID. What you can see for cluster 4 is a clear predominance of people with long COVID have this particular transcriptomic subcluster of alveolar type epithelial cells in the lung. So we're finding distinct subclusters within assigned cell types that associate with disease recovery status via dimensionality reduction, which is hugely exciting in terms of figuring out what's actually going on in the lung. So just to summarize and give you the next steps, No significant differences in cell type proportions in the lungs comparing the two groups, but significant differences in gene expression among identified cell types in the lung, comparing the two groups with a particular long COVID specific or associated cluster in alveolar type epithelial cells. These provide really exciting emerging signatures at the single cell level and a unique opportunity perhaps to unravel pathogenic mechanisms at the initial site of infection with SARS-CoV-2, where we suspect there's huge amounts of damage and residual virus and other mechanisms going on. In terms of what we're doing now, well, Alan Phillips in Chris's lab is doing, we're continuing the analysis for each cellular type to figure out if we can identify other long COVID specific subclusters. We're also collaborating with Chris Ponting at the University of Edinburgh to do analyses of cell state using an approach called STATOR, and that's unique. It's recently developed. It does not rely on proximity and transcriptomic space. Instead, it iteratively reassigns cells based on things like activation status or differentiation status. And we, at the end of the refinement of the data set, we hope to pursue confirmatory studies using spatial transcriptomics. To thank everyone, my group at Cardiff, they're all fantastic. Everyone at the JCVI, it's been an absolute amazing pleasure to collaborate with them. They're all fantastic, particularly Chris for this analysis, Marcus Buggert and everyone in his lab, and everyone in the Karolinska University Hospital who collects the samples, including Sue Ellerman. Huge thanks to PolyBio who made this study possible. They're the best funders we've worked with. Thanks also to Helen Davis for collecting our samples locally in Llandoch Hospital, Balvi Canro for generous donations to the cause, and particularly to all healthy volunteers and the heroic people with long COVID who have volunteered to try and make advances in medical science. And thanks for listening.
Great. Thanks so much, David. Really good. Our next speaker was Peter Broden, but he could not attend last minute. So it will actually now be Michael van Elzakker, and he'll be talking about long COVID and ME/CFS neuroinflammation and neutrophil function. And also Mike is an assistant professor in the Division of Neurotherapeutics at Harvard Medical School.
Thanks, Amy. I appreciate it. So let me just get my slides set up here. So I'm going to talk today a little bit about a neuroinflammation study that we're doing, and as well as some peripheral measures that we think are interesting and important. So we have a lot of studies kind of going on in the lab. I'll just kind of like open these up. And what we've done is the vast majority of the things that we're working on, we work on both long COVID and in ME, people that got sick before 2019, and in some cases, have a lot of overlapping symptoms. We're largely focused in my neuroimaging lab on the convergent symptom pathways, but then also we're trying to work on the potentially divergent drivers or root cause drivers or triggers of those symptoms. I think both of those are sort of valid and valuable options for research, and even can both inform treatment, one at maybe the root cause level and one at the symptom management level. So everything we're doing except for the one way at the bottom is done in both long COVID and ME patients. So what I'll talk about first is from this study here that we put out last year, where we showed evidence of some neuroinflammation in long COVID patients. In this case, we were sort of trying to move quickly, and our control group was healthy controls that had already been scanned in the same scanner, a PET scan, and then we recruited new long COVID patients to do that. And what we do when we look at neuroinflammation is to study glial cells, one type of which is microglia, which are the resident macrophages in the central nervous system. So when glia or microglia come in contact with pathogens, and importantly, it could be any pathogen. It could be something that didn't exist before 2018, like the SARS-CoV-2 virus, or it could be something that's been around a long time. Or when it comes in contact with inflammatory markers like pro-inflammatory cytokines, these cells actually kind of change their morphology. They change shape and then they activate. So when they turn on and activate, that's one of the things that happens in neuroinflammation. They release those pro-inflammatory cytokines, which of course can signal to other neuroimmune cells, and they release excitatory mediators like glutamate, the things that excite neurons. That is likely to throw off the natural signal to noise signaling of brain circuits. And we think that may be important for cognition. And they also release this thing called the translocator protein, which is what we can actually measure using PET scans. So in that first long COVID cohort, we studied 12 individuals with long COVID. These are people that qualified for a modified MECFS international consensus criteria because we were specifically looking for patients with that type of symptom presentation, as opposed to, for example, patients that have a lot of trouble breathing or patients that have lost their sense of smell or something like that. These are patients that were largely not hospitalized and largely got sick before they were able to be vaccinated. So this is sort of the PET scan protocol that we used. We did not get arterial line in these folks, but used a cerebellum baseline. And this is the analysis that we did, basically pretty standard PET analysis. And this is sort of the pattern that we found in those long COVID patients versus the pre-COVID controls. Areas of red are areas where we find increased signal from that translocator protein normalized to the cerebellum. So we extracted the data from each of the participants from the entire brain, and that's over here on the right in red. And you can see the dots that are in open circle, one way up high and one kind of in the middle. Those were the hospitalized patients. So it didn't seem to be the case that that was driving the effect, that those were big outliers. And you can see there's quite a diversity of signal in healthy controls. So these are sort of the brain regions that were activated. We thought that it was largely centered on sort of, subcortical regions, especially places where there's a little bit of a window in the blood-brain barrier, which I'll talk about in a minute. This is what happened when we extracted from each of those different brain regions. And basically, the point here is to show that it didn't seem to be kind of one brain region that was really driving the effect and that sort of thing. So given that we found signal in areas where there's kind of naturally and normally a window in the blood-brain barrier, makes it easy for the brain to sample what's happening in the blood, we thought that maybe the blood might have some clues. So we took the blood that we had collected from our patients right before they got into the scanner and analyzed it and found that there were several mediators associated with vascular problems that correlated with the neuroinflammation signal in these long COVID patients. So here on the left, for example, is SL-selectin, which is what's called an adhesion molecule, which means that it is expressed on the vasculature and pulls immune cells in, right? So immune cells are circulating through blood, and there has to be something that makes it stick to the vasculature and then get pulled into the tissue. And that's what this does. And it correlated quite well with the neuroinflammation signal. A potential argument that that's what we're seeing with the neuroinflammation. And here on the right is fibrinogen, which also correlated. If you know anything about biology, a 0.8 correlation is pretty good in biology, or 0.73. So we really think that that may be important. So we followed up with controls that had COVID, but then reported feeling better. So these are COVID recovered controls as opposed to COVID naive controls. It does seem, now that everybody, basically most of humanity has had COVID, that's an important thing to sort of look at. And I'll just show these. I have just an amazing postdoc named Mario Murakami, who's an MD that works in my lab, and he'll talk a lot more about this, including sort of why we think it might be happening. But this is signal in the brainstem of long COVID patients. And you can see there's a pretty good split, although you can't really just look at a scan and say, there it is, there's the long COVID. So then we also have pre-2018 ME patients, again, using those COVID recovered controls, and found signal up here in what we call the anterior mid-cingulate. again, sort of a split here, as well as signal in the brainstem. That brain region, the anterior mid-cingulate, I'll talk about in a second, because we think it's important for the symptom of brain fog, difficulty concentrating. A lot of conditions have this issue, but we think that there's mechanisms behind it that we could actually understand and maybe even be part of treatment. So, when you are concentrating on something, you use what's called cognitive control. So if you're looking through a crowd for your friend with a red shirt, you will be sort of focusing while ignoring distraction. And that's called stimulus, that's called goal-directed attention. So you sort of see your friend in the crowd. But then let's say a balloon floats up in the crowd. You weren't looking for that, but it will capture your attention and make you lose track of your friend, and you'll sort of focus in on that. That's something that you didn't intend to look at, but it captures your attention. Interestingly, that is a phenomenon that's largely driven by this anterior mid-cingulate cortex, which is this region up here. And it's something that we can actually test in a task. So we use what's called the multi-source interference task to probe this phenomenon. And it is really something that is driven by this anterior mid-cingulate cortex. And importantly, that's a region that is sensitive to inflammation, as I showed you in the pre-COVID ME/CFS patients. So we essentially engaged in this task while they were in the fMRI. That's the same scanner as they were in for the PET scanning. And basically the way that it works is you pick the numeral that is different than the other numerals. So here the answer is 1, here it's 2, here it's 3. Those are easy, but then this is 3, which is harder because it's on the left where you would expect 1 to be. Or here the answer is 2, 1. So when you do that, it actually engages the cognitive control network and you can study it. So this is the task as you would be seeing it in the scanner. And we find that in long COVID patients, it basically takes more effort from the anterior mid-cingulate to engage in this task. Here's long COVID patients, and you can see that it takes more effort in this area right here with the mid-cingulate. And then also in the pre-COVID ME/CFS patients, it also takes more effort. And you can see that is the region that we're talking about. So let me just jump ahead real quick to these neutrophil data. So in the blood of patients that we collect right before they get into the scanner, we put their neutrophils into these little wells where they can swim in and grab bacteria that we put in there. And it's live cells that have never been frozen. And what we found was that patients with ME/CFS showed significant differences from healthy controls. And so far, the long COVID patients have only been trending. So we intend to follow up on that and try to understand how it's related to other symptoms. Okay, I think I'm at time.
Yeah.
Thank you.
Great. That's a lot packed in there, Mike, but really cool data. I know you're going to be publishing a lot of that soon, which will be really exciting. So the next speaker is Luane Spammer. She's a researcher from Ricio Pretorius's lab at Stellenbosch University in South Africa. And she'll be talking about let me grab this, a development of a point-of-care microclot detection and characterization device.
I just want to thank PolyBio for the opportunity, as well as my supervisors at Stellenbosch University, namely Professor Ricio Pretorius and Professor Willem Perald. Okay, so recent work in our research group at Stellenbosch University has identified fibrinolytic microclots in the blood of patients with systemic inflammatory and coagulation disorders, including type 2 diabetes, COVID-19, and long COVID. These microclots are able to block microcapillaries, leading to tissue ischemia and hypoxia. This in turn may cause a variety of the symptoms encountered in the aforementioned disorders. Further studies showed that the size, abundance, and morphology of microclots often correlate with disease severity. The figures show representative micrographs of plasma stained with THT from healthy and long COVID patients. At present, our microclot detection relies on high-end fluorescence microscopy. The Zeiss Axia Observer 7 used in our test was acquired in 2019 for roughly $100,000. Our group has developed a fluorescence-based grading protocol for the microscope, which involves manually going through the sample and grouping microclots according to size and distribution. The scoring system is summarized in the table with a visual representation of the scores seen in the figure. This method is resource intensive and lacks full quantitative robustness. There is a need for a simple, low-cost, point-of-care device capable of detecting and automatically characterizing fibrinolytic microclots, not only using standard THT staining, but also employing specific amyloid detection antibodies to characterize inflammatory content. To address this need, I have developed the Microclot Detection and Characterization Device, or the MDCD for short. In 2003, the World Health Organization published a set of criteria for the ideal diagnostics test that can be used at all levels of the healthcare system in the developing world. These criteria are known by the acronym ASSURED. A point-of-care test should be affordable, sensitive, specific, user-friendly, rapid and robust, equipment-free or simple, and deliverable to end users. The first prototype was designed to achieve sensitivity and specificity comparable to the Zeiss system. Particular emphasis was placed on minimizing component requirements and ensuring a simple design. Furthermore, cost-effectiveness was a key consideration to enable widespread adoption, particularly in resource-limited settings. The Zeiss Axio Observer 7 is an epifluorescence microscope. As seen in the diagram, an excitation light source provides the energy required to excite the fluorophores in the sample. An excitation filter selectively allows light of specific wavelengths from the light source to pass through. A dichroic mirror reflects the excitation light down towards the sample. The objective lens then focuses the excitation light onto the sample and collects the emitted fluorescence. Hereafter, the dichroic mirror transmits the emitted fluorescence upwards towards the detector. The emission filter blocks any residual excitation light and selectively allows only the emitted fluorescence to pass through a tube lens to the detector. A benchtop computer is used to process and display the captured microclot images. The MDCD employs the same fluorescence microscopy principles as the Zeiss. The composition and layout was, however, altered to decrease cost and complexity. Here we have the first MDCD prototype. It consists of 3D printed parts, namely emission filter housing and connector, microscope tube, LED holder, Z-stage top, camera sled, tube sled and bottom, sample holder and base, and the XY stage. The MDCD functional electronic units include a single board computer, CMOS color camera, external peripherals, microcontroller, PCB for Z-stage motor control, and a PCB for LED state and brightness control. To verify that the MDCD can indeed detect fluorescent particles, microbeads were used. They have similar excitation and emission peak wavelengths to that of microclots stained with THT. With a known shape and size, they provide a reliable reference to determine or verify a fluorescent microscope's resolution, focus level, and scale. Their high emission light intensity, stability, and reproducibility also make them ideal for initial prototype testing before moving to human plasma samples. Here we see fluorescent microbeads imaged using the MDCD. The beads used have a nominal diameter of 22 micrometers, which is well within the range of reported microclot sizes. To compare the MDCD with existing fluorescence microscope, characterization tests were performed. A calibration slide with a 70 micrometer dot was imaged. A magnification of approximately 12 and a field of view of 0.54 by 0.40 millimeters were computed. The ability of the MDCD to image fibrinolytic microclots was validated using plasma from confirmed long COVID patients stained with THT as well as PAC1 FITC. A unique clot was found within a long COVID sample stained with THT, enabling a direct comparison between the imaging capabilities of the Zeiss and the MDCD. Here we have another unique clot found in a long COVID sample stained with PAC1 FITC. After confirming that the MDCD can image microclots with comparable sensitivity and specificity to the Zeiss microscope, I wanted to address some important questions. Namely, how can I make the microclot testing protocol more quantitative and less prone to human bias? And how can I automate the microclot characterization process? My proposed solution was the use of microscope grid slides like the one seen in the figure. They were manufactured using photolithography with biocompatible red photoresist. The grid blocks were designed to be the same size as the camera field of view. Additional LEDs were meticulously chosen and configured to allow the user to identify the grid blocks without exciting the fluorescent particles. As seen in the figure on the right, the grids are not visible when the grid-finding LEDs are disabled. This enables the imaging of fluorescent particles without added background noise from the slide. Here we see Microclot images taken with the Zeiss and the MDCD of the same grid block. The lower contrast observed with the Zeiss system results from a combination of the red-colored slides, the available filter set for the Zeiss, and the type of camera used. Thresholding the Zeiss image using their Zen software gives a comparative view of the imaged microclots. In contrast to the Zeiss setup, no background noise is introduced by the red grid slide on the MDCD images, leaving the resolution unaffected. This is due to the type of camera employed by the MDCD. Next, let's look at what the testing procedure for the MDCD and grid slides entail. The first step in the testing procedure with the grid slides is plasma staining using THT. The sample is then loaded in the MDCD. This is followed by light source control, focusing, and grid finding, which are all manual processes. Next, microclot images are captured and processed. Finally, the microclot data is extracted. The graph seen shows the clot area fraction of each grid block of a single long COVID sample. In addition to imaging microclots, the MDCD includes an automated image processing module that quantifies microclots within each grid block. This enables the extraction of fully quantitative data, such as that illustrated in the accompanying graph, representing a significant advancement over the semi-quantitative results obtained using the current microclot testing procedure. Statistical analysis across multiple samples, as well as comparison with the results from the Zeiss testing protocol, remains to be conducted. So, let's do a cost comparison. Today, the Zeiss Axial Observer can be acquired for about $200,000 US. The first MDCD prototype came in at $1,200 US. That is less than 1% of the Zeiss system's cost. This prototype achieves performance comparable to advanced systems at only a fraction of the cost, offering a pathway to democratize microclot test diagnostics in both research and clinical settings. The future vision for the MDCD is sample in, microclot data out, where the MDCD forms a black box that performs the test protocol. The next phase of development will, therefore, look at adjusting the testing procedure to reduce operator dependency. One solution is to automate the system by motorizing the stages and integrating the individual functional units into a unified system. A better solution, and one that we wish to exploit, is the use of lab-on-a-chip technology to isolate the microclots to a predefined area, removing the need for mechanical adjustment of the system during testing. We are also looking at increasing the device's functionality, which includes expanding the MDCD's fluorescence detection capabilities to other wavelengths. The aim is therefore to make the MDCD and testing procedure more user-friendly, rapid, and robust. Thank you.
Amazing. That's really great engineering, Luane. Really cool. All right, our next speaker is Marcus Buggert. He's an assistant professor and docent and group leader at the Karolinska Institute in Sweden. He'll be talking about SARS-CoV-2 gut and lymphoid pathology plus long COVID longitudinal study.
All right, thank you very much. Thank you, Amy, for the presentation or introduction, and thank you very much for inviting me. Today, I'm going to talk about SARS-CoV-2 gut and lymphoid pathology and plus long COVID longitudinal study that we're conducting as well. For the sake of time, I'm mostly going to focus now on the SARS-CoV-2 gut studies that we're doing and the data which has been generated on this cohort. So just a brief background, a little bit about the Karolinska cohort that we have in Sweden. We have almost 400 patients, which we are collecting data from longitudinally up to 10 years now is the plan, where we have disease data, symptoms, lab, clinical examinations, and self-assessment questionnaires that we are conducting, and we're collecting different type of data, which includes some of the parameters here listed on the right side here in the yellow box. And in addition to that, we're also collecting longitudinal serum plasma samples from all participants, and we are also collecting PBNCs on selected subgroups, which which essentially gave rise to a recent paper that we published this summer between our group and David Price group. But what we also are doing is that we are focusing on 3 subgroups, a little bit based on their symptoms, where we also are collecting bronchoscopies, lumbar puncture, and also do colonoscopies from these individuals. And this last part here is what I'm going to focus on today, specifically for this talk and the data which we have generated so far. I don't think I have to tell the audience here the reason for why gut is an interesting site. This is probably the first paper which sort of showed it from Michelle Nussenzweig's lab with Christian Gabler as the first author, which essentially could see in asymptomatic people, long time after SARS-CoV-2 should have been cleared, they could see essentially the SARS-CoV-2 nucleocapsid stains were present in many donors in these individuals, as depicted here by these images, partly due to the fact that ACE2 most likely is very highly expressed within the gut. Christian is actually part of our consortium as well, and he's conducting ddPCR measurements for our reservoirs in the samples that I'm going to talk about soon. So particularly for this cohort, like I said before, what we have decided to do is that we have different individuals, they have specific type of symptoms. All the individuals that we have in our cohorts have severe long COVID symptoms. It includes brain fog and other type of symptoms. But some of the individuals, about 20 to 25%, has these persistent GI symptoms for more than 4 years. And these are IBS-like symptoms that includes abdominal pains, bloating, cramping, diarrheas. But importantly, when the colonoscopies have conducted analysis on these individuals, they are not able to find any microscopical findings so that they can essentially link it to inflammatory bowel diseases like UC, etc. But as I'm sure you're going to see soon, we can definitely still see specific patterns coming up in these individuals. So essentially what we decided to do for this study was to conduct high-end analysis. I will show soon which kind of analysis on 10 long COVID patients and 7 healthy controls. They're all recruited from Sweden, from the same site. And we are then taking biopsies, multiple biopsies from the sigmoid, from cecum and ileum to get a full representation, as good as possible at least, of different sides of the GI tract. The type of analysis that we're conducting is VGJ sequencing that we can conduct through single-cell sequencing. We're conducting 16S sequencing, in other words, microbiome analysis, FIPSeq, ddPCR of our reservoirs that I mentioned recently, and we also conduct confocal imaging. Specifically for this talk, I'm going to focus on the single-cell data which we have been generating on our donors. So far, what we've been able to do is to generate on fresh biopsies. That's important. It's probably a big difference also from other data sets which is out there. Fresh biopsies with very deep sequencing, so over 50,000 reads per cell on over 600,000 cells. And this includes cells which is not only immune cells, it will include epithelial cells. We have mast cells, fibroblasts, T cells, plasma cells, and B cells including as well. And from all these individuals, what we can do is to conduct different type of analysis. We can divide them up into, and this is just to sort of illustrate that we can combine the data to harmonize it, but we can then basically go in and we can determine what some of the differences are between these different sites when we look at the data later. I'm not going to go in on that, what the site-specific differences are. It's interesting. There are site-specific differences. You can almost actually distinguish that already here in some of the on the top right side here in cecum, that you have more, for instance, plasma cells there. I won't go in exactly on that, but it has some important meanings a little bit later on, and I think it will most likely when we do more analysis on this in the future. But if we then focus on the immune cells, so this is how the UMAP looks for the immune cells. That's a little bit more than 300,000 300,000 immune cells that we have in here. We have T cells on the left side, CD4s and CD8s here in different clusters, B cells, plasma cells, and we also have different type of ILCs, mast cells, and also macrophages here in the middle. And in general, if one just looks broadly speaking, over the different clusters, it's not so that there's major changes that are happening between healthy controls and the and our long COVID patients, which we here called the CLIM patients, because that's what the name of the cohort is. But essentially, you can still see that there are some features, such as increased TRM populations, increased plasma cells. I will come back a little bit later to in the talk, that is of interest for us as well. But there are also some differences, that there are some lower levels of naive B cells and some certain memory B cells as well. So there are some shifts, but in general, it looks to us that the major compartments are still there, and it's not something that is majorly different between them. What is different, though, is that when you start to dissect every single cluster here, we definitely see differences between healthy controls and long COVID patients. And one of the major features that we do see is that interferon-stimulated genes, so ISGs, they're broadly upregulated in long COVID. And ISG expression is primarily elevated in adaptive immune cells. And for that, for us, that sort of suggests that there might be an upstream or extrinsic signal that drive these features. If it would be viruses driving it, we would probably see it also within these compartments here in the middle. There could be still a virus component driving this, but I think some of these features that we see, given the fact you see them adaptive cells, it rather for us implicates that there is probably some upstream and extrinsic signaling driving these features. Another interesting feature that was part of the grant also that we put together was to understand if there's reduced killing capacity that we can see. We can do more sophisticated analysis than I'm showing here, but in general, it doesn't look like there's any reduced killing expression patterns for T cells in general within the gut. If anything, it looks that effector functions are moderately elevated across long COVID donors, and there's some strong upregulations observed for several chemokines and cytokines, which include CCF5 and actually TNF, which is of importance. And this is just showing that for TNF, that across different T cell subsets, but even within germinal center B cells, we can see enrichment of TNF signaling at the baseline is elevated across the multiple T cell subsets, and also, like I said, germinal B cells. But in addition to that, and this is a striking feature, which is the opposite, is actually that TGF-beta signaling is reduced in long COVID. And the reason for why that's interesting and important is because TGF-beta is important to essentially create immune tolerance in the entire gastrointestinal tract. But what this data at least is suggesting is that there's multiple subsets that are showing reduced levels of TGF-beta expression in general. And just to zoom in on the cell type that is best associated with TGF-beta expression, and is also this year, particularly for someone from Karolinska, a cell type that we focus a bit on more this year because of the Nobel Prize, is that we can see that there is reduced baseline expression of TGF-beta across multiple Treg subsets. And you can sort of see it here in this UMAP quite clearly, and you can also depict it by the violin plots on the right side as well. It doesn't only end there, and like I said, I'm not having time to show all of this, but essentially what we see in addition to that as well is that there's a reduced expression of Proal protein convertases that are important to maintain immune tolerance, like furin, for instance, that is usually expressed within a peripheral Treg population here, 0 and 15 specifically here, that you have less of in long COVID. And you have nuances and distinctions between CD40 ligand that is slightly higher within long COVID as well in comparison to healthy controls. To some degree, if that is meaningful or not is hard to say. I mean, what we do here is mostly correlative analysis, but what we can see as well when it comes to B cells is that in addition to the fact that these individuals have actually higher levels of plasma cells, we do see over the entire span of B cells, there seems to be sustained B cell activation, interferon signaling, like I talked about before, and persistent differentiation towards antibody-secreting phenotypes rather than a return to homeostasis. So this is, like I said, just a summary of some of the analysis that we have done so far. This is, there's way more, but I don't have time to present it during these 10 minutes. But the summary here is that interferon signaling and ISGs are broadly upregulated, especially in adaptive immune cells, including B and T cells, that indicates that there's a chronic interferon exposure or extrinsic stimulation that is taking place in these donors. But what we also find is that effector and specifically cytokine programs, such as TNF expression, seems to be elevated across multiple T cell subsets. That is usually linked in different inflammatory bowel diseases to sustain inflammatory activity. We see on the other side reduced TGF-beta signaling, which is reduced across T and B cells, including Tregs, which highlights that there could be impaired tissue repair and immune regulation at these sites. There's a regulatory imbalance between Treg and TFH cells to show altered expression, also these other regulatory components like furin and activation ligands. And we can see that the B cell phenotypes, such as decreased BLIMP1 expression, but elevated CD38 and TNF pathway genes in plasma cells, which sort of points to that there's most likely short-lived inflammatory plasmablast bias rather than stable long-lived plasmablasts that are occupying this niche within these donors. So these data essentially points towards persistent immune activation and an incomplete resolution of mucosal immunity, potentially maintained by local interferon TNF signaling loops. Some of the future aspects of what we'll do now from here is to expand the molecular analysis to conduct GI, so site-specific analysis between the different sites within GI, compare these findings to peripheral blood to see if we can find out. We do ligand receptor mapping to non-immune cells to see where this network sustaining the interferon TNF signaling comes from. We'll integrate some AI-based tools for that most likely in the future. We'll conduct functional assays to validate the interferon TNF sensitivity, assess plasma cell function, antibody reactivity through autoantigen and microbiome cross-reactivity testing. And we'll also conduct broader systems perspective by integrating microbiome and ddPCR viral persistence data to link immune activation with possible reservoirs, where the long-term goal is to define very persistent immune activity that we see, particularly for interferons and TNF activation represents itself, sustained in mucosal inflammatory circuit and how it might be therapeutically reversed. So that's it from me. Particularly, I would like to thank Waitao Sheng in my lab, who has conducted most of these studies with help of other members in the lab. Also thank to clinicians and other researchers at Karolinska University Hospital for collecting these samples, David Price, Christian Gabler, patient health volunteers, and our funders as well. Thank you very much.
Thanks, Marcus. That's great data. It'll be really interesting when you do the ddPCR for SARS-CoV-2 as well to see if there's correlation there. So great. Cool. Our next speaker is Michela Locci. She is an associate professor of microbiology at the Perelman School of Medicine at the University of Pennsylvania, and she'll be talking about lymph node immune responses in long COVID.
Okay, my pleasure to be here and give you an update on this project where we are trying to better understand the biology of B cells in the lymph node that are reactive against SARS-CoV-2 during long COVID and as well as in convalescent. And just for the people in the audience who are not familiar with these, so my lab studies germinal centers, which are these really complicated reactions that take place in lymph nodes. And these reactions are incredibly important during viral infection, also during vaccination. And the goal of this reaction really is to educate your B cells and to become high affinity for the pathogen in such a way that when they're selected, they give rise to plasma cells that secrete high affinity antibodies, as well as high affinity memory B cells that when re-encounter the pathogen can give rise quickly to a burst of plasma cells producing antibodies against the pathogen. So it's extremely important in the setting of a viral infection to mount robust germinal center responses. The difficulty in studying these responses is that they take place in lymph nodes, and so if you work with humans and you have access only to blood, you can really study germinal centers there. And so we have to be creative and find a way to access the lymph node directly. The approach we have used in the past is using this fine needle aspirate, in which you select the lymph node of interest, you visualize it with an ultrasound, and then you enter with a fine needle. As you can see in this video, there is a fine needle literally poking the cortex of the lymph node, which is the site where the lymphocytes are, and you collect enough cells for downstream analysis. It's not enough cells, but for the most part, we can get very valuable information. And that's what we did, for instance, in the past when we study germinal center responses against COVID-19 vaccination in people. And so in that case, we were probing the axillary node. Of course, this would not be ideal for studying responses triggered by SARS-CoV-2 infection because this is a respiratory virus, and so we thought a little bit about what lymph node to target for that particular study, and we decided to try and collect by FNAs samples from cervical lymph nodes, which drain the oropharynx. And so we started a couple of years ago now this study. First in collaboration with Ben Abramov at Penn, who runs the Long COVID clinic, and then more recently in collaboration with Michael Peluso, UCSF. And the idea is that we are collecting either healthy convalescent that get COVID-19 and then recover, as well as individual affected by long COVID. And we basically collect the blood and fine needle aspirate of the cervical lymph nodes upon reinfection, and ultimately stratify the sample into sample collected between 1 and 6 months post-infection, 6 and 14 months post-infection, or later on, which for the sake of today's presentation, I will not talk about. I removed actually for sake of time the slide describing the characteristic of the individuals in our cohort, but for the most part, it's individuals with a neurological manifestation. All right, so we did in all samples that we collected fresh flow cytometry analysis, specifically focusing on the SARS-CoV-2 specific GCB cells. And this is the way we define antigen-specific cells using a combination of fluorescently labeled tetrameric probes. And what we found was that early, within 6 months post-infection, germinal center B cells against SARS-CoV-2 were detectable in most healthy convalescent, with a few exceptions. Whereas in individuals with long COVID, for the most part, these responses were severely disrupted. So you can see that only very few individuals had detectable germinal center responses. And then the situation, it's interesting because later on, at a time where the responses are, for the most part, clear in healthy individuals because the virus is not there anymore, in a fraction of the long COVID individuals, you actually see that these germinal center become detectable later on. So the overall kinetic is completely different. What does this mean in terms of the quality of the antibodies against SARS-CoV-2? So if you look at the binding antibodies, these, for instance, are antibody binding S1 of SARS-CoV-2. At the time point, early post-infection, the overall levels of these antibodies are relatively normal. But then a difference that is significant is that in the long COVID group, these antibodies wane faster. And this is consistent with the fact that there are disrupted germinal center, which are usually the source of these long-lasting antibodies. Neutralizing antibodies were also significantly reduced. And interestingly, we found in the healthy convalescent a very nice correlation between the frequency of SARS-CoV-2 specific GCB cells and the amount of neutralizing antibodies, suggesting that in healthy convalescent, most of these cells of these neutralizing antibody have a germinal center origin. But this does not seem to be the case in the long COVID, where there is absolutely no correlation. So more recently, we have done site-seq data. We have generated site-seq data on multiple type of samples. And what I'm going to show you today is site-seq data from sorted IgD negative B cells from FNA. And so IgD negative is basically a way to enrich for non-naive B cells. We have run on these transcriptome, BCR sequencing, and also we have tetrameric probe, which are barcoded, and so we can essentially infer by binding of this probe the cells that are SARS-CoV-2 specific. So this is basically how the cells look like. You see there is a very large cluster of memory B cells, some naive contaminant from the sorting, a nice population of germinal center B cells, and a small population of plasmablasts, which are the precursors of plasma cells. And first glance, this data largely confirm what we found by flow cytometry. So you can see that total GCB cells are reduced in long COVID at T1 in comparison to the convalescent, whereas they seem to be larger, more abundant at later time point post-infection. Overall, it was interesting to see that there is also a significant reduction in clonal expansion in long COVID, which is particularly evident at T1. As well as a reduction in the number of mutations, somatic permutation, which is an indication of poor affinity maturation in long COVID. Then also using these probe that are barcoded, as I mentioned, we try to infer on the antigen-specific cells, and these are also largely recapitulate the data that we found by flow cytometry. But even though I should say that we obtain a very small number of cells with this approach, and so we are currently in the process of generating a second data set where we sort by flow cytometry on antigen-specific cells and to perform a subsequent transcriptomic analysis in order to learn more about the alteration in the SARS-CoV-2 specific GCB cells from the lymph node. What I can show you though is some more data on the memory B cells, and so there are different clusters of memory B cells, and there are alteration in these, and in particular it appears that there is an expansion of memory B cells which are IgM positive, and so these would be cells that come before the germinal center, or the so-called extrafollicular B cell responses. And these differences are particularly evident at T1. Going deeper into these differences, what was interesting to see was that the pathways that are upregulated in convalescent are pathways associated with mitochondrial metabolism, such as oxidative phosphorylation, which is considered to be important for the selection of high-affinity B cells in the germinal center. And then, if you look by contrast at the pathways that are upregulated in long COVID, there is a pretty massive upregulation of many pro-inflammatory pathways, including interferon responses that were pointed out by Marcus in the previous presentation. So we also see that some individuals, but not all, have this massive ISG signature in the memory B cells. Moreover, we could specifically look at the somatic permutation of the memory B cells that are SARS-CoV-2 specific, and we found that just at T1, but not at T2, there is a significant reduction in somatic permutation. So these cells really seem to have like a major impairment in their capacity to undergo affinity maturation. So in conclusion, what I show you today is that we were able to find, by directly looking in tissue, major alteration of germinal center responses against SARS-CoV-2 in long COVID in comparison to convalescent, linked to an overall reduction in both clonal expansion and somatic permutation, and also connected to alteration in the metabolism, but also in a particular subset of the memory B cells, and reduce somatic permutation in the memory B cells. And I would like to point out that these differences are very evident within the first 6 months post-infection. So really looking at the particular time post-infection matters to find differences that are more clear. Lastly, I would like to acknowledge all the people involved in this study. The study was started by Anna Sharp, who left the lab then, and was continued by Joy Chu and Mohammed, who did the analysis of the site-seq data. And I already mentioned Ben Abramov and Michael Peluso for helping us with obtaining these very precious samples. And excitingly now, we are starting to collaborate with John Werry's lab to learn more about the T cells and to look at the T cells, the CD4 T cells, as well as the CD8 that are in the FNA samples and that are specific for SARS-CoV-2 and EBV. And hopefully at the next meeting, we will have exciting data on this topic. And of course, I want to thank PolyBio for the funding. So I'm going to stop here.
Great. Thanks so much, Michela. Very interesting. Okay. The next speaker is Sara Cherry. She's a professor of pathology and laboratory medicine at the Proman School of Medicine at the University of Pennsylvania, and she'll be talking about long COVID and tissue reservoirs.
Thank you so much for having me today, and I'm really excited to give you an update on the work that we're trying to do to understand the relationship between long COVID and tissue reservoirs, and to define whether or not we can consider that in our biomarker utilization, and whether or not we can use this information to come up with additional treatment strategies for this disease. So as we all know, SARS-CoV-2 initially infects people in the respiratory tract, where you get a peak viral load a few days after infection. And then for most individuals, there's clearance of the infection. But of course, in this group, I don't need to bring that up in any real way, except to say that for a subset of individuals, there's symptoms post the acute phase of the infection that can have consequences in many different tissue types and has a lot of complexity. And there's a lot of effort to try to understand what's driving the symptomology for these individuals post the acute phase of the infection. And many people, including most of the people on this call today, understand that there's been huge amounts of evidence to suggest that the infection is not restricted to the respiratory tract, at least in some individuals, and that's been observed through shedding in sewage, biopsies, and autopsy samples, where you can see viral antigen in other tissues, suggesting that there may be a reservoir. And some of our own work from autopsy patients during the pandemic, we were able to look in a variety of tissues, and we also observed, as many others did, that you could detect viral RNA in tissues throughout the body, but for a large number of individuals, that was in the GI tract. And similar to the studies that have shown that during acute infection, people are shedding in sewage, we've been looking in long COVID patients with the clinic of Ben Abramoff here at Penn, where we've been looking at the stool from individuals that are either healthy or come to the clinic with long COVID. And we've observed a distribution of viral antigen detection in the stool, where patients that report long COVID are more likely to show significant levels of viral RNA in their stool. So this is really striking and I think important in considering the fact that we don't have gold standard biomarkers for long COVID. And so the question is, can we use stool as a way to read out, at least for a subset of patients, whether or not they have viral antigen in their bodies, which patients have this, and can we use this not only to as a biomarker for therapeutic treatments, but to consider whether or not we might treat patients with SARS-CoV-2 RNA in their stool differently, perhaps, than patients that don't. This also leads to another really important question, which is, where is the virus antigen coming from? Is it coming from the GI tract? Is it coming from other tissues? In which cell types might it be coming from? And again, can we consider this in terms of how we might treat individuals that have SARS-CoV-2 RNA in their stool? So this has led us to try to model tissue infections and to ask how modeling of these tissue infections may impact our understanding both of local infection of this potential reservoir, as well as how it may be interfacing with the systemic immune system, as well as other tissues that have defects in long COVID. And so we and others have used these air-liquid interface models as a 3D model of the human respiratory tract, and we can infect these, and we can show that the infection kinetics resemble that of acute infection in humans, where we have peak viral loads at around day 3, and then over time, the viral load in these models go down, and that's accompanied by destruction of the monolayer, as we observe in acute infection in humans. Furthermore, we can ask if we can treat these air-liquid interface models with the therapeutics that are used clinically, such as Paxlovid or molnupiravir, and we can show that we can get very striking reduction in viral infection in this model using these approved therapeutics, at least for acute infection. And so again, we're now trying to ask questions about whether or not we can model other potential tissue reservoirs. And so we have a 3D model of the GI tract, where again, we can infect this model, but interestingly, the kinetics of infection are completely different, where the infection of the lung model or the respiratory model has high levels of infection that go down over time. What we see in the GI model is that it takes longer for there to get peak viral infection, and it goes out for more than 21 days. So this converts the infection from an acute lytic infection to a more chronic infection, which may resemble that of a reservoir. Furthermore, we asked if the same drugs that are active in the air-liquid interface cultures are also active in these 3D gut models. And no, they're not all active. Where we see activity of, for example, Paxlovid, we do not see activity of molnupiravir, suggesting that different direct-acting antivirals may have different activities in different tissues, which we should consider in cases where we think that the disease may be driven by tissue-resident reservoirs. Furthermore, we were interested in comparing the host response to infection of these two different tissues. So this is RNA-seq data infecting either these air-liquid interface model or this GI model, and asking what are the gene expression changes. And what we can see is that there's a subset of genes that are induced in both the lung and the GI model, where there's a larger number of genes that are induced only in the GI model, suggesting that there's fundamental differences in the host cellular response to infection with SARS-CoV-2. If we look more carefully at that, again, if we take the genes that are induced in the lung, this is all of them, then most of them that are expressed in the GI tract are also induced. And indeed, these are exactly what you'd expect, antiviral signaling pathways and ISGs. However, if we look the other way around and what's happening in this potential reservoir, what you can see is if we look at the genes that are induced in the GI tract, they're not induced in the lung. There's a completely different gene expression program, suggesting again that there's fundamental differences on how the cells are responding. And if we look at what pathways are being induced now in the GI tract, what we can see there's many more inflammatory pathways, including TNF signaling, for example, and other NF-kappa B-dependent inflammatory signaling pathways. So this, I think, is telling us something fundamental about what may be happening if indeed patients have a GI reservoir during infection. And so we're looking more carefully at what the cytokines and chemokines are that are being secreted by this GI model and asking about what cell types it may be reading out on. This is just a subset of the cytokines and chemokines that we've identified. And we're really now in a position to ask how the secretion of these factors may be impacting the systemic immune system, and how that may also be signaling to other tissues, such as endothelial cells, which have also been shown to be dysregulated during long COVID. So the questions we're now asking is how these secreted factors impact these systemic responses. Are they leading to these past symptoms? Can we directly test how these secreted factors are altering immune function? So now we're taking the supernatants off of these models and adding them to either peripheral cells, and we're developing tonsil organoid models to ask more fundamental questions about this crosstalk and whether or not we can use this information to treat in terms of blocking or using immunomodulators that will prevent these tissues from talking and activating these cells inappropriately, to suggest both the most direct-acting antivirals that might block the infection in the reservoir, as well as to try to define which anti-inflammatories may be most active to block infection in trans or dysregulation in trans. Furthermore, now that we're developing these 3D models of infection in other tissues, we're hoping to expand it into other potential tissue reservoirs, including the CNS and others that have been defined through a variety of different contexts. This is just the beginning of our ability to understand what may happen if infection is being established in these other tissues. So with that, I'd just like to thank all the individuals that have contributed to this, our collaborators, including Mayan Levy, Christoph Dias, and Ben Abernoff, who has the long COVID clinic here at Penn, as well as Benjamin Gabriel and Tamana in my lab, who have done most of these infections. Thanks.
Great. Thank you, Sarah. Really cool. All right. Our next speaker is Christopher DuPont. He's a professor in the Genomic Medicine, Environmental Sustainability, and Synthetic Biology groups at the J. Craig Venner Institute, and he'll be giving updates on the tissue analysis pipeline.
Perfect. All right. So as Amy mentioned, I want to provide an update on the tissue analysis pipeline. This is a group of, I believe, something like 8 small interconnected grants, a huge team. There's probably people I'm missing off of this team. The big impetus behind this initiative is that we're trying to move beyond blood or even just saliva as the accessed human specimens that can be used for tracking either microbiome disruption or human cell biology disruption. And the tissue types that we're diving into include lung, intestinal lining, endometrial tissue, skin, and nerve and ligament. Now, the overall approach, and this is why it's such a big team, really has a lot of multi-omics in parallel. So we get tissue samples directly from the surgeons, often by providing them with sampling gear that allows them to preserve it directly on the surgical floor. From that, we do unbiased shotgun metagenomics and metatranscriptomics. This can recover the genomes of any organism, from viruses to eukaryotes. We use single nuclei RNA sequencing. This is particularly important for tissue samples. This is to get at what human cell types are within a specific tissue, but also what gene expression profiles. So when we go to, say, endometriosis versus non-endometriosis, we can ask, are there specific cell types that are enriched? Or of cell types that are in both conditions, is there an altered transcriptional profile? And now, well, not for all of the projects, there is also a very large spatial transcriptomics approach. After that, we really dive in with machine learning and discriminative AI methods to identify the features within these huge data sets, often hundreds of thousands to millions of individual features, such as the relative abundance of a given taxa, and identify individual features or sets of interacting features, like I'm showing down here on the lower right, that are diagnostic for that condition. And by doing a discriminative AI approach, we can actually quantify that accuracy. Some kind of high-level highlights. You're going to hear about this later for Beverly Peng. She's going to discuss this some more. We've developed the methods for getting single nuclei out of endometrial tissue. We've put together a cell atlas for endometriosis versus control tissue from these samples. And most recent result is we found that E-stromal matrix metalloproteases are enriched in endometriosis relative to control samples. Those of you who have a background in cancer biology, this should be ringing an alarm bell. This is involved in a lot of tumor progression, ovarian, skin, et cetera. So this is somewhat of a hallmark of potential cancer progression. You've heard some of the data about the lung single nuclei RNA-seq data set from David. One thing that I wanted to pop out there is that we are also very sharing with our methods. All of our informatics code goes into GitHub and is open access. All of our protocols, Mark Novotny is currently writing up this protocol to drop into protocols.io, which is also an open access protocol. One nice thing about this is that we found the $30,000 box that's supposed to facilitate getting nuclei didn't work on lung biopsies. And Mark, who really led up the development of single nuclei RNA sequencing in general, came up with an extremely low-cost method. About $300 worth of laboratory equipment is needed to get large yields of single nuclei out of lung biopsies. And this will be deposited in protocols.io shortly. One of my specialties, and what I'm going to spend a lot of the rest of the talk on, is some of the discovery pipelines we have for the human microbiome and human virome. Now, you hear a lot about the microbiome, particularly with regards to gut health, but the reality is that pretty much every external and internal epithelial site within our body is colonized by microbes. And these run the gamut from viruses all the way up to eukaryotes. What is very clear, though, is that it is very niche defined. Your oral microbiome and your skin microbiome have a little bit of overlap, but not a ton. They're both very, very different from, say, your gut microbiome. And this is a combination of environmental factors, but also the human immune system is essentially dictating which microbes can settle into those environments. However, those systems can become dysbiotic as well. One thing that when you see a lot of microbiome work, most of the time you'll see lots of bar charts. Essentially, they are showing the data as relative abundances of these microbes. Because Amy was generous enough to fund us on multiple of these different projects, we've also taken an approach inspired by the ocular microbiome projects funded by NIH to quantify the absolute abundance of bacteria, viruses, and archaea within these different tissue types, and really start to get an idea of what abundances of microbes are there. And maybe in some cases, like these two endometriosis samples, this can actually be a better indicator of dysbiosis than other metrics, such as the abundance of different individual taxa. We also, when we generate shotgun metagenomic or metatranscriptomic data, we do not take a read-based analysis approach solely. We actually actively specialize at JCBI. We develop the methods to assemble metagenomes or genomes out of metagenomes. That is, we can assemble the metagenome and then using machine learning, bind together the pieces that belong together and pull that genome out. And in one of those studies, specifically the ligament and nerve study for patients undergoing CCI surgery, We had 2 samples where we recovered complete Burgundomonas genomes out of those tissues. Literally, we stitched together entire genomes out of them. Now, Burgundomonas is a known reagent contaminant, so we have gone on a quest of validation verification. We have done separate extractions on these tissue samples with extraction blanks and run them through ddPCR. The same 2 samples where we recovered genomes, we had appreciable amounts of Burgundomonas rec. So we have a secondary confirmation done. Working with Marcelo Freire, we are developing a fish assay to dive into these same tissue samples and scan to see if they're there. Moving forward, we wouldn't want to do shotgun metagenomic sequencing, but those 2 validation assays are low-cost, high-throughput assays, that is FISH and the ddPCR, that then can be applied to hundreds of samples that are already archived, as well as samples going forward in a very inexpensive fashion. This is just some of the power that we can get out of this. There actually is Burgundomonas has been recovered from human samples previously. It's also found in other organisms' microbiomes. And I mentioned some of these ongoing experiments. I also want to acknowledge PolyBio for funding, as well as the Cohen Foundation, National Institutes of Health, and the others. And thank you for your time.
Great. Thank you, Chris. Great to see the pipeline coming along. Okay, the next speaker is Margaine Bomsel. She's a senior researcher in mucosal entry of HIV and mucosal immunity at the Institut Cochin in France. And she'll be talking about SARS-CoV-2 persistence and impact on long COVID megakaryocytes and platelets.
Good evening. So, okay. So in the lab, we focus on the viral persistence as a potential master regulator of long COVID symptoms. Indeed, viral persistence mechanism are poorly understood. And for instance, a viral spike has been found by several authors in the circulation, but its origin remains unclear. Also, as it has been shown just before, viral components have been detected in many tissues of long COVID patients. But question remains of whether this component correspond to actual viruses, if these viruses are produced, if these viruses are infectious, or even if with these viruses, infection can propagate. We investigated in the lab this question using megakaryocytes and platelets as a model. So our reason to focus on megakaryocyte is because during acute COVID, we have shown in the lab that megakaryocytes were infected with SARS-CoV-2 in the bone marrow and in the lung, and that this infected megakaryocyte could produce during thrombopoiesis platelets containing infectious viruses. And this infected megakaryocytes are mainly found, were mainly found in non-survivor, but also in a small proportion of COVID survivors that, when re-questioned recently, were mentioned that they reported that they developed symptoms compatible with long COVID. So, for this study, together with Dominique Salmon at Institut Fournier in Paris, we established a long COVID cohort named Megalong, for which we collected blood from long COVID patients that was compared to COVID recovered individuals. So briefly, so this cohort is mainly female. Most of the individuals were vaccinated and have undergone several reinfections. Importantly, the median sampling time was 16 months after acute COVID onset, And this patient reported quite high, so roughly more than 6 type of symptoms on 16, and which included mainly asthenia and cognitive and neurological dysfunction, indicating that our cohort study severe long COVID. So we first looked at megakaryocyte and showed by confocal microscopy after immunolabeling and in situ hybridization that this megakaryocyte contain spike, but also viral RNA, both sense and double-strand RNA, suggesting viral replication, but this was found only in sample from long COVID, but not from COVID recovered individuals. We could quantify that using a fish flow technique, which reveals that roughly 30% of circulating megakaryocytes were positive for viral RNA and spike. And this is associated to a higher content in circulating megakaryocyte, suggesting emergency megakaryopoiesis, as we had already shown for acute COVID. So this indicates that megakaryocyte may form SARS-CoV-2 reservoir in long COVID patient, and that megakaryocyte may produce platelets sheltering infectious SARS-CoV-2. So we then look at platelets, also by the same morphological technique, and show that they contain spike, always located inside the platelets, but also viral RNA, again, both positive strand and replicative negative strand of the RNA. Fish flow help us to quantify this infected platelets, and the blood of this long COVID patient contain roughly 0.8. The platelet in the blood of this patient was roughly 0.8%, which correspond to roughly 10 to the 9 infected platelets per liter of blood, which is quite significant. So we then ask the important question of whether platelets, whether this viral component corresponded to infectious viruses located inside platelets. Therefore, we took platelets, activate them to allow its content to be released, and this release was tested for infectiousness on reporter cells. And as you can see here, most of the long COVID patient sample were infectious for this reporter cell, and we can estimate that 1 million of platelets contain 30 infectious infectious viral particle, which is roughly 10 million of infected platelets per liter. So we then ask whether the platelets could also transfer not only the virus, not only to reporter cells, but also to primary monocyte-derived macrophages. And as you can see here, it is indeed the case, in contrast to platelets from COVID-recovered individuals. And next, we also ask whether these infected macrophages were able to produce viruses and initiate a second round of infectiosity. And therefore, the supernatant of these infected macrophages were tested for infectiousness on reporter cells. And indeed, when macrophages were infected by platelets from long COVID individuals, they become infected and produce infectious viruses, in contrast to platelets from COVID-recovered individuals. We then focus on the capacity of these platelets to form aggregates, which I think are a bit different to the one described earlier today. So we just focus on platelets and use microscopy to characterize these aggregates. And as you can see here, platelets from long COVID individuals form larger aggregates than those from COVID-recovered individuals. And more recently, we also ask whether these COVID aggregates contain spike, and it is indeed the case, as you can see here, using imaging cytometry, image stream, and this technique shows that indeed the aggregates made of platelets contain spike. Spike is detected either in almost all platelets in the aggregate, but often, but sometimes not in all these platelets from the aggregate, and sometimes just one plate or one or two platelets from the aggregate contain spike, in contrast to COVID-recovered individuals. We then address the metabolism of these platelets and study the ATP production by these platelets using the Huot technology. And as you can see, the analysis of this data indicated that there is an increase in proton click, so indicative of defective mitochondria. And indeed, concerning ATP production, the data shows that these platelets from long COVID used glycolysis to produce ATP at the cost of mitochondrial produced ATP. And interestingly, this higher glycolytic ATP correlates with platelets infection we had shown previously. We then looked at the platelet transcriptome by RNA-seq, as you can see here. And the initial analysis shows that the main canonical pathway differentially expressed split in a very limited number of functions, such as mitochondria ATP synthesis dysfunction, mitophagy and microautophagy, emergency megakaryopoiesis, platelets and serotonin pathway dysfunction, membrane trafficking, and MHC class 1 presentation. Finally, I want to mention were results on the spike found in the plasma of this long COVID individual. We have now studied more than 140 patients for that. And as you can see here, roughly 20% of these patients had quite significantly high level of spike in the plasma, whereas only 30% were totally negative. Importantly, the spike in the plasma is totally independent of vaccination. It doesn't come from the spike that could remain from the vaccination, also that is quite unlikely. And also the concentration of spike found in the plasma correlate both with the infection score of platelets and the glycolytic ATP production by these platelets. So in conclusion, this study shows that megakaryocytes and platelets are infected with SARS-CoV-2 in long COVID patients. Megakaryocytes could form a viral reservoir. Platelets, this reservoir produce platelets which are can spread infection. And finally, platelets are dysfunctional. They can form microclots that could contribute to cardiovascular signs found in long COVID patients, as well as immunothrombosis. And the glycolytic ATP production, favored glycolytic ATP production could lead to contribute to asthenia. So I think I will stop here. I want to acknowledge the people in my lab, especially Stefan Hay, who performed most of this study, Gabriela Sarango, and the bioinformatician Zicheng Zhu, who just joined the team, as well as Andreas Cotigny-Calamart and Bo-Xin Huang. This study is performed together with Dominique Salmon, who built this cohort of patients and a set of clinicians that contribute to this study. And of course, we thank all participants, funding, especially PolyBio, which is quite instrumental in this study. So thank you very much.
Thank you, Morgane. It's incredible data. It really is. Okay, our next speaker is Isan Safik. She's an assistant professor in the Department of Immunobiology at Yale University School of Medicine, and she'll be talking about a humanized mouse model of SARS-CoV-2 RNA persistence.
As Amy mentioned, we've developed a humanized mouse model of SARS-CoV-2 RNA persistence, and the way we have been studying SARS-CoV-2 infection is infecting our humanized mouse model, which recapitulates the human immune response that we see in patients, including persisting lung pathology. In this mouse model, the infiltrates come early as 2 days post-infection, but as seen in patients, they persist, and about 28 days post-infection, converts into fibrosis. And we do see the 3 histopathological phases of the disease, both the exudative phase with the early infiltration, the organizing phase, and the fibrotic phase. So more recently, we've been using these mice to study how myeloid cells contribute to viral persistence and inflammation. In our earlier work, using this mouse model, we described that macrophages are infected by SARS-CoV-2. This infection and replication of SARS-CoV-2 in macrophages activates inflammasome, which is a very inflammatory pathway, yielding to maturation of inflammatory cytokines such as IL-1 beta, IL-18, and CXCL10. And blocking this inflammasome was curative and helped with the lung pathology. We also validated these mouse findings in humans in looking at autopsy lungs, where we've seen about half of the macrophages at the time of death being infected with SARS-CoV-2, and the inflammasome activation was also recapitulated and validated in human studies. So more recently, we've been focusing on other myeloid cells, particularly human neutrophils, and trying to determine how they do contribute to viral persistence and the resulting inflammation. So to be able to do this, we had a challenge because none of the humanized mouse models actually had human neutrophils. So we developed 2 mouse models, one targeting GCSF, cytokine and receptor axis, and one targeting CD47 SERP alpha, do not eat me signaling pathway. So today I'll be mostly talking about the second mouse model, which targets CD47 because it gives us more consistent data with human neutrophils and better engraftment in general. Sophia Peng in my lab, a new graduate student, has been tackling this question of how human neutrophils are driving inflammation and are they really contributing to viral persistence. So we call these mice magic mice. It's an acronym for the genes that we have humanized so that we get a human immune response in these mice that is recapitulating monocyte macrophage response as well as neutrophils. These mice do have platelets, so human platelets, which is another advantage following on the earlier talk, and then as well as the adaptive and the lymphocyte response. But we've been specifically looking at how SARS-CoV-2 is influencing the human neutrophils. So the first thing we asked was, well, is there a difference? Do human neutrophils actually make the disease worse? So we compared a humanized mouse that has human neutrophils with one that doesn't have, so magic and the non-magic humanized mice. And we've actually seen that human neutrophils do worsen lung pathology, and you can see the quantification of the scoring here. But I would like to remind you that the mice that don't have human neutrophils still have human macrophages and other human immune cells which contribute to pathology. So our quest has been, well, what about the unique aspects of human neutrophils? And we have found that the unique response of human neutrophils to SARS-CoV-2 was forming nets. And nets are neutrophil extracellular traps where neutrophils package and secrete their nucleus out. It's very rich in DNA, histones, as well as antimicrobial proteins and that are secreted by the neutrophil. But this netosis or net response was very unique to human neutrophils. So in mice that are not infected, mouse or human neutrophils do not really respond to SARS-CoV-2 infection. In an infected mouse, mouse neutrophils do respond to SARS-CoV-2, but not by forming nets. They are secrete inflammatory cytokines. They release some of their granules, and produce reactive oxygen species, but they do not make nets. And this net response is specific to human neutrophils. So then what we wanted to see is, well, is this a defect of the mouse neutrophils? Are they still functional after they're infected? And yes, they are. So if you take these neutrophils and stimulate them PMA ex vivo, now these mouse neutrophils start making nets. So it's not a defect. It's a very unique response of the human neutrophils. So we said, well, maybe human neutrophils are infected, and they do indeed express really high levels of ACE2, which is a receptor that we all know is required for SARS-CoV-2 entry. So we took our approach to in vitro, and isolated human neutrophils and infected them with a reporter strain of SARS-CoV-2. When a cell is infected with this reporter strain, it lights up an MNG. So indeed, human neutrophils, when they are co-cultured with SARS-CoV-2, have MNG signal in them, and this MNG signal can be, is lost when we treat them with antivirals such as Paxlovid. But we first wanted to say, well, neutrophils are also known to be, very phagocytic. Maybe this is not there might be other parameters here. Maybe we should test their phagocytic potential. Maybe that is the pathway, and this is not. So when we blocked phagocytosis, there was no effect in netosis. However, when we blocked viral replication, either with Paxlovid or remdesivir, we could see very clear reduction in netosis, but not a complete loss. So then we took this approach to next level, and we wanted to see, well, this was only looking at neutrophils. How about if we look at them together with epithelials? And we've developed these neutrophil co-culture system with primary epithelial cells with an air interface system. And this is just a schematic of you can see that our epithelial cells are here, our neutrophils are here, and then we add the virus on top. And indeed, in this system too, epithelial cell infection was sufficient to induce netosis. And this netosis, again, was reduced when we used Paxlovid, but it's not completely gone. So we're now trying to see if there is a antiviral regimen or a combination therapy that could help us bring us to completely baseline levels in netosis. So, and on top of this, we were also interested in looking at the effect of vaccination, whether using an mRNA spike vaccine could prevent netosis. We developed and optimized this in our mouse model, where we would give a single dose of the RNA vaccine, wait 4 weeks, and then boost it, and then infect with SARS-CoV-2. And then we collect blood and the lungs 4 days post-infection. In this mouse model too, so without the infection, we see very clear nets. As you can see here in the non-immunized section, and this is histology that was done by a very talented postdoctoral fellow, Simon Millet. You can see very clear nets. But in the immunized mice, there are no nets, suggesting that vaccination is actually protective in this net response, which could have therapeutic implications for the patients as well. So overall, in the laboratory, we're very interested in studying myeloid cells and how they do contribute to viral persistence and persistent inflammation that we see in both severe COVID-19 earlier, but also in long COVID patients. We've defined that viral infection entry could happen in 3 ways in macrophages and at least 1 way, ACE2 and neutrophils. It appears that CD16 is not involved in the neutrophil infection or the Fc receptor. In both of these myeloid cells, actually, SARS-CoV-2 is good at replicating. We've measured this either by a reporter virus or looking at subgenomic RNA, which is a product of replication. And in both of these cell types, it leads to a very inflammatory death, which really is important for the persistence of inflammation. In the case of macrophages, it is pyroptosis. In the case of neutrophils, it's netosis. And we're currently trying to find the magic formula of antivirals and other therapeutics to be able to control both the inflammatory consequence of these viral reservoirs in the myeloid cells, as well as blocking the viral replication itself. This has been a very collaborative effort. The lead in my lab in this project is Sophia Peng, but we've been working very closely with Floval Lab from the beginning. And our PolyBio team of Anna Pyle, Akiko Iwasaki, and Craig Weiland from Yale has been very good. And we are also analyzing blood samples that Michael Pelusa have collected from long COVID. And we do think there is a net aspect in these long COVID patients as well, and we're trying to determine what the mechanism is. So with that, I will end sharing, and please let me know if you have any questions. I'll be happy to answer them in the chat.
Thanks, Susan. All right, great. Our next speaker is Benjamin Reedhead. He's an associate professor at Arizona State University Banner Neurodegenerative Disease Research Center, and he will be talking about towards precision Alzheimer's, a salivary cytomegalovirus biomarker for patient stratification.
Thanks so much, Amy. Are my slides showing up okay?
Yes.
Great, terrific. I'm so glad to be here. Thanks everyone for your time and attention. Yeah, as Dr. Proal mentioned, I'm here to share some work we've been doing at ASU on a particular subtype of Alzheimer's disease and some efforts we're putting into trying to find a biomarker to find these patients in the clinic. And it's a little different from a lot of the terrific COVID-related research that you've been hearing about today. But I think there's also many shared themes. And so, unfortunately, many of us already know a lot about Alzheimer's disease. Many of us will have been touched by the disease directly or indirectly. It is the commonest form of dementia, characterized by loss of memory and impaired cognition and behavioral changes, and linked with these really classic hallmark neuropathologies that have been known and described for over 100 years. Nevertheless, it is within the top 10 causes of disease. It sits at number 7 within the US, and of those causes of disease, it's the only cause that doesn't really have any powerful disease-modifying therapies. At least that was true up until a couple of years ago. We do have some that are kind of offer some modest improvement, but there's still a long way to go with Alzheimer's disease. And sadly, it's a very common disease. Every 65 seconds, someone in the US develops Alzheimer's. And to give you an intuition for this is what a full house at Stanford Stadium looks like, 50,000 people. And just in the US, 6.7 million people are living with Alzheimer's disease. So it's a big problem, obviously. And it's a very heterogeneous disease. Many different factors seem to be important for understanding Alzheimer's disease, genetic, environmental factors. And the little corner of biology that I focus on is the impact of chronic infections, peripheral infections, as well as central nervous system infections, and the ways in which this might contribute to or accelerate aspects of the disease. And what I want to share with you is, as kind of the context for our current efforts are a couple of quick vignettes that we published on last year and early this year that kind of lead us to what we think is an interesting Alzheimer's subtype. And this came out of a collaboration with terrific partners here in Arizona. And what we were doing was applying some of the latest next generation sequencing technology, looking at single cell level resolution to brain tissue from over 100 people with and without Alzheimer's disease, and then looking at all the different cell types that are present in the brain and those that might look different between patients with and without Alzheimer's disease. And there's a ton of complexity that we could talk about here, but one of the areas that we zoomed in on was a subset of microglia that we saw as being more commonly observed in patients with than without Alzheimer's disease. And microglia are tied intimately with kind of core pathological processes in Alzheimer's disease for a number of reasons. And so we're interested to know whether or not these microglia can tell us anything interesting about the disease, because we weren't seeing them in all Alzheimer's patients. We were seeing them in maybe 35 to 45% of patients. And so we did a bunch of network modeling. That's, one of my particular biases is a very kind of network-centric approach to trying to understand biological complexity. There's a lot that we could talk about with these microglia, but essentially for several months, we were scratching our head trying to work out what is it that is different clinically or neuropathologically about these patients. Does it tell us anything actually useful? We started to realize they actually look very much like what's called a senescent type of microglia. So these are cells that are kind of riding the edge of burnout. Microglia, for anyone that's not familiar, these are the most abundant immune cell type in the brain. We saw that these were Alzheimer's patients that had a little bit more neuropathology than other Alzheimer's patients, but beyond that, they didn't really differ. They hadn't had the disease for longer. There was no difference in ApoE or age of onset of disease or sex differences. Where our breakthrough came from was on a bit of a hunch. We generated some proteomics data from transverse colon tissue samples from these same patients where we profiled brain tissue. And what we saw was when we compared Alzheimer's patients with versus without these particular microglia, which I'm calling here CD83 microglia, just on the basis of some markers on the surface of the microglia. What we saw is that the biggest difference actually was an increased abundance of a certain immunoglobulin, a certain antibody called IgG4. And it's an antibody subtype that doesn't get a ton of attention. It generally makes up a minority of circulating antibodies in a healthy subject. It's somewhat considered to be an anti-inflammatory IgG. That is, it'll bind to an antigen, but then it won't initiate phagocytosis or complement activation. And so that's kind of where this first vignette finishes. We've got these subset of Alzheimer's patients that have these interesting microglia, and one of the key differences seem to be that they've got more of this IgG4 in their gut. And then where we picked it up in this next study, which we published in Alzheimer's and Dementia very late last year, we were trying to work out, what does this mean? Does it tell us anything interesting or useful? We've got this CD83 microglia linked with increased immunoglobulin G4. And what we report in this paper was that this actually seems to be associated with human cytomegalovirus. It's a very common human herpes virus. Human cytomegalovirus in the gut and in the vagus nerve and in the brain of these patients. And so what I'm showing here is some tissue sections from transverse colon in some of these same patients, and the dark staining is the IgG4. So showing a consistent story as to what we'd seen in the proteomics. So what we wondered then was whether or not, so we've got more IgG4 in the gut of these Alzheimer's patients. Is this specific or non-specific? Are these antibodies against particular antigens, or are they somewhat non-specific? And so we use this terrific technology called serum epitope repertoire analysis, which gives us a somewhat data-driven, unbiased readout of which pathogens or antigens in individuals antibodies are seeing. And we did this on the IgG4 component taken from cerebrospinal fluid. And what we saw was actually a really clear signal that these IgG4 antibodies especially were enriched for HCMV. So this was a comparison between Alzheimer's patients with versus without these CD83 microglia. So we started to feel like maybe these were specific antibodies, that is against a specific class of antigens. And so we went and looked in these same gut tissue samples from these same individuals for HCMV. What we saw was evidence of an active infection, seeing both late and immediate early antigens being produced. And so then we wondered whether or not it was actually getting up into the brain. And maybe the CD83 microglia are a response to that. And so what you see here are tissue sections in superior frontal gyrus. And we're actually seeing a bunch of late and immediate early antigens from CMV in the CD83 positive Alzheimer's patients, but not in the Alzheimer's patients that didn't have these microglia. And we looked at a bunch of patients and put a bunch of statistics around this, but essentially the story is we're seeing very tight correlation between the presence of CD83 microglia and the presence of IgG4 in the brain and the presence of cytomegalovirus in the brain. And I'll skip forward a little bit, just to quickly say, we think one of the ways it might be getting into the brain from the gut is through the vagus nerve. And we did some interesting organoid studies where we saw that the more virus is within a neuron, the more amyloid beta 42 it will produce, and the more phosphorylated tau, which is kind of a molecular precursor to tangles, these hallmark plaques. So we're building up a picture for how it is we think that this virus might be tweaking Alzheimer's relevant biological pathways. There's a lot of complexity to cover in a short amount of time. So, but just to bring you to the point of this presentation, like where are we looking next? What we actually would like to check is whether or not there could be a role for like careful use of antivirals or some other immune-themed therapy in this CMV-associated Alzheimer's disease. We don't know whether or not this virus is actually causing the disease. We've got some evidence, consistent with the fact that it might be contributing to it in some people. But really, I think it's such a complex question. The way to answer it might just be empirically in a clinical trial with drugs that are known to be safe. And there is a precedent for this. There is a recently concluded trial being run out of Columbia University in New York, looking at valcyclovir to treat a different subset of patients who have HSV1 associated early mild cognitive impairment. So there's a precedent, at least, for antiviral use in Alzheimer's disease. And whether or not it ultimately ends up being therapeutic, we'll have to see. But this is one of the directions we'd like to push towards. But what we need as a precondition for that is a good non-invasive biomarker that can, so all the data that I showed you before were in body tissues generously donated by patients that have been studied in life and then generously donated brain and body tissue after they passed away. So we need a way to non-invasively tag these quite complex brain and gut states that led us to this story. And so we spent a bunch of time looking at whether or not there might be a serum IgG4-based biomarker. And this was really generously supported by PolyBio, who we're tremendously grateful to. And what we ended up seeing was when we measured IgG4 in the serum, we were seeing certainly a positive signal. We were getting a classification area under the curve of 0.73, so certainly significant, but it wasn't the slam dunk home run strength of a biomarker that we'd really like to see for it to be really powerful in the clinic. And so where we're at the moment in our thinking is actually thinking that the salivary compartment is actually very neatly tagging this virus when it's acting in a way that we think is relevant to this patient subtype. And in retrospect, it's not that surprising that saliva should be so relevant to CMV. It is known to be a viral reservoir for cytomegalovirus. And so by virtue of some of the tissue that we have access to at the Banner Brain and Body Donation Program, and as part of this same cohort that we've been studying, we're fortunately able to look at submandibular gland tissue sections. And what I'm showing here is a sample of submandibular gland tissue sections being stained for CMV on the left and no CMV being detected on the right. And what we're seeing is a very strong enrichment in Alzheimer's disease. It's not everyone that has this. maybe about 60%, as measured, using these assays, which is about a 24-fold enrichment between Alzheimer's patients and age-matched controls. And so there are devices on the market made to detect salivary CMV. In conversations with some of these vendors, this sort of approach hasn't been used before in Alzheimer's disease. But what we really want to check is whether or not detection of salivary CMV might tell us something helpful. And so what we're pointing towards at the moment, and what we're kind of trying to assemble some of the pieces for, is a proof of concept study where we'll look at adult salivary CMV detection in patients with Alzheimer's disease and biomarker evidence of Alzheimer's disease, and then some age-matched healthy control subjects. And what we expect to see is very strong enrichment for CMV in the saliva of these subjects. CMV has been studied in the context of Alzheimer's disease before, and it's kind of yielded mixed results, but these have been studies looking at antibody presence usually in serum. What we think is that the saliva is really going to be where the signal is that we need to focus on. And so this is one of the directions we're looking at the moment, kind of a proof of concept study, not an intervention trial, but we're simply testing the saliva of a sufficiently large set of Alzheimer's and control subjects and see if we see the enrichment that we're expecting to see. So it's a lot to cover in a short period. So thanks for sticking with it. To conclude, we think that CMV infection in the gut seems to be a precondition for eventual passage to the brain with apparent impacts on Alzheimer's pathobiology. We think we're seeing promising findings that there are non-invasive ways to detect this phenotype in a way that could be clinically useful. We think saliva is one of these ways. And with that, we think there's all sorts of exciting biomarker and also therapeutic opportunities to help improve the sorts of treatments and patient stratification, so that we can serve patients affected by this disease, in a more powerful way. Really grateful to be able to present this work on behalf of many terrific friends and collaborators. Really grateful to PolyBio for their generous support of this work. And with that, I'll wrap it up, and I'd be happy to take any questions at the end if anyone had any.
Awesome. Thank you, Ben. That was really great. All right, our next speaker is Ed Breischwartz. He's a professor of medicine and infectious diseases at North Carolina State University College of Veterinary Medicine and an adjunct professor of medicine at Duke University Medical Center. He'll be talking about developing an advanced diagnostics to identify the Babesia parasite.
I first want to thank PolyBio for the invitation to present this data today, and I want to acknowledge my co-principal investigator, Dr. Ricardo Maggi. No, I can't get perfect. So our research for literally the last 30 years is focused on the genus Bartonella. Over that period of time, we have focused on Babesia as a pathogen in domestic animals, wildlife, and production animals. But we've kind of wandered back into the world of Babesia as a result of developing a droplet digital PCR assay for the detection of Babesia, Bartonella, and Borrelia species. The 3 Bs, as they have been referred to recently, are seemingly important in patients with Lyme disease and chronic Lyme disease. And we then wanted to clarify, our initial focus had been on trying to determine what species of Bartonella, and we wanted to have better clarity on what species of Babesia might be involved in those patients. I want to point out with the research testing strategy on the right that our initial strategy, which is the left side of this diagram, was to focus on Bartonella serologically and with PCR and enrichment blood culture. I think the importance of what we have generated in publications that I want to be clear about is the fact that for each individual that would enter our study, we are actually looking at 15 independent samples that have been DNA extracted from serum, from blood, and from 7 and 14-day enrichment cultures. And to my absolute amazement, what has helped us better identify on a DNA basis the presence of Bartonella is also helping us identify the presence of Babesia. And this is one of the most recent case reports that I would highlight, in as much as this is a child that was scratched on the face by a feral cat in Canada at 2 years of age. And cats are the primary reservoir for Bartonella henselae, and fleas are the primary vector that infects cats with Bartonella species around the world. At 4 years of age, the child developed a rash, and both of these photos were provided by the parents, but there wasn't a tick, and the rash actually disappeared within a few days. Within a week after that occurred, the child started seizuring. Those seizures progressed to status epilepticus, multiple hospitalizations, which persisted from 4 years to 8 years, when a brain biopsy was obtained to determine whether the child had Rasmussen's encephalitis. It was actually the parents that insisted, because of the cat scratch, that we test, and we were able to document not only the presence of Bartonella henselae in the brain, but also the presence of 2 Babesia species, either in the brain or the blood, that we collected over several months. The second observation that was facilitated by digital PCR that told us there was something there, and then by what we'll discuss next, and that was to develop species-specific probes and primers to identify what Babesia was actually infected, was Babesia coli. This is a Babesia, as you'll see on the subsequent slide, that is very prevalent in wild ruminants throughout North America, Central America, and South America. And digital PCR was able to tell us that there was a pyroplasma within the blood samples, and then ultimately the techniques we'll describe next allowed us to actually obtain amplification, sequencing, and confirm the Babesia species. The importance of the table, and again, this is very brief on this particular publication, but the age range went from 2.5 years, a child in Oklahoma, to 60 years of age. You'll see that veterinary workers, predominantly because I'm a professor at a veterinary college and our research is focused on Bartonella and veterinarians early on, are perhaps overrepresented. You'll also see that this spans many states within the United States as well as Mexico, where both a mother and her daughter were co-infected with Bartonella quintana and Babesia coli. So this particular slide illustrates sequences either generated in our lab or in GenBank from deer, reindeer, elk, and caribou from North America. And what is very obvious is there is tremendous sequence similarity amongst the various isolates that have been reported for Babesia coli. And based on 18S sequencing, which is used by researchers around the world for the pyroplasmas, it had been suggested that Ota coli also existed in Europe. And I just have 2 references down there supporting prior publications of that in GenBank. And what we were able to find once we got a good sequence for Babesia venatorum, which again is another ruminant Babesia from the Europe, is that these were probably misclassified Ota coli cases and are actually venatorum. So the focus that Dr. Maggi took was to amplify regions of the intergenic spacer region between the 18S and 28S ribosomal RNA. And obviously, compared to the previous slide, you'll see that there's tremendous variability. And our primary focus was to look at both Babesia species that are known to infect humans in the United States, which is Babesia otocolii, Babesia microti, the most important one in the northeastern United States, transmitted by Oxodes scapularis. Babesia duncani MO1, which was originally found in Missouri and has been reported in just a few case reports from humans within North America. And then Babesia duncani, which is transmitted by ticks in the western United States, essentially the other side of the Rocky Mountains. So using sequences that we derive from the different ITS1 and ITS regions, we generated specific primers for digital PCR and qPCR, and ultimately use those for conventional PCR. And this is just to depict the fact that we could multiplex this with the QIAGEN Acuity digital PCR equipment, and in a single run actually determine whether it was one of these 4 individual Babesia species. So ultimately, that data was published in Pathogens in great detail. Fortunately, on the veterinary side, we were able to get numerous Babesia species. It's estimated, based on visualization, that there's over 100 Babesia species that exist. And many of them animals are reservoirs for, so it's easier to find the Babesia in the animal, visualize it microscopically, and then have samples that you can actually test and extract DNA, do PCR and sequencing. So we were able to look at a very large number of Babesia species for this multiplex process, and then actually focus specifically on the ones that are transmitted by ticks in the United States and cause babesiosis in humans. I just indicate one more publication that resulted from this particular research, in as much as this family, the mother contacted me because the son had neuropsychiatric illnesses, and the mother and father, using the techniques that we had developed, were co-infected with Babesia divergens MO1 and Babesia microti, and the children, 2 daughters and son, were all infected with Babesia divergens, and both of the dogs in the family were also infected with Babesia divergens. Interestingly, shockingly, and surprisingly, the entire family and the dogs were infected with Bartonella quintana, for which humans are considered the only reservoir, and for which the human body louse is considered the only competent vector. So we have a very unusual outbreak situation. And I think the importance for this, for me as an internist on the veterinary side, is the complexity of these vector-borne organisms that can persist within either cells within the blood, erythrocytes, macrophages, neutrophils, and ultimately contribute to very complex disease presentations. So in summary, by targeting non-conserved regions of the 18S region, we were able to develop species-specific multiplex assays for the detection of the most common Babesia species infecting humans in the USA. ITS targets increase both qPCR and cPCR detection levels that are close to dPCR. We were able to facilitate species identification by DNA sequencing down to the strain level, which varies tremendously amongst the Babesia otocolii that exist in ruminants in North America. We were able to detect and confirm with 2 to 3 different gene targets Babesia infection in chronically sick individuals, either as single or co-infections. And I'd certainly like to acknowledge the team in our laboratory that's contributed to the development and validation and the studies that we've done in documenting both Bartonella, Babesia, and co-infections with both vector-borne organisms in humans. In regard to PolyBio, I certainly like to thank them for allowing us to continue research with Babesia, and we are currently applying the techniques that I just briefly discussed in a long COVID study for which we should finalize and be able to provide data by the end of the year. Thank you very much.
Thank you, Ed. This is great, and I'm excited for that long COVID study. All right, our next speaker is Jesus Estefes. He's a postdoctoral researcher in the TAN lab at the J. Craig Benner Institute, and he will be talking about persistence of viral antigens and immune endocrine features in long COVID.
Awesome. Thank you, Amy. Can you guys see my slides?
Yes.
Perfect. So thank you for that introduction, Amy. Today, I'm really excited to share some ongoing research that I'm currently doing at Dr. Tan's lab, in which we are going to dive into persistence of viral antigens in long COVID and potentially as a biomarker to better understand long COVID. So without further ado, let's go ahead and get started. So as many as you know, long COVID is a chronic disease where typically these patients are experiencing symptoms beyond 3 months. And in spite of great progress, there's still so much to understand. And that is something that we are is yet to discover, and I hope to discover and hope to dive into in this case control study that we currently have here at JCVI. So now I'd just like to introduce the current case control study that we have here at JCVI. This case control study is from Cardiff University, collected by Dr. David Price. And I'd like to point your attention to the left side of the screen where you can see that we have a total of 217 long COVID participants, and we have a total of 118 convalescent controls. So these long COVID participants are patients who experience these chronic symptoms 3 months post initial infection, while these convalescent control participants are people who recovered from the acute infection. So I like to also point out that this is mostly dominated by females and also dominated by Caucasians. So we just have to take that into account when doing our analysis. So now on the right hand of the screen, I like to show some overall comparison and age differences between both groups. And I just want to highlight that there is no significant difference, which really shows how well this case control study was written or looked into. So now that I've also want to introduce another important factor of this case control study, a huge part of this is the date in which these samples were recruited. And I just want to highlight that both long COVID and convalescent participants were recruited 3 months post-diagnosis, and even some were recruited 6 months post-diagnosis. So this is just important because it kind of gives us clarity that this is a true case control study, because these long COVID participants were recruited post those 3 months, which gives us more emphasis and clarity in a way. So now that I've introduced the case control study, I want to have this question that most researchers, including myself and other researchers, are trying to dive into, and that is what is underlying the cause of these chronic disease endotypes seen in long COVID. So some potential mechanisms that can explain these long COVID symptomology is viral persistence, cytokine dysregulation, autoantibodies, mitochondrial dysfunction, and endothelial inflammation. For this specific presentation, I want to dive specifically into viral persistence, and I just want to highlight that this can be a multitude of multitude of mechanisms, not just one. But for the specific presentation, I'll just dive into viral persistence. So diving into viral persistence, I want to highlight that as many as you know, in acute infections, typically the spike protein usually resolves within 2 weeks. However, we're finding out that these long COVID participants potentially can have this spike antigen floating within their plasma well beyond that acute infection. And that could be a potential reason as to why these long COVID participants are experiencing these symptoms. So now that specifically, I want to highlight that we're going to look into spike, not necessarily into the N or the S1, where we eventually will like to look into with the current assays that we have in hand. So now that I've introduced the cohort and our question of looking into viral persistence, I want to introduce the platform where we're able to detect viral persistence. So the Simoa, or known as a single molecule array assay, is essentially a very sensitive sandwich ELISA where it can detect protein or antigens up to the picogram level. In this specific case, we're interested in the full-length proteins, so we are using antibodies to detect a certain region of the spike. So we have two antibodies. One will go ahead and look into the S1 region, while the other one will look into the S2, and that will give us the overview of that full-length protein. So now I'm just explaining that I want to look into some specific preliminary data that we currently are doing in Dr. Tan's lab. So this is some very preliminary data, but I just want to highlight how we're seeing that these long COVID participants tend to have higher plasma spike in their plasma compared to the convalescent control. Although we see no significance, there is a trend where long COVID participants tend to have more variable of spike, while convalescent controls tend to group towards the left bottom of that graph. So we also did overall analysis where we compared the overall amount of spike between the two subgroups, and as you can see, there is a statistical significance between long COVID and convalescent control. So now that we've introduced that there is residual plasma spike in this plasma, I want to highlight some that we're interested in some host biology, where on the right hand of the screen, you can see that there has been journals that have been shown that the spike actually does bind to the estrogen receptor. So that could potentially be a reason why these long COVID participants are experiencing these symptoms. And I just want to show some relevant work that we've shown previously, where we see that long COVID participants experience higher amounts of estradiol compared to convalescent controls in both females and males. So now that I've shown that, I want to do some correlation analysis between estradiol and spike. Although, again, there's no significance because we haven't done the entirety of the cohort yet, we can see this trend where convalescent controls have low amounts of estradiol and low amounts of spike, while long COVID participants have a more variability in this platform. So with this into account, we are hoping that we can essentially use this as a biomarker and as a possible explanation why these long COVID participants are experiencing this. So in conclusion, I want to just state that the circulating SARS-CoV-2 spike protein persists in current subset of individuals beyond 3 months, and that long COVID participants generally have a higher amount of this spike compared to the convalescent control. And while all this is really great, we also have to be just careful of interpretation, because recent exposure to SARS-CoV-2 can affect that, and also recent vaccination. So also I want to highlight that there is no significant correlation as of yet, but we are hoping to see some significance as we expand our cohort. So some future directions that we hope to explore at Dr. Tan's lab. We want to definitely finish off this Cardiff cohort and see if these trends hold. And essentially, we want to look in other parts rather than just plasma itself. We want to look into exosomes and into other biofluids. And also, we want to look into longitudinal studies to track spike in immune endocrine markers over time to understand dynamics and persistence. And then lastly, we would like to establish a long COVID animal model to look at estradiol correlation spike in a more controlled environment. So I'd like to make some acknowledgements. I'd like to thank my mentor, Dr. Gina Tan, Dr. Max Chin, Dr. Christopher Dupont, Dr. Marcelo Ferrer, Dr. Richard Sherman, and a huge shout out to Emory Proal for giving us this platform, and a huge shout out to Cardiff University for providing these samples. Without them, this work would not be possible. So I'd be happy to take any questions in the chat if you guys have any. Thank you.
Thank you very much, Jesus. All right, great. Our next speaker is David Izquierdo Garcia. He's an assistant professor in radiology and an assistant in biomedical engineering at Harvard Medical School. He'll be talking about evaluation of tissue fibrin accumulation in long COVID via PET imaging.
Okay, fantastic. Thank you very much, Amy and the PolyBio Foundation for inviting me once again to talk about this super exciting project. I'm going to talk about detection of fibrin non-invasively in vivo for the first time in long COVID using positron emission tomography. Let me go again into the presentation. Here we are. So why fibrin? Well, fibrin is one of the main components of blood clots. And there's been a lot of evidence. Already explained that in our last symposium. A lot of evidence for a lot of colleagues. Probably you have heard it already today quite a few times. Now, clotting has been a major issue in COVID, but also in long COVID. There is evidence that there is a microclots being formed in the blood and accumulating in certain parts in the body, above all, especially in very sensitive areas like lungs and the heart, the brain. And we are in a very unique position. We are a lab that has developed for the first time a non-invasive way to visualize and detect fibrin and blood clots, basically in vivo. So we are going to be using PET radiotracer. What is PET? PET is a positron emission tomography technique where we use a radioactive probe, basically something that we inject in blood in patients. That probe is functionalized, in this case, to target fibrin, to target blood clots. And because it's radioactive, it's going to emit radiation, and we are going to be using that radiation to locate, detect non-invasively where this fibrin accumulation, these blood clots accumulating in the body. Why, you would say, why are you using PET? Why are you using this particular technique? Well, the idea is that PET is the most sensitive technique across all the imaging modalities. So we only need a very, very tiny amount of the compound to accumulate in the body to visualize this. So in order to put a very simple examples, around only 250 cells that have this compound accumulated is enough to detect what is going on. And that's very, very little. PET is basically the workhorse in the nuclear medicine department, and mostly for tumor and metastasis detection, yes. So we wanted to find out if we can use our new probe that binds into fibrin to detect microclots in long COVID. So basically, that's what we've been doing in the last months. So far, we've scanned 12 subjects. Like the previous presenter, Dr. Steven, we've seen mostly female prevalent population. So out of the 12, it's been 10 females and 2 males. So 8 on the long COVID and 4 controls. Age, weight, height, BMI, injected, those are all very similar. So no significant difference among the different groups. And our imaging protocol, basically, we are going to be using the PET tracer injecting at the same time as we scan simultaneously the positron emission tomography, it has the fibrin imaging non-invasively in vivo, together with some MRI. So we are going to acquire, obviously, anatomical images for the MRI. So the red part is basically on the brain. And then after brain, because basically most of the symptoms are on COVID, not uniquely, but most of them are basically on the lungs, on the heart, on the brain. And we wanted to see what is going on. So this was our first result. We first acquired a patient with a lot of symptoms and with already clinical visualization with a CT. This is not a CT, this is an MR image, and we already seen some basically some formation of a scar, basically. So there is some lung involvement already, and it was new. We just wanted to show for the first time. and potential fibrin accumulation. So that was our first subject. So we knew we can detect clotting in long COVID. Now the question is like, what can we do the same with patients that have symptoms but don't have any clinical evidence? So what we are trying to do here is, can we get a biomarker for the first time that defines whether long COVID has got a signal that we can measure, quantify it, and detect it in vivo, non-invasively? And we've been acquiring a few patients. We have, so these PVFs are all non-COVID subjects. And what I'm showing here on the right lower part of the images is always, it's not a fully healthy control, but it's what it was the equivalent of it. It's an atrial fibrillation patient before the long COVID time. So no lung involvement, or mostly no lung involvement. And that's basically properly a patient control right now. So what you see is quite a lot of difference. Yes, a lot of lung involvement in some of the subjects, but we have others where there is very little lung involvement. We've done more, and we see basically the same pattern. Some subjects with some involvement, and some subjects with little or very, very little involvement in the lungs. So then we acquire more healthy subjects. And what we are saying is that healthy subjects have indeed very little involvement, but not quite little to the healthy subjects we had prior to long COVID. And that's something very interesting. Let me get a summary in a table of the PET results, or quantifications, numbers, basically, yes? So those are 4 different flavors of the signal that we obtain in PET. SUV is a standard uptake value. TBR is basically taking the SUV mean versus the signal in blood. And then we can do the same with maximum values, and the same with the ratio with blood, but taking the maximum values. Okay, so SUV means controls versus long COVID. So this is controls that already had COVID, but are not long COVID subjects. We don't have any significant differences, neither with TBR. We see some a trend on the SUV max or TBR max, but again, nothing is significant. Now, what happens if I am adding our 24 healthy controls that were atrial fibrillation, so not properly healthy controls, but no lung involvement before COVID? The numbers change quite dramatically. So the first thing they will notice is that TBR is very low compared to the previous healthy controls. The SUV is lower, but not as dramatic. But then the SUV max and the TBR max are higher. Why is that? Well, these are atrial fibrillation patients. Yes, they are cardiovascular, very heavily involved patients. So most of these cardiovascular patients have renal impairment. They are older people. So basically the blood, the reactivity in the blood basically remains more time, because there is renal impairment. So the trace is not excreted as fast as in real healthy subjects. So to confirm that, we just measuring the signal on the left ventricle, basically blood pool. And what we are confirming is that the controls on the long COVID is very similar, but in the atrial fibrillation is much higher. So yes, there is very few excretion. So which means that basically we cannot have any ratios with the blood, because they are useless with this patient population. And why am I going to read the SUV max to? Is because, again, these are cardiovascular patients. So a lot of the cardiovascular patients develop also lung symptoms. So it doesn't need to be going on through the whole lung. So the SUV mean is a good measure to quantify the lung, but they may have small, very tiny lesions. They may have a very, very large signal in terms of fibrin. Okay, so if I'm plotting this in a nice violin type of plots where we see where all our subjects are, this is our long COVID cohort. This is our healthy controls post-COVID cohort, and no difference with SUV mean. What happens if I'm adding this healthy, pseudo-healthy controls before COVID? That's why we say BC. Well, everything becomes now significant. So there is difference between the long COVID and these, pseudo-healthy controls, and even difference between the healthy controls post-COVID and the before COVID. So just to summarize all the things you just said today, we have completed 12 subjects, 8 long COVIDs and 4 healthy controls, and we had 24 prior, not completely healthy, but in terms of lung involvement, mostly healthy controls. What we are seeing is a bimodal distribution of the long COVID signal. So they are long COVID subjects that have a lot of lung involvement and some subjects that have very little involvement, very similar to the healthy controls. The healthy controls that we are acquiring in the post-COVID area, they have a higher signal than the prior healthy controls. And we wonder if there is anything that has changed due to the COVID. All these patients have already passed COVID. So if there is any slightly change in the signal and the baseline is already higher. So that's something that we need to understand more. Of course, we need to still make correlations with clinical scores. We have spirometry levels. We need to all the lung symptoms. We need to identify if patients with more lung symptoms or worse spirometry are the ones with higher PET signals versus the ones that is lower signals, because we can actually differentiate both populations. And because we already acquired DC in the lungs for these patients, we need to measure and see if permeability in the lungs correlates with this fibrin accumulation or not. And because we already did brain and heart, we need to also find out if some of the symptoms also correlate to the same in the brain and the heart with the brain with the same fibrin signal in those areas. So the question is still open, but we are seeing promise. We see, this bimodal distribution where patients have been heavily affected, even though they are not having any other clinical biomarker, either CT or MRI or any other thing. We are seeing signal on the PET side, and others where there is no involvement. Now, we need to identify who are those type of people and why, some of them are having this type of signal. I hope you enjoyed this. Of course, acknowledgements, first of all, to Amy and Mike, the PolyBio founding research foundation, basically, for all the help and the money of the founders and the donors and volunteers, obviously, for coming forward and making this project possible, and all the members in the lab. Thank you very much.
Thank you. Thanks, David. Great. Okay, I'll introduce our next speaker. It's Lael Jonker. She's a physician scientist and assistant professor of pediatrics center at Harvard Medical School. She'll be talking about neutrophil spike protein interactions in pediatric long COVID.
Great. Thank you so much. Thank you for the invitation again to speak. Let me just start these slides up. And so one change from prior talks is I have left Mass General and I'm going to be starting up at UT Southwestern Medical Center and continuing the work there. And so the overview of this very brief talk is I want to just touch on, emphasize that children can be affected by long COVID. I want to talk about what we've been learning about how spike can elicit inflammation and injury, and then briefly touch on what we're trying to do about this. So children are often sort of put in the background of the long COVID discussions, and maybe about other diseases as well. But for long COVID, it's likely unrecognized in children for many reasons. First, it's underappreciated in pediatric providers, or symptoms are minimalized, or symptoms are attributed to other causes. And then there's also stigma within the families and the kids themselves to not seek care and to try to sort of not be noted as different. And so it's challenging to be able to make progress when we're having a hard time identifying it in long COVID. So another plug for biomarkers. But kids are known to have symptoms of long COVID after acute infection, and this work is really built on really great multi-center work led by Rachel Gross, looking at symptoms that are increased in children after they've had a SARS-CoV-2 infection. And even down in toddlers, they can have increased symptoms of stuffy nose, wet cough, difficulty sleeping, as compared to those who've never been infected with SARS-CoV-2. And then in older kids, you see more of the things that older kids meaning, 6 and above, you start to see symptoms more common in adults, like brain fog, difficulty with attention, pain, stomach complaints, and some anxieties too that can come up. And so, there's lots of evidence. I'm sure this was talked about from other people. Actually, I know there were some talks focusing on viral reservoirs after SARS-CoV-2, and certainly most of this literature is completed in adults, although we've published some studies showing that kids can have these prolonged reservoirs. And so there's no reason that that children aren't affected in the same sort of way. And having these viral reservoirs, here's a slide of Amy's, has a lot, or adapted from Amy's slide, is that it has lots of effect on the body. And so what we wanted to do is further understanding of how this worked in general, sort of focusing on neutrophils and focusing on children. And what I really like about working with samples from children are two things. First, it highlights that there are objective physiologic differences between children who have long COVID and children who don't. And second, children often don't have these accumulated age, related inflammatory disease like like just high cholesterol, diabetes, metabolic syndrome, or other things that can happen to us as we age. So they're a little bit more straightforward, and you're dealing with one problem at a time generally. And so what we did was we collected blood from children with long COVID, also the post-COVID syndrome, multisystem inflammatory syndrome in children. And we did a bunch of different assays to try to characterize neutrophils in long COVID. And I'm not going to go through all of these today. I'm going to focus just on one form of activation, but we did cytokine profiling, we did RNA sequencing, and we did some functional assays. But I want to focus on one that really jumped out, and this was increased neutrophil extracellular trap formations. And that's when basically the cell breaks down, the cell wall breaks down, and the DNA is expelled from the neutrophil in an attempt to ensnare, encapsulate any pathogen that's in the area. And so it's sort of a last-ditch attempt, like Spider-Man casting a web on whatever virus or bacteria to try to contain it. The problem is when it does that, it also can cause irritate surrounding tissue. But what we've seen in long COVID is we use this little chip. I'm working with Daniel Aramia's lab, where we put blood, actually isolated neutrophils, on these devices, and we image them over time. We just put them in and we image. We don't treat them in any way. We just look at what happens to them. And you can see these neutrophils over time. The dense DNA swells, and then once the membrane breaks down, the cells turn green based on the dye that we're using. And so when we quantify the amount of cells that undergo this netosis, this swelling and then breakdown of cell membrane, you can see in MIS-C, there's certainly a lot. We've published that there's a lot of spontaneous migration, but we also see this in long COVID. And so when we look at here's actually a quick video. So these are healthy neutrophils. They maintain their round shape. And they're happy and floating along. And then here are neutrophils in long COVID. And you can see they sort of stop moving. They get a little bit stickier. They start to swell. And then all of a sudden the green starts popping out because their membranes are breaking down. And that's netosis. And so we've looked at, try to understand why this is happening. And so we've done these assays where basically we add spike, we add antibodies to these neutrophils, and we try to make these immune complexes. These immune complexes then are added to the neutrophils. And what we see is when you add more spike, so increasing concentrations of spike along with antibodies, you have a high amount of netosis. And so that, shows us that these spike in the blood is activating these neutrophils. And so this seems, conceptually like a problem when you have these neutrophils expelling this DNA within blood vessels. It seems as though it could be a problem, but we wanted to better understand that. And so we again did extensive profiling on kids who never were infected with SARS-CoV-2. So these were samples collected early on, and then kids who've reported of long COVID. And I would say kids, I mean 25 and younger, so young adults as well. And when we look at the symptoms in this cohort, certainly they're symptoms consistent with what we're seeing across the ages. So lots of cardiovascular symptoms with feeling dizzy, lightheaded, persistent palpitations, chest pain, things like that. And we did some in vitro assays where we took endothelium and we grew in a cell culture, in a tissue culture dish. And we added different components. We added spike alone, we added neutrophils alone, and then we added these spikes with these neutrophils with spike immune complexes that were inducing the nets. And when we did that, we saw a lot of cell death, which is captured here. We also saw that, when we had the endothelium on a tissue culture plate, and we added these neutrophils with spike together and the immune complexes, so the nets were made. We also saw some loss of barrier function within the endothelium. And so again, not quite as dramatic as the cell injury with the LDH marker, but some increased cell barrier breakdown. And so this has been shown in adults. This is a lot going on, so I'm going to walk through this. But we saw increased microclots in long COVID patients as well, long COVID kids. And these have been shown in older adults, but not in kids. But here we do show that there's increased microclot burden. And we showed there was some endothelial injury. Here we showed that components of the microclot, like the amyloid P, for example, but also fibrinogen, were increased with cell-free DNA. And cell-free DNA is a marker of netosis. And so these markers are, suggesting that there's correlation. And again, we'll need models to better understand causation. But we do see, that the nets can injure endothelium, and we see lots of increased markers of endothelial injury, and we see this microclot formation. I'm not showing here, but we do have the increased cytokine profiles showing endothelial injury as well. And so we propose that this comes together as a potential hypothesis that contributes to long COVID. So we have the viral reservoirs in the gut. Zonulin-mediated leak of the gut allows spike to leak from the gut into the blood. So now you have spike protein in the blood. Spike protein is bound by antibodies, either from prior infections or, having gained immunity over time. And these immune complexes activate neutrophils. These neutrophils are released in the extracellular traps, are released into the bloodstream and, adjacent to blood vessels. That injures the endothelium, and that causes activation, inflammation, and correlates and potentially contributes to microclot formation as well. And so, I think we're learning a lot about what's causing long COVID. Now, what can we do about it? And so we have had a study looking at multisystem inflammatory syndrome in children, where we showed this stepwise pattern that we're understanding is what's, some underlying pathology in long COVID-2. So there's virus in the gut, there's increased zonulin release that allows for breakdown of the tight junctions, and spike leaks into the blood. As I mentioned, we're seeing similar sequelae in long COVID. Virus in the gut, increased zonulin, spike in the blood. And so we have this lorazepatide treatment, and lorazepatide competitively inhibits the action of zonulin. And so the tight junctions restore and they remain tight. And we've actually treated kids with MIS-C with lorazepatide, and we've seen shorter on time. So I'm just showing you that we've seen positive effects when we've looked at it through open label study, but also as part of a randomized placebo-controlled study. GI symptoms got better, they cleared spike faster, they returned to their usual health more quickly, and they returned to their usual activities more quickly when they were treated with lorazepatide as compared to those who were treated with just standard of care. And so now we're running this study in long COVID, and we're enrolling 150 patients. Here's a QR code if anybody's interested. And these are for patients ages 7 through 50. So we are extending our age group, 2-to-1 randomization, and it's a 3-week treatment with lorazepatide. Again, what this aims to do is keep the viral reservoir in the gut and prevent spike leaking into the blood, because we're having growing evidence that having spike leaking into the blood activates neutrophils and injures endothelium. And that, correlates with a lot of the cardiovascular symptoms that we're seeing. So in summary, post-COVID symptoms are common, and long COVID is likely underdiagnosed in children. Spike from persistent viral reservoirs triggers neutrophil activation and can elicit endothelial injury, and targeting spike trafficking into the blood may be an effective strategy. So thank you again for this opportunity. I feel like I ran through a lot quickly there, and I'll pass the mic on to the next speaker.
You did a great job. Thanks, Leo. Okay, and our next speaker is Shannon Stott. She's an engineer and an associate professor at Harvard Medical School. She'll be talking about microfluidic capture of SARS-CoV-2 particles in long COVID blood.
Wonderful. Well, thank you so much for the introduction. I think it fits that I am right after Lyell because similar to her, I'm currently at Mass General, but starting on December 1st, I'll be a new position in the Department of Biomedical Engineering at Boston University. So I recommend if anyone would like to follow up, you could use either email address as we go through, because I'd love to increase our collaborators as well on this work. To give you a quick overview, I'm a big fan of biomarker-related research, mostly doing all of this in cancer. So I think that biomarkers have a great impact in patient care, but I also think that diagnoses and therapies are delayed until those biomarkers are detectable. And so I think our approach is to find those rare biomarkers and see if we can get that detection as early as possible. And for the scope of this symposium, thinking about this in long COVID. So for our approach, jumping right to it, we're a microfluidics lab. And so we have a viral herringbone chip, and you can see here is the microfluidic device where we have our sample go in. And what makes it unique is we have microfluidic mixing inside the device. So we have staggered herringbone grooves where the particles will twist and turn as they go through. We also have some antibody engineering where we elevate our antibodies or our capture moieties so that you can have better access to small nanoscale sized particles. And then finally, we've done a lot of different coding efforts so that we can make sure that everything is stable over time. So our goal is to see if we can address this for high sensitive viral detection in blood, also having amenability to other biofluids such as saliva, stool, and urine. We work with very small volumes, so the smallest volume that we can work with is 50 microliters, but we can go up to 2 milliliters of sample. And then we're also thinking of looking at this also in long COVID patients, which is where PolyBio came in, as well as MISC-C patients. So how do we bind? This is the most common question. So I'm going to give you some high-level overview and then get to some of our long COVID data. How do we get the virus to bind to our surface? And so we use a receptor trap that models ACE2. So we coat this receptor trap onto the surface so that the spike protein will bind to that ACE2 receptor trap. We did comparisons along the way where we looked at antibodies against the spike protein versus this ACE2 capture versus a negative control. And our readout is the ddPCR probe detection for SARS-CoV-2 using the FDA-regulated assay because we run all of this in Mass General's CLIA lab. So we found that using ACE2, we had significant enhancement in our ability to pull out virus from these different fluids. Here's just a high-level overview of when we look at traditional just trying to detect virus in plasma without using any enrichment. We're not able to see anything, but when we use our microfluidic technology, we can pull out all of these viral copies. We can do this in whole blood as well as banked plasma, and then we can also look and confirm that we have intact viral capture. So I'm going to do a little extra on this intact viral capture part because I think it's one of the things that differentiates from some of the other talks that we've had. And both of measurements, whether it's the spike protein or intact virus, have value in us trying to figure out what's happening for these patients. So in our case, we have the virus flow through the chip and stick to the inner surface. Again, we have that ACE2 receptor trap on our surface. The virus sticks to it. We then flow through an RNase treatment. So if there's any free viral RNA, which is what we're measuring, this should dissolve any of that RNA. And then we do an extraction of the RNA from the chip and we see, do we see any loss of viral RNA because we've done the RNase treatment? And we found them to be statistically similar. If anything, we have a cleaner readout when we include the RNase treatment. So between our imaging as well as our various analyses, we are fairly confident that we are detecting intact viral particles from all of these different biospecimens. We've also been able to do this work funded from the NCATS program at the NIH, where they were able to give us blinded samples of all the different variants of viral particles of SARS-CoV-2. This is our limited detection work that we did at the CLIA lab at Mass General, where we were able to determine that we had a limit of detection of 3 viral particles per mil of plasma. So that's data here. We also looked for cross-reactivity and specificity of our assay. And then because we were very excited about this and processing our samples for acute COVID infections, we started to process samples over time to see if we could start to measure trends of viral load in relation to clinical care at Mass General. And it was here when we started looking at longer time points, even out to days at 100 days, we started to still see this presence of viral particles. So when we published this work, we were really excited to talk with Amy and thinking about, could we think about repurposing our technology to look for long COVID patient samples? So this is, again, our viral detection for acute patient samples. So we were able to detect in plasma 72% this intact viral particles, and then our data for saliva and stool. But we really think, and we're excited about this application of looking at long COVID, because the more we've learned after starting going down this pathway, we see this real need for a diagnostic for these patients. We want to help to be able to move the needle, as so many of you do on this call, of moving past the diagnosis of exclusion. And then also, if you are on a particular therapy, being able to have a quick readout that would be able to understand, is that therapy benefiting you as you go forward? We also not only want to think about measuring viral load for the patients that we're analyzing, we'd also love a readout of tissue-specific injury and end organ damage. And where this comes from is we're also an extracellular vesicle lab. So we wanted to see, can we measure both those viral particles as well as those extracellular vesicles? And to give you a little bit of what that would look like, so here's our flow. I talked a little bit, but quickly, of that viral capture on our chip. But our device is non-destructive, so we can just take the flow through as it enters one chip, and then we can put it into another chip. So we can put a chip there where we pull out epithelial EVs, another one where we can isolate T-cell EVs, and another one with innate immune EVs. Then you kind of separate each chip, you extract out the RNA for each one of those individual chips, and then you look at what are those signals and how they're changing. Here's just a quick peek at some of the data that we had from our early acute COVID patients that we were serially monitoring, looking to see what different RNA transcripts. And so we're still working on publishing this work and finalizing our analysis, but we were starting to see some differences across these EV populations that also correlates with what their viral load was. But now moving towards what really we're most interested in now is what's happening in these long COVID patient samples. So we're very grateful for the grant from PolyBio to be able to do a pilot investigation of, can we detect intact viral samples in plasma banked from patients across 4 different cohorts that we're looking at. So the first is long COVID samples, convalescent control samples, mild acute COVID samples, and then pre-pandemic control samples. And then after we complete that work, we want to be able to then test additional long COVID samples. So last time I presented was right after we got news of the award, and we were just starting to bring up the whole system again and getting things ready to process the plasma. So I wanted to acknowledge Dr. Roma Parikh, who's done all of this amazing work in our lab. She joined, learned all of our technology every step along the way. And then we also had to work on getting new receptor traps fabricated, because that was also something that slowed down after our COVID work was finishing. So I want to really acknowledge the laboratory of Jim Wells for doing a whole new production line, giving us those ACE2 receptor traps. So Roma took those new traps, as well as all of her learnings from how to use the technology, did a spike-in of inactivated virus. And then this is just our readout for, if anyone's familiar with the ddPCR assay, the N1 probe versus the N2 probe. And then we average the probe, and then we look to see, in this case, we spiked in 100 copies of the virus, 1,000 copies of the virus, 100 copies of the virus, 25 and 5, making sure that we still had that detection limit that we were seeing 2 years ago when we were processing the first acute patient samples. And so here's just an early look of the data of the first 10 patients that we processed. Now, I show this because I want to show that here's the progress that we're making, and we are excited about this data. We don't know, because we're still blinded to who these patients are. But for the first 10 patients that we've processed, in 9 out of the 10 patients, we are seeing intact viral particles, and these are the levels that we are seeing for the intact viral particle load. We're going to continue to process the remaining 115 samples that we have on hand, and then we will be unblinded. We'll work with Dr. Peluso. He'll share with us what were the samples and start to look at how does viral load impact. But we're very excited to see these levels, because what I want to point out is when we were looking at acute patient samples, the viral loads were much lower than what we're seeing now. So there we knew we had that really low limit of detection, but it was still very much typically under 10 viral particles per mil. But if we go back to what we're seeing in the long COVID, again, we don't know if these are all definitively long COVID patient samples, but we are seeing viral particles And we're just excited to see what's going to come. So right now we're leading with enthusiasm. We're very much looking forward to processing the remainder of the samples and seeing where those viral loads are. And then our next step is to then also explore the EV component in addition to now knowing what is the viral load, what are the EVs telling us as well, so we can think about that end organ damage and other systemic effects that are happening. And then of course, expanding that to serially test the long COVID samples. So I want to first and foremost thank all the patients who donated all of their plasma so that we're able to conduct this work. Really grateful to PolyBio, as well as all of the members of my lab and many of my collaborators at Mass General, as well as Jim Wells' lab. And thank you so much for your time today.
Great. Thanks, Shannon. That's really encouraging data on those first samples. I know it's early, but it's really cool. All right, our next speaker is Chiara Gianarelli. She's an associate professor in the Department of Pathology and the Department of Medicine at New York University's Grossman School of Medicine. She'll be talking about deep characterization of SARS-CoV-2 cardiovascular reservoirs and plaque formation.
Thank you, Amy, for the introduction. So my lab is really interested in focusing on cardiovascular disease. We study how the immune system responds, contributes to vascular inflammation and particularly atherosclerosis. And what we are interested in particular among all the different cardiovascular complications of the virus is myocardial infarction and ischemic cardiovascular disease. Particularly because despite the association between COVID infection and long-term complications in patients with a prior infection being increased risk of myocardial infarction or stroke, there's been a little bit of uncertainty as to whether this was secondary to inflammation-related plaque destabilization or related to systemic inflammation and increased metabolic demand with hyperexemia, which is considered a different mechanism that leads to myocardial infarction. And based on this question, we performed an initial investigation where using human coronary vessels, we were able to answer some of these questions. And we leveraged our access to coronary autopsy specimens from deceased patients with a severe COVID-19. And we were able to classify these lesions based on their severity of pathology from normal vessels to more advanced lesions being atherosclerosis. And based on the clinical data and also our access to autopsy report, we understood that these patients had pre-existing conditions that were making them more prone to severe COVID and outcomes. These patients also were enrolled before vaccination, where tissue was collected before this the vaccination was available. So we all these results will be pertinent to unvaccinated subjects. And some of these patients during the course of the disease had some acute cardiovascular manifestations being recognized as myocardial infarction or stroke. Some of them, unfortunately, died very acutely from the infections, while others recovered from the infection, but then due to persistent what we will call now post-COVID, they had complications, cardiovascular complications like this patient, like heart attack. And this happened almost 140 days after the first hospitalization and from testing positive for the first and only time after the first infection. These are the type of analysis that tissues that we have analyzed. As I mentioned, the pathology was very various, so this applies to a normal vasculature as well as to diseased vasculature. Atherosclerosis is the narrowing of the arteries that is the underlying pathology for a heart attack and strokes. And we performed an analysis that was very comprehensive and was based on spatial analysis of the presence of the virus in the tissue, where we classified the tissue from the arterial wall and the surrounding perivascular fat. We look at the cells and the quantification of viral RNA in these cells. And we found that the vasculature was really impacted with a higher number of copies from, in this specific case, in RNA encoding for the spike protein in the vasculature, including also viral RNA that is only expressed during viral replication. And there was a detection also in the perivascular fat, but not so much compared to the vasculature. And we also found, this is a very complicated plot, but it wants just to show you that there was no difference based on pathology, although the replication seemed to appear more in a pathology that will correspond to the early lesions developed in these coronaries. This is what we found. We also found, looking at the type of cells that contain the virus, that were in fact macrophages. Those are cells that infiltrate the atherosclerotic lesions, and they phagocyte lipids to clear the vasculature, and they also contain spike cells. And of course, we thought this might be just an epiphenomenon of macrophages engulfing the dying cells from infected by the virus, but we actually found that these cells also contain the spike cells and viral transcripts. And in particular, with subsequent investigations in vitro and in vivo that I don't have the time to explain now, we found that indeed the virus can infect these cells, although the infection is abortive and not being able to produce infectious particles. But nevertheless, this is sufficient to create an inflammatory state at the site of the at this vascular site, which is known to trigger cardiovascular events. This analysis will show, just very briefly, that macrophages contain the virus in the different pathology. Again, we found that this was particularly true in early stages of the disease, so what we call fatty streaks. And the number of cells that were infected, again, was higher in the vasculature and in macrophages contained in these early stages of the disease, where macrophages are considered to be more healthy cells compared to more advanced disease. So based on our access to a large number of patients, we decided to, and with collaboration, of course, with PolyBio, thanks to their support, we decided to leverage our existing biobank to perform an analysis of the vascular reservoir, but particularly as a reflection of it, of the immune responses that can drive cardiovascular disease. And we also will integrate in our data other data that were generated beyond PolyBio to give a broader understanding of what's going on. These patients that we have access are patients from pre-COVID. We have an enrollment that started in 2013 up to 2020. And then we have patients that were exposed to the virus, with some with documented PCR tests, some others that will report the diagnosis, and others that had an uncertain history. Maybe they were asymptomatic patients and they were never tested or never had the documented diagnosis. So to address this, and this is what we have done, we have analyzed, and we are still doing this analysis, is ongoing, carotid plaques, coronary plaques, and also we have blood samples. And in particular, to really try to understand better what the history of these patients was, we decided to partner with colleagues here at NYU who had developed a Luminous XA that is able to detect the exposure to the virus for up to 2 years after infection. And then we are performing bulk RNA sequencing. I will show you some of the preliminary results, and we will integrate our data that were generated in a subset of these patients for single cell analysis. So I mentioned that part of this is of our study is related to sequencing of these samples, but also we have plasma samples, and we use this Luminesque XA because we really face the uncertainty of this reported diagnosis. So using this Luminex serology that has been done in collaboration with Dr. Durr, we are able to distinguish patients that were exposed to the virus or that were exclusively vaccinated with a window that goes up to 2 years. So here you see the infected patients that were documented through this analysis, and also other in the assay. This was developed with clearly documented diagnosis or negative diagnosis as well. And in this case, you will see that these patients are patients that were exposed to the virus within 2 years. And here you have the pre-pandemic patients. And in this area, you have patients that were vaccinated, but have no documented or historical exposure within 2 years from the diagnosis. So based on this analysis, based on our preliminary deep characterization, deep RNA sequencing, we characterized the transcriptional profiles. And just at first glance, it was obvious that a lot of inflammatory pathways and genes were upregulated, and these were pertinent to interferon signaling, to TNF-alpha signaling, and some reactive oxygen species, apoptosis, a lot of the typical pathways that are involved with inflammatory response, that based on this analysis that was adjusted for covariates and is including the type of pathology, the comorbidities, and the history of the patients, there seems to be a persistent, localized at the vascular site, interferon response. So a persistent inflammatory state that we're trying now to understand whether it is related to a sustained vascular reservoir, or rather to epigenetic changes that were triggered by the virus, and that condition, regardless of the presence of the reservoir, the inflammatory response in the vasculature. So with this, I would like to thank PolyBio for supporting us in this endeavor, and I hope that we will conclude our analysis soon and will be able to share the final results with you very soon. Thank you.
Great. Good presentation, and we look forward to that data at the next symposium. All right. The next speaker is Michael Peluso. He is an assistant professor on the Division of HIV, Infectious Diseases and Global Medicine at the University of California, San Francisco, and he'll be talking about CHIME, the new ME/CFS program.
Hey, everybody. Can you hear me? You can hear me?
Yep.
Great. Great to see everybody. Thanks for the opportunity to be here. I'm going to talk about our plans to do some biological work in pre-pandemic ME/CFS. So as has been discussed in this forum before, our post-COVID program is called LINK. And we've been running this program since March of 2020 here in San Francisco. And, you know, in addition to sort of the background cohort, I think what really makes LINK unique is all of the sort of deep mechanistic work that we support via many collaborations with PolyBio, you know, that are supported by PolyBio, but also a lot of the tissue work, which has also been supported by PolyBio, our imaging program, to really be able to understand what's going on both immunologically and, you know, related to infections on a total body level in people with long COVID. We have, you know, been very interested in expanding that beyond long COVID for some time now. Last year, we and others wrote this article in Science Translational Medicine, basically arguing, which I think many people believe, that progress in understanding long COVID really should be translated to the study of other conditions that are suspected to be infection-driven or post-infection, and that have been really understudied and under-resourced for many decades now. And so, based on that rationale, we wanted to think about how we could expand the activities that we were doing and kind of leverage the infrastructure that we've developed for LINC to study ME/CFS. And the spirit of this is very much LINC was built upon a large infrastructure that had been developed for HIV, and so this is kind of trying to expand that into yet another condition. So our program that is generously funded by PolyBio to study ME/CFS is called CHIME. And I just want to acknowledge all of the work that two of our team members, Beatrice Wong and Brent Abel, have invested over the last year in helping to put together this program so that it can get to the point of being launched soon. And we're basically going to apply the LINC model in CHIME. So I'll just walk you through what our plans are. This is not fully set in stone yet. It will be next month when we're having our first meeting of our scientific advisory board, and so we're finalizing a lot of these details now. So the way that this is going to work is that the first portion of CHIME participation is going to all be done remotely. So a person will request contact from the study, fill out some basic contact information, and then have a informed consent discussion where we'll tell them about the study, review some basic inclusion-exclusion criteria, and then send them some screening surveys to get an idea of both their ME/CFS history as well as kind of their medical record documentation, understanding what records we need to collect. Then we initiate a medical records request and gather their medical records, and our clinical team basically puts together a summary of the full medical history for each participant. There will be some back and forth with the participant during this time to make sure that things are complete and we're not missing anything. And then, as is typically done in some other ME/CFS studies, then there is a review by our advisory committee to make sure that we're confident in the diagnosis, that the participants we think will be a good fit for the study, and to determine whether there are any major gaps in the history that we've obtained, or, for example, in the testing that needs to be done to inform what can happen at the initial visit. We're then going to have the in-person components of the study. So there will be an in-person baseline visit with some more questionnaires, with biospecimen collection, and with additional objective testing. All of the details of this are still being worked out. We're being very mindful of participant burden and issues of post-exertional malaise, for example. And then, the purpose of this study is not to repeat what has been done in other ME/CFS studies over the last decade. It's to really build upon that with the unique resources that we have here at UCSF. And so really we're going to be targeting this study toward people who are interested in and willing to do some of these more advanced procedures. In particularly our tissue-based procedures, we're going to begin with a focus on gut biopsies, and then our imaging procedures. And then we will have follow-up visits after the initial visit, but the details of that are still being worked out. We're currently assembling our advisory committee. We've identified 5 people so far for the advisory committee. We are waiting to hear from a couple of more people. And these are individuals with kind of deep experience caring for people with ME/CFS, studying people with ME/CFS. We're also going to incorporate 1 to 2 people with lived experience onto the committee. And this committee will really be very helpful to us in kind of providing scientific guidance to the program, and making sure our recruitment is on track. Currently, we're planning to do a combination of patient-reported measures of fatigue and functional capacity. There's a lot of interest in this FunCap instrument, particularly in the patient community, and so we're going to plan to incorporate that as well as kind of more traditional metrics of fatigue, assessments of post-exertional malaise, and some assessments of cognition. And then we have the ability to do a wide array of objective measures and are trying now to kind of achieve the right balance in terms of intensity and completeness. But we've stood up a lot of this infrastructure in the long COVID program, so excited to apply it to ME/CFS. So as I mentioned, this is still being finalized and will be finalized next month. A lot of the rationale for this program, and maybe I should have put this up front, is based on this very provocative work that John Chia down in LA has done over the last few decades, and work by others investigating viral triggers of ME/CFS. And so I won't spend a lot of time talking about this, but there's a robust and I think compelling literature about the persistence of enteroviruses in ME/CFS. And we're really looking to try to replicate some of this foundational work that has been done in a different patient population up here in San Francisco, and also applying some of the newer technologies, including RNAscope and our transcriptomics technologies in this condition. We're also going to really lean upon the collaborations that we've developed as part of the PolyBio consortium here. And so, people at UCSF like Tim Henrich and Henry Van Broeklen and Nadia Rohn will be involved in the immune phenotyping and the imaging and the virology. We're hoping to work with the Kiko at Yale on some of the immune responses and autoimmunity. John Weary and Mark Painter at Penn doing longitudinal T-cell work, looking at virus-specific T-cells. David Walt, who's capable of doing ultrasensitive measurements of various viruses and immune responses, and Michela Locci at Penn as part of the lymph node program. And this is really based on the rationale that we need to be looking in blood, but we also really need to be looking in the tissues. So as I mentioned, the intention is for everyone in this program to at least get a gut biopsy and PET CT imaging to understand what's going on with immune responses and potentially viral persistence in the tissues, and then also planning to expand to some of the other tissue-based procedures that PolyBio has supported. So last slide here. So we got IRB approval this fall. We opened recruitment on the internet last month. We've gotten about 15 referrals so far. We've begun doing consent calls and requesting medical records. The first advisory committee meeting will be next month. And then we're hoping to begin the in-person activities. This should be 2026, early in 2026. So I will end there and looking forward to providing an update on this at the next meeting. Thanks.
Definitely. Great job, Michael. I'm really excited that CHIME is moving forward. It's very exciting.
Thank you for PolyBio's support.
Of course, it's a hugely important study. So the next speaker is your colleague, Steve Deeks, who's a professor of medicine in residence at the University of California, San Francisco, and he'll be talking about VIPER, a long COVID diagnostic test validation program.
Thank you, Amy. Everybody. I'm going to give a slight update on some of the previous reports here. Just let people know that we're trying to make progress in building up this program, which we call VIPER. Just as a quick background, let's see here. As Michael just said, LINC, all the programs here that we're doing with these various post-infection complications is largely built on, a 20, 30-year experience that we've had studying HIV. And I got my start in this business back in 1993, working in HIV. And at that time, as most of you know, things were pretty dreadful. It was dismal. There was no biomarker, and we certainly had an antibody test and we had a CD4 count, which was helpful, but we really had no idea who was going to progress fast, who was not going to progress. We didn't really have a good sense of their prognosis. We didn't have any way to sort of engage industry on targeting the virus, because at the time it couldn't be measured. And so the only way that we could figure out whether or not antivirals actually did anything was basically give people the drug, cross our fingers, and see if it actually prevented people progressing to AIDS or death. Those were very complicated, big studies to deal with. As a consequence, industry engagement was pretty weak. There was massive investment at the time from the NIH and so forth, so we were progressing in terms of our understanding of the disease, like we are right now with long COVID. But without industry, it just didn't seem like we were actually going to get very far at all in terms of coming up with a treatment. And then something magical happened, right? We actually had this measurement, this diagnostic, the plasma HIV RNA test, which emerged in the mid-1990s, and it basically showed with great precision who was going to progress and who not, how fast they're going to progress. But most importantly, it gave companies the ability to target something and measure it. So a company actually could tell within 2 to 4 weeks of a small 10-person study whether their drug was going to work or not. And when that happened, the floodgates opened. And the late 90s, there were every single major pharma company was engaged. And billions of dollars were pouring in. And drugs were coming out left and right. And they continue to do so with 30, 40 different drugs. And it really all depends on the fact that pharma realized that they can actually very quickly, very efficiently generate a drug, get it across the finish line, and so forth. So this would, of course, be transformative for long COVID. I don't think any of us here thinks long COVID is going to be as easy. But we do believe that a biomarker that is perceived externally as having been validated and having gone through all the rigor that is necessary for a pharma to use these assays, for regulators to prove drugs based on this assay. If we had such an assay, things begin to move quickly. Now that was the 90s. All right. Now we have people who are on long-term treatment, and now actually the agenda is, can we cure people? And basically we're going back to the same problem. In the HIV cure space, the big problem is that we don't really know what we're measuring. That we know there's this reservoir of virus that's really hard to find. It's tiny, just like with, the SARS-CoV-2 issue. People on long-term treatment, there are these rare cells that contain the virus. Sometimes it's intact, sometimes it's not. We don't know exactly what we should be measuring. Should it be in blood? Should it be in tissues, and so forth? And so the Gates Foundation, now the NIH, is sponsoring a pretty massive, well-resourced network called Raven, that basically is a one-stop shop for people who are interested in this field to come share samples, use a central repository, get blinded samples, get blinded analytic standards, the spike stuff, and share the data centrally, have the data analyzed independently, do fresh versus frozen, do all this sort of validation stuff if necessary. And this is now inducing a whole new industry of companies that are getting engaged in HIV cure research. And it's this Raven work upon which we believe we should actually model the next step in this process, which is, of course, the Viper work. Now, a biomarker for long COVID. What does the field need? What do clinicians need? What do patients need? What does pharma need? What does the FDA need? Everyone really wants the same thing. They need a biomarker that's scalable, that's affordable, that's reproducible, that uses an easily accessible body fluid, basically blood, that it has performance characteristics that have been well established, that it actually has some prognostic value. Typically, does it actually correlate with disease cross-sectionally? Does it correlate with disease over time? And has all this been done independent of the group that's actually discovering and moving these things forward? And so Viper hopes to fill that need. Here's the current situation. We've seen some amazing work already in terms of different individual labs, accessing different cohorts. But at the end of the day, there's no consensus, there's no external validation. People who do assay discovery have skill sets that might not necessarily be the type of skill sets that's necessary to do this validation work at the level that we think is needed. These data sets are all independent. They're all being done on different samples. There's no merging, there's no collaboration, there's no synergies. But still, and as we've heard today, and as our team has actually also been involved with, these studies, which are ongoing, have some value. We've, the Link team has supported a number of these types of exploratory studies, and you've heard about ongoing work already today. But again, at the end of the day, these studies are probably not achieving the level of impact that they could because the assays are not undergoing independent qualification, validation. These are convenience samples that we basically try to fix retrospectively. Everyone uses different definitions of the clinical phenotypes. Analysis is not centralized and so forth. All right, so we mean to fix this with Viper, in which all the data that will be generated will be shared, will be analyzed independently. It will allow for deep phenotyping of the patient population with support, of course. We could do repeated measurements because we're going to have longitudinal samples. Again, data will be managed independently and so forth. So that's the intention, the Viper, that we are now slowly building up. And of course, as we've heard today, the low-lying fruit here in terms of coming up with a potential biomarker that could have that transformative effect in long COVID like it did in HIV. It's just a measure of the virus persistence. The rationale for going after the virus is sort of the first step here is pretty straightforward. At the end of the day, this is a disease that actually is started by the infection. And now we've seen multiple reports that the virus can persist. It's sometimes associated with long COVID, sometimes it's not. We think it is more readily found in gut. We're not sure about other tissues. When we do find it, it's sometimes associated with local tissue damage, so forth. So clearly, this is where to start. This has been a central part of the PolyBio brand and collaboration since day one. And so we've actually begun, as we build up Viper, as we raise the support that we need to do this in the best way, we began to do some preliminary work. We are working very carefully with this Raven team, which has deep connections in the blood bank world, where a lot of the expertise exists for the kind of work that needs to be done. Michael and I on our work in HIV, supporting some studies to figure out exactly within whole blood, where is the virus. It turns out actually that even though we end up measuring most of the virus in plasma, it tends to accumulate and is stored on platelets, which tends to be it's a much better, it's a much more enriched source of the virus, particles at least during long-term treatment. So no one's really sort of asked this in a really rigorous manner in the context of SARS-CoV-2. And so we, in preparation for Viper, are doing some of this really basic work, trying to figure out exactly, where the virus particles and various different things might best be found in blood. And we're also at the same time asking questions whether or not it's best to do this with fresh versus frozen sample and so forth. The kind of work that is really necessary to sort of jumpstart this type of collaboration. We've, Tim Hendrich, who's going to be speaking later, had a really beautiful paper with Michael and others in Science Translational Medicine arguing that the gut might be the best place to find SARS-CoV-2. And we heard some earlier presentations along these lines today. And so basically in Link, we've actually built up with PolyBio support the capacity to look deeply into the gut, both in rectal tissue and sigmoid, and in some people as far up as the ileum. But we haven't stopped there. we have, in the context of LINC, also now beginning to look at lymph nodes. We're doing a bone marrow biopsy program, pretty intensive stuff. We've already enrolled 30 people, which I just find amazing. If nothing else, it just illustrates how incredibly engaged our patient population is to undergo either bone marrow biopsy or lumbar punctures. I mean, it's a big ask. But we've done many of these procedures. You'll hear about the PET work that Tim is doing a little bit. And we've even started an interesting collaboration looking at endometrial biopsy and so forth. So we have all this will be available within the Viper ecosystem to try to also help with basic discovery, basic characteristic of where these biomarkers might perform best. Shannon Stott in her presentation described some really elegant work using these sort of gold standard, highly curated samples in which the target, for example, the spike protein, whatever it is spiked specifically into plasma samples to the degree that investigators know exactly how much is there. These are the types of things that are necessary. This is hard work to do, and we intend to actually build up these panels for external use. And we'll be way beyond the virus, right? Virus is just the first generation stuff, but we all know that it's probably not going to be that simple. We need to look at inflammation, immune dysregulation, and a whole series of other things can be done with these samples. All that data can be fed eventually into the same system and see if AI and machine learning can figure this out for us. So with that, I'll turn it back to you, Amy. And again, thanks for your support.
Of course, Steve. We're going to make Viper happen. All right. Okay, our next speaker is David Petrino. He is the Director of Rehabilitation Innovation in the Mount Sinai Healthcare System, and he's the Nash Family Director of the Cohen Center for Recovery from Complex Chronic Illness, or CORE, a clinic where I work with him to innovate care for patients with long COVID and other infection-associated chronic conditions. And David will be talking about combination antivirals in the treatment of long COVID.
Okay, so it is a pleasure to be here. I always love PolyBio Symposium Day. So many amazing talks. And I'm going to be, speaking about the use of combination antivirals for treating long COVID. I just want to sort of touch on something that, a few people have said now at this point, but like, what are the things that we need? What are the things that people with long COVID need from the research world? We need multi-therapeutic approaches, and this is a paper that was led by Amy Proal, and many of the PolyBio team were listed on this paper, and really what we've tried to point out in this paper is, in order to target reservoir that is going to be very resilient and very hard to kill off in people with long COVID, we're going to need multi-therapeutic approaches. We're going to need long doses. Short doses of single drugs just will not get the job done. If we know anything, we know that. We also, beyond this, we also need to be learning from everybody. We need to be reaching out to clinicians who are out there figuring stuff out for themselves, and listening to patients who are saying, I go and see this doctor in this location because he seems to be onto something, she seems to be onto something, they seem to be onto something. And that's how we learn. That's how we get sort of signal in the noise to try and think about which therapeutic combinations to treat. And most of all, we just need to move fast. Patients can't afford the time, nor can they afford the out-of-pocket expense of just paying for off-label drugs endlessly. We need to get some of these interventions FDA approved and get out there. So it's with this sort of mindset and this level of urgency that we're sort of pushing into the data that I'm going to be presenting today. I met Skip Pridgeon, who's pictured here. He's a physician in Tuscaloosa in Alabama, and I met him because some of our patients were like, yeah, have you heard of this guy? Dr. Pridgeon. He's doing a novel combination of different antiviral drugs, and it's really helping a lot of folks with long COVID. So Amy and I got to meet Dr. Pridgen, and he sort of told us about his approach. He told us why he's pulling these drugs together. So it's a combination of Celebrex, which is often thought of as a non-steroidal anti-inflammatory drug, but in its ability to block COX-1 and COX-2 receptors, which is what these anti-inflammatory drugs do, it also blocks these enzymes that are produced by COX-1 and COX-2 receptors that are used for viral replication. So it blocks viral replication. It helps to sort of disrupt membrane surrounding the viral capsid, and suppresses SARS-CoV-2 activity as well. There was a study showing early on that in acute COVID, Celebrex could be used to suppress SARS-CoV-2 activity. So he was combining this with, of course, Paxlovid, which we all know inhibits is a protease inhibitor for SARS-CoV-2. So it doesn't exactly eradicate SARS-CoV-2, but it stops it from replicating. And then valacyclovir, which has a sort of broad action on many different herpes viruses. And it does that by inhibiting viral DNA synthesis. And as we have seen, like myself, in our collaboration with Akiko Iwasaki's team, and many, many other researchers have published this phenomenon that we see on COVID who have reactivated herpes viruses, such as Epstein-Barr virus, HHV6, etc. So it seemed like a very clever, common sense combination of antivirals to try. But Skip was also using these drugs for a long period of time. So I spoke with Skip and I said, can we have a look at some of your data? And he was absolutely forthcoming, and he said, here's some data that I have so far. And you'll see it's a small N. It's just, he approached 27 people. And what I really respect about this data set is often when you speak to people who are trying things out in the wild, they'll say, I've treated 1,000 patients. And you say, okay, can I see your data? And they'll show you data from 30 patients. And I'm like, but you said you've treated 1,000, what's going on? In this case, it was a small N, 27 people were approached. One of those people, Skip wasn't really sure about their long COVID diagnosis, so they weren't offered the interventions. Everyone else was offered one of two interventions, a combination of Celebrex and Valtrex, or valacyclovir, and on its own, or they were allocated to this group where they got the Celebrex, the Valtrex, and they got the Paxlovid. So we got to have a fairly good balance of 13 people purely on their own, selecting one intervention or the other. And we got to see quite a balanced group. Even in both groups, one patient dropped out of either group, which left us with 12 people who completed. And a little bit further down the track, we'll get into this, but also from the group that tried the IMC only, 3 of those, after trying the IMC only, actually said, you know what, I want to try the other treatment as well. So we even got to see some data from a crossover. So let's actually get very concrete about what the protocols look like. So the people who received IMC2 only, that means that they received a combination of 1,500 milligrams of valacyclovir taken twice daily and 200 milligrams of Celebrex taken twice daily. And they took that for roughly 120 days. In the IMC2 plus Paxlovid group, it went a little different. So they started off with that same regular dose that we were seeing in the IMC2 only group. Then around 2-3 weeks in, they moved into a 15-day course of Paxlovid. During that 15-day course of Paxlovid, they were taking the same dose of Celebrex, but they actually dropped the valacyclovir dose down from 1,500 twice a day to 750 twice a day to reduce strain on kidney and liver as they were taking the Paxlovid. And then after the Paxlovid course was over, they went right back to taking that same dose that the IMC2 group were doing. So that's the dosages and the sort of protocol that was going on. Now let's think about outcomes. Now, this was a clinical case series, so we weren't, Skip wasn't exactly using this huge battery of outcomes, but he did ask patients using questions using the patient global impression of change. Now, this is actually a really well-validated way of asking a patient, do you think that this thing made a difference for you? And you can go from no change at all the way through to a great deal better and a considerable improvement. And what we see is that at 120 days, so as soon as they finished that 120 days of taking the drugs, the people who took IMC2 only, they were scoring on average a 4.8. So moderately better, a slight but noticeable change. Whereas the people who took IMC2 plus Paxlovid, they were saying, I am feeling a great deal better, considerable improvement that has made all the difference. And what was even more exciting was when we look at follow-up at different time points. So at the end of the 120 days of treatment, then at 305 days from the commencement of the protocol, all the way out to 730 days from the commencement of the protocol. So more than 600 days post-treatment, people who took the IMC2 plus Paxlovid were still feeling better. This tells us it's a durable effect, which is very, very exciting, because often, as we all know, some people will get improvement from a clinical protocol, but then sort of crash back down to earth. Also, just want to name the fact that in this short case, we have a case series in a case series. We have these 3 patients that did the crossover. And what we saw was they completed 120 days of the IMC2, and they were scoring, like a 4 and a 5 on the PGIC. On average, after completing the IMC2 only arm, on average, these patients waited about 108 days before starting the IMC2 plus Paxlovid. And as you can see here, at day 120 of IMC2 plus Paxlovid, they're all scoring a 7. We see one person scoring a 6, but the other 2 scoring a 7 at 305. And at 731, everyone's back up to a 7. So really encouraging results here. So next steps, this is a case series. This is not placebo-controlled, this is not randomized, and it's a small sample. So this is not generalizable to every single person with long COVID. We need a large-scale randomized controlled trial. Pridcor, which is the company that produces IMC2, is going to be launching a clinical trial called SHIELD in 2026, and our team will be launching that clinical trial, so we're really excited about that. But I just wanted to share some preliminary data that I hope gives everyone, an understanding that some of the things that we're doing with multiple drugs and longer time courses really do have the capacity to work in a subset of patients. So really excited for what's to come. I want to acknowledge all the wonderful people I get to work with and all the wonderful organizations that support us. And thank you all for your time.
Great, David. Thank you. I'm excited to be part of the SHIELD trial when it happens. Okay, our next speaker is Beverly Peng. She is a postdoc in the Chris DuPont's lab at the J. Craig Venner Institute, and she'll be talking about mapping the landscape of endometriosis tissue.
Okay, so my name is Beverly Penn, and I am from JCVI, and today I will be going over my work using cell type annotation for endometrial tissue. So the endometrium is the lining of the uterus that sheds and regenerates during each menstrual cycle. And endometriosis is when tissue that is similar to the endometrium grows in other places in the body. So this can be close areas such as the fallopian tubes and ovaries, but can also be further away, such as in the bladder and intestines. And endometriosis is a chronic condition with varying levels of pain and symptom severity. And it is due to this variability that makes it difficult and invasive to diagnose and retrieve samples surgically. And among many treatments include surgery, anti-inflammatory or hormonal drugs. And there really is a lack of RNA-seq studies for endometrial tissue. And it's really unacceptable considering half the population have endometrium and about 10% of them could be affected by endometriosis. So I'm grateful today that I'm able to present on the data that is from the University of Pittsburgh from Dr. Nicole Donnellan to hopefully advance this research. Okay, so this is the general pipeline for single cell and single nucleus analysis. For the purposes of this presentation, single cell and single nucleus are considered the same thing. And then today I'll be going over the bioinformatics analysis, specifically about the quality control, the clustering, and the cell type annotation. And the main package that I used was the Python ScanPy. Just a little bit background on the wet lab things. I'm not a wet lab person, so I don't know much about this, but if you want to know more about our tissue processing, then you can, I can get you in contact with our lab manager, Mark. Okay. So after going through all of our pre-processing, we get this UMAP and then we run Leiden clustering. And we identified 20 Leiden clusters. So Leiden clustering is a community detection algorithm that groups individuals that are densely connected to each other versus all other groups. So an example of this would be biological network analysis. And an example is protein-protein interactions, where we can assume proteins within the same community are more likely to interact with each other. And this is like the basis of biological pathways. And then these other colored UMAPs show different, or I guess how our covariates are integrated together. And then taking a quick note on our number of samples. So we have a little bit uneven between our endometriosis samples and our control samples, which you can more clearly see in the endodiagnosis shaded UMAP. But this figure does form the basis of analyzing the differences in cell type proportion between the control and endo. So you can kind of see, or we can predict about which Leiden clusters would be more enriched in Leiden and endometriosis versus the control. Okay. So now that we have our 20 Leiden clusters, we want to annotate these cell types. And we're really lucky that last year, the Human Endometrial Cell Atlas was published. So this is known as HECA. And this paper did a really good job at defining the broad cell types and then going more granular. So we can see the main cell types, which is epithelial, immune, mesenchymal, and endothelial. So this study is what we want to replicate with our data. And now that we have, so we use HECA as our reference, and there are a couple steps towards our cell type annotation pipeline. So I helped co-develop the NSForest and FRMatch algorithms. NSForest is a machine learning algorithm that helps us calculate the minimum set of marker genes for cell type classification. So this is run per data set. So we ran this on the HECA dataset, and we got 1 to 3 markers per cell type from the HECA dataset. So we have about 60 marker genes that we're using to define our feature space. And then what FRMatch does is it subsets both datasets, so the query and our reference. It subsets that to our new feature space, and then it matches them and provides p-values. And then kind of a visual representation of what's happening is in the bottom left. So we can see the HECA markers for arterial are these two, and then you can kind of visually see how it is the combination of these two markers that makes us think that Leiden cluster 14 is arterial. So if we repeat the same process as before, for all of our Leiden clusters, I picked out the ones that we were most confident in. And then we colored them according to the broad cell types. And then you can see on our UMAP that if we map our high confidence over to our Leiden UMAP, we can see that they do break down into the same broad cell types as the HCCA data. So I think that's a promising sign that we're in the right direction of annotating these cell types. And then the final step of this analysis is proportionality testing. So the main question we're trying to answer is there a difference in cell type proportion for endometriosis samples versus the control? So here we take the FR match annotation from the previous slides, and then we calculate the fold change. So this is the difference in proportion between the control versus endometriosis nuclei. And then on the right, we see that for each Leiden cluster, we are plotting the proportion of nuclei that were grouped within each endometriosis and control samples. And then it's generally shaded by the strength of the p-value, so darker means higher significance. And even though we only have a sample size of 28, this is still pretty promising that we do have some significant p-values. And specifically, the most significant is Leiden cluster 7, which Chris DuPont mentioned earlier, we identified as estromal MMPs. This is pretty cool because it's associated with cancer progression and metastasis. So there's like a clear, connection between that and endometriosis, which is the abnormal growth of endometrial tissue. So we hope from this study that we're able to publish the results, and then we can hopefully enroll more participants and continue to sequence more data, and then we can increase our power for this study. And then for next steps, we want to finish annotating the rest of the unassigned Leiden clusters. And hopefully we can define these as novel cell types. And then with help from the HCCA and NS-forest markers and annotation experts, hopefully we can properly categorize them and then publish our results as a new HCCA adjacent atlas. I'd like to have a quick thank you for PolyBio, University of Pittsburgh, for giving us the samples, and my team at JCVI and NIH who have been helping me with this. And I'm free to answer questions in the chat.
Awesome. Thanks, Beverly. Great job. Okay, I'll introduce our next speaker. It's William Eimer. He is an instructor at Harvard Medical School, and he'll be talking about viruses as drivers of Alzheimer's disease pathology.
All right. Looks like it's sharing. Amy, thank you for the introduction. Thank you for having me at the symposium. Some of you may be wondering if I accidentally wandered into the wrong symposium, but no, I promise this connection between viruses and Alzheimer's disease will loop back around to COVID in an important way. All right, so for a brief description as to where my work originates, Alzheimer's disease is a progressive disease. It starts years, decades before we start seeing symptoms. And the classic idea is that we see amyloid. Amyloid leads to tangles, and tangles leads to neuroinflammation. Neuroinflammation leads to the damage that we see in Alzheimer's disease. And this is called very heavily based on the amyloid cascade hypothesis that was established decades ago. And this has been the primary junction point for where pharmaceutical industries have entered into trying to help patients of Alzheimer's disease. They look at amyloid, say, hey, that's the bad guy, and then we try to eliminate that. We have 2 FDA-approved drugs out there right now that came in the last 2 years, but they have very modest success. And what this has caused the field to do is backpedal a little bit and say, okay, if amyloid is not the sole reason for why we're seeing the progression of Alzheimer's disease, where other influences could be. and pathogens has popped back up. Even Alzheimer's himself, who discovered the disease, noted that it looked similar to neurosyphilis at the time, and that's over 120 years ago. So there's been a lot of work. Ruth Itzhaki has been pivotal since the 90s of looking at viruses in Alzheimer's disease. And it's really blooming right now, this idea that Alzheimer's could be driven by microbes. And viruses are probably the top of that list. But there's still a black box in connecting between pathogens and Alzheimer's disease. We know amyloid beta is involved, we know tau is involved, but we don't know exactly how. Are the plaques and tangles we see an active part of the disease or a byproduct, or are they involved in something else? Looking at viruses and its impact in Alzheimer's disease, one of the best studies out there is from 2018, and it was a retrospective case study looking at Taiwanese patients over the course of nearly a decade that they knew had positive HSV-1 infection. And they broke it down into 4 categories. We had those that don't have HSV, those that have it but never treated it, those that have it and treated it, and those that, well, those are the big 3. So what they found is that if you had HSV-1, your risk went up 2 to 3-fold. If you treated it, it went back to baseline. It went back to the same risk factor as those with HSV. Now, the important part here is it didn't cure Alzheimer's. We didn't get rid of Alzheimer's because we treated HSV-1, but it brought it back down to baseline. This suggests that there is a microbial component to Alzheimer's disease. Now, when we look at HSV-1, which is the basis for herpes infections, and we look at COVID, they do share some important commonalities. They're enveloped viruses, they're both neurotropic, and they both rely on surface glycoproteins for infection. HSV-1 has the glycoprotein spikes. We talk about the spike protein on COVID and its interaction with the ACE2. These are key components, and it's also the core area where we think that there's been interaction with amyloid and tau. So the interesting piece is this is where COVID and Alzheimer's start to overlap. In 2022, right in the middle of the pandemic, there were some cohort cases of people that they had tracked through military service. So they knew whether or not they had Alzheimer's disease because they had all been tested and scanned and everything. And they managed to bring some of these people back in who had been positively infected with COVID. And some of these people who ended up unfortunately passing, they had plaques. They had amyloid plaques in their brain. They looked like dense core or diffuse core plaques. And as you can see in the red, in the Columbia cohort, 10 out of 10 of the COVID-19 patients came back positive for amyloid stain. And last year, when they turned their view to tau, they found that with N being the non-infected, with the acute infection, they started to see increased phospho-tau signal inside of neurons that's not there in people that were infected. And even in people that had survived and recovered from COVID, there were signs of phospho-tau increase in these patients' brains that similarly look like the neurofibrillary tangles that we see in Alzheimer's disease. So the research and the overlap between the two over the last 4 or 5 years has really accelerated. We're seeing commonalities in the immune response, in vasculature changes. I just showed you the examples of amyloid beta and tau pathologies. And they've even seen EEG shifts. So this is where our work here comes in. We're of the opinion when we look at Alzheimer's disease and amyloid and tau that they're actually antimicrobial peptides. They're a natural part of the body, and they're meant there to fight infections. So we did a look with mice and in vitro culture systems, and we infected these systems with HSV1, and we saw amyloid forming plaques around and incorporating the virus. You can see here that even after 15 minutes of exposure between a virus and synthetic amyloid, we're seeing these tails of aggregated amyloid starting to form. And after 2 hours, they're starting to agglutinate and connect all these viruses together. But antimicrobial peptides go beyond just being able to have nets and to capture. There's a whole host of different modifications that antimicrobial peptides can do. They can regulate the inflammasome. They can be part of cell signaling. They can promote and depress different responses to infections in cells, both immune-based cells and non-immune-based cells. And we wanted to take what we had learned with amyloid and apply it to tau. Could tau actually be an antimicrobial peptide just like amyloid? And what we found is that when we pre-treated our neuronal cultures with a synthetic tau and then exposed them to HSV1, it decreased the infection. We had fewer viral plaques. And not only did we decrease the infection, but the infections that did actually make it were smaller. The plaques didn't grow as much. When we went and looked at the cells and the virus themselves, similarly, we could see tau aggregates starting to form around the capsids. Now, I want to just quickly clarify that this is capsids. These are the inside parts, because this is what gets ejected into the cells from the virus, not the envelope. The envelope was with amyloid. We can see even here, we've got immunogold of tau sticking to the surface of the capsids. We analyzed our cultures looking at this kind of spread and progression of tau, and we broke it down into regions. This is the infected region, this is the near infected region, and this is the region of our culture that didn't show any infections and was away from it. And interestingly, what we found was that the infected cells, as we anticipated, increased phospho-tau signal. But there was actually an increase in phospho-tau signal in the uninfected cells that were right next to the infection. And there was this very strong correlation between the amount of viral signal in the vicinity and to the signal of uninfected p-tau. And what this implied was that p-tau was actually being released and taken up by neurons in advance of the infection itself. So we devised this experiment where we added a tagged amino acid so we could see where peptides and proteins were going. We infected the top culture, and then we had pores that were small enough for proteins to go through, large enough for proteins, but too small for virus. And what we saw was that the healthy uninfected cells in the layer below the infected was taking up these tagged phospho-taus in preparation for a potential oncoming infection. So where this leads us is that we have this growing evidence of a strong involvement of not just virus, but we've seen fungus, bacteria, and parasites, and their ability to influence and possibly direct neurodegenerative diseases. Our example is Alzheimer's disease, but with the plaques and phospho-tau that we've seen in COVID, we think this is also directly involved. And our amyloid and tau antimicrobial hypothesis plays into this idea of an innate immune ecosystem in the brain that's really kind of undiscovered and untapped. And we hope that by continuing to unravel this, we will understand better not only how it's working in relation to Alzheimer's disease, but anything that makes it into the brain that is triggering the immune system. Is if there's this undiscovered immune kind of play out that's not just either microglia or neutrophils, we need to understand it so that we can better treat the infections and better understand how things that may be causing the memory loss or the haziness or other low-level inflammatory results that may be driven by this amyloid tau immune process can be interrupted and managed in a way that's going to help patients both for acute infections and chronic infections. So I'd like to acknowledge Rudy Tanzi and my previous mentor, Robert Moyer, and MGH and Amy for having me. Thank you.
Of course, well, it's amazing research with such huge implications. So thank you for sharing it. Our next speaker is Tim Henrich. He's an associate professor of medicine at the University of California, San Francisco, and he'll be talking about the long COVID tissue biopsy program update.
Thank you, Amy. It's a pleasure to speak again here at the PolyBio. I'm going to give a quick update the next 10 minutes on the LINC tissue biopsy program. As you know, LINC here in UCSF, as both Steve and Michael have discussed, follows over about 11 or 1,200 patients longitudinally, and it gives us an opportunity to really do a deep dive, both in tissues and also in peripheral blood in the setting of clinical trials, interventions, imaging studies, et cetera. So really provides us a really amazing opportunity. We really focus mostly on gut tissue as well as bone marrow, based on some initial imaging findings we had with our PET program, showing that there was an increase in T-cell activation inflammation post-COVID within these compartments. And I just wanted to thank everybody up front here in the LINC pathogenesis team who made a lot of these slides and data for us today. So I think just briefly to say what I think is now obvious to most people, that long COVID is a tissue-based process. Most of our study has either been in the peripheral blood when we think of pathogenesis and biology, but really this is a deep tissue disease. As we previously showed that we could identify SARS-CoV-2 many years or many months after initial infection. Now, this was in people before there was a lot of reinfection, et cetera, which we're now seeing quite a bit of in our current cohort, which is clearly providing some challenges when we're looking at viral persistence in tissues. But we did see myeloid-associated viral persistence of both single and double-stranded SARS-CoV-2 RNA in gut samples in our initial cohort. And these were all in people that were reporting long COVID symptoms. So far, we've now processed, I think, over 100 gut samples, focusing on treatment responses in clinical trials. We've looked already in 17 or 18 people before and after the Outsmart monoclonal antibody study that is completed, and also about 18 now or so pre and post Incitrovir trial, which just wrapped up a few months ago here at UCSF. So we really have this opportunity to look before and after and do a deep dive in the setting of trials. Just to show you some cross-sectional data of our initial look at people with long COVID versus fully recovered, 43 and 13, fairly well matched cohort for gut biopsy program. But what we did see interesting, and these again are gut samples that were collected later temporally in time than that initial publication. And what we're seeing is actually two different patterns where we actually see double-stranded and single-stranded RNA and epithelium in people that were more recently reinfected, which is actually quite common now in our cohort. In fact, most people are reinfected at least on a yearly basis, or so I think, at least from what we've seen so far in blood and tissue. But as we look farther out, we're seeing a similar pattern where we do see some lamina propria, likely myeloid-based persistence over time. When we look actually at gene analyses in our gut cohorts, we actually see an interesting pattern when we look at gene transcripts in bulk gut tissue, and that's actually we see an increase in certain innate immune response. We see interferon signaling, we see some IL-1 receptor, we see some other inflammatory markers, but we actually also see a dramatic downregulation in many immune response genes that are responsible for either attracting immune cells into the tissue, granzymes, for example, HLA expression for presenting antigen or for clearing those infected cells, which to us suggests that there is a perfect storm of inflammation, but also decreasing the capacity for the immune system to clear virus in that pro-inflammatory state. Just to show you a heat map here, the little red asterisks on top are participants where we found RNA using RNAscope or other methods within our cohort. And you can see in the orange clusters is our fully recovered cluster versus blue on the top here, which is long COVID. And you can see that there are clear differences within these gene sets. Again, upregulation in some, but not all people with long COVID in inflammation and downregulation, relative downregulation in setting of some of these adaptive immune response gene pathways and genes related. And so, but again, not a huge, again, but we see kind of infected virus spread throughout. One thing we do not see is we actually do not observe RNA in fully recovered tissues thus far. Granted, we have fewer of those in the gut samples, but we have been unable to identify so far in fully recovered gut tissues, at least in this cohort. We are now moving on to, we've performed digital spatial multi-omic analyses as well as single-cell RNA-seq within these gut samples. And I won't go through the details here for time, but essentially we're seeing that there is this complicated interaction between certain immune cells like macrophages, which may actually be increasing inflammation, but other lymphocyte subsets that are actually acting as regulatory cells, which is inhibiting adaptive immune responses from clearing virus within some of these tissues. I next want to actually talk about our bone marrow biopsy program. And this is really fascinating because we actually saw there was quite a bit of bone marrow uptake on our T cell activation imaging, lymphocyte imaging tracer that we published a few years ago. And so what we've done is actually brought in now 30 people, about half with long COVID and fully recovered, trying to get a mix of demographics within each of those cohorts for good control. And we do bone marrow aspirates as well as core biopsies in the participants. And I just want to thank the participants up front. I mean, this is not an easy procedure to do, but we've really had quite a bit of success and participation, and want to thank everybody, not just the oncologists and our bone marrow biopsy staff, but obviously the participants themselves. This is just an example of when you look superficially at some of the marrows from people that are with long COVID LC here in purple or fully recovered in tan, although now we have 15 and 15 or so. You see that they look somewhat similar. There's nothing hugely strikingly different on that superficial level, but when we take a closer look at these are, remember, all post-COVID participants, we actually see both lambda and captor-restricted B cell proliferation within the marrow. Now, whether this is just background signal or this is related to either viral persistence, ongoing viral infection, unfortunately, one of these participants, our 69-year-old female, actually, this was diagnosed as a CLL, a chronic lymphoid leukemia, unfortunately, in this cohort. So we're now trying to understand what the relationship between this SARS-CoV-2 persistence and also inflammation, immune response, and hematopoietic cell development within the marrow compartment. So initially, when we looked at bone marrow aspirates using qPCR, we used the metagenomic next generation sequencing. We used nCounter, which is not a very good assay for viral detection. Unfortunately, that's a different story altogether. We actually did not see any definitive detection of SARS-CoV-2. And we also looked at both viral and protein as well in the core biopsy sections and did not see any protein production through our clinical lab. However, we now already 2 out of 6 long COVID cores. We don't see this in the fully recovered. We're now seeing SARS-CoV-2 RNA in multiple sections, so it's not subtle. Every section you cut, you can see multiple cells that are infected within the bone marrow. We're now almost completing this to see within the 30 how extensive this is this double-stranded or single-stranded RNA. But there's certainly persistence within the marrow compartment. We do a lot of negative controls on each slide, et cetera, for gating out potential noise. When you're just looking initially, some of it are smaller cohort. Again, we're now doing this on a much larger scale. There does seem to be some clustering within those fully recovered and long COVID, especially in interferon-stimulated genes, excuse the typo, and other innate and adaptive immune-related genes that we see as well. So there definitely seems to be some differences. What we also saw, interestingly enough, in that people both with long COVID and fully recovered, that we saw hyperexpansion of B cell clones, and some of these may actually map to SARS-specific sequences. So it looks like there is a SARS-specific B cell clonal expansion within the marrow, and this is something that we're obviously ongoing and looking at as we go forward. And you can see here that this participant was also floridly positive for SARS-CoV-2. Oh, that was a fully recovered. I'm sorry, that is a typo. I apologize. So I think our overall hypothesis here is that SARS-CoV-2 evades innate and adaptive immune responses in tissues to viral persistence, but it still induces chronic inflammation. And I think it's essential to boost tissue-based immune recognition and removal of these cells, which brings us into our Interrupt-LC trial. I'm not sure if Michael had a chance to talk about this yet, but boosting CTLs and NK cell activity, and we just enrolled our first participant. And we are officially open. So if interested in the San Francisco Bay Area, let us know. We are now enrolling for our open label IL-15 superagonist trial to enhance CTL, cytotoxic T cell, and NK cell responses to try to purge viral reservoirs that we are seeing. And the take-home message, just one last point, this is a tissue-based disease. We need to be looking at the tissue when we are looking at interventions, very much like we do with other viral infections. Now, that does not mean that we should not be looking for biomarkers in the blood. We should absolutely be doing this because it is a challenge, obviously, to do these types of tissues, especially in a larger cohort and in larger clinical trials. So just want to give Viper a plug and some of the other work for biomarker development. Tissue is the key, but we also need to have a window into the tissue through the periphery. And with that, just want to thank Michael and the Link team for making all the pathogenesis studies possible.
Wow, Tim, such great data. It really does show why you need to study tissue in order to understand the long COVID disease process.
And I forgot to turn on my camera. I'm very sorry. So I am here in person as well. That's my fault.
No problem. Okay. Now we have our next speaker, who's Frances An Lee. She's a professor at Emory University School of Medicine, and she'll be talking about use of Mensa to identify the active immune status for long COVID and other chronic infections.
Well, I just wanted to talk about Mensa, and I'll just talk to you briefly about it because I think I only have 10 minutes. I have some other slides about Lyme. I don't know if we'll have time to go into a chronic Lyme story as well to kind of show that this method could also work. But let me just give you the kind of the updates here. If I can move my slides. These are my disclosures. We're trying to commercialize this diagnostic, just so you know. So the basis of this diagnostic, for those of you who may not know our strategy, basically what we are very interested in is as a plasma cell immunologist. So I was really excited about the last talk with Tim, who talked about bone marrow cells and all those plasma cells in there, because that's what we really focus on and would love to talk more about it. But nonetheless, the B cells get activated in the lymph node, then basically differentiate. And when they do, you see new plasma cells forming and they dump into the bloodstream as they migrate into the blood and then go into the bone marrow sites or long-lived tissue sites, for example, other sites like the gut or what other places that it may be. And we identified a long-lived plasma cell compartment. But we've been really interested in capturing what's happening in the early phase, these early minted ASCs and these blood ASCs, and what do they look like? What are they all newly generated? And this is just some data that shows that naive cells are really all nucleus, but the ASCs that we find in the blood are antibody secreting cells, is what I like to call them, because they're not mature plasma cells and they're not purely blasts. So I just like to call them antibody secreting cells or ASCs because they basically are here. And they're very different compared to what we find in the marrow spaces. They're much more mature, and you can see that there's a lot of endoplasmic reticulum. There's a lot because they're antibody factories and they're making lots and lots of antibodies. And so they have a lot of mitochondria because secretion of antibodies is a high energy state. So early on, it looks a little bit different than it is early. They look like blasts, but they have blasted is really the bottom line, because all the cells that we find in the blood compartment are Ki-67 positive, so they're newly generated. So what we've done is we've capitalized on this specific feature of these early minted ASCs, and what we can do is use it as an immune snapshot of what's wrong with you today to understand the viral drivers or whatever, bacterial drivers or whatever other drivers. And so we've been very interested in understanding this process. So we agree that long COVID is a tissue-based disease, but the bottom line is that you can probably use these biomarkers that we've identified in the blood compartment to tell you what's stimulated in the body at that moment. So what we do is this technology is called Mensa. We media enrich with newly synthesized antibodies, our little smart fluid. And what it does is separates out the plasma from all the cells. So these newly generated ASCs in response to what's wrong with you today, secrete antibodies, we capture that, and then we basically look at it on, scan it on whatever we want. Luminex, VeerScan, Mesoscale, you name it, you name your favorite ELISA platform. Or some sort of antibody platform. So the antibodies in the plasma or the serum tell you what's wrong with you today. The ones that are in our Mensa the ones that are in the plasma and the serum tell you what's wrong with you historically, and the ones that are in the blood compartment from the Mensa tell you what's wrong with you today. So this is today's news. And this is some data in COVID. So in COVID, when somebody came in with the infection, their Mensas were positive, and then it goes down to a negative value. You get vaccinated to RBD or the spike protein, it elevates, comes back down. If you get boosted again, goes back up, comes back down. And then if you get infected here, I think this one was the Delta strain or Omicron for this patient so we follow this person over like 850 days, and you can see it goes back up. Whereas a serum, it goes up, comes up, and stays up. I don't have the responses to the nucleoprotein, but the bottom line is those only go up with the infection and not with the vaccine. So that's basically what this data shows you. And so using this approach, what we said was in long COVID, can we see if there's any other drivers that are basically causing us to understand what's the trigger for those patients? And can we capitalize on our Mensa technology? So we used the VeerScan, and we used COVID recovered or fully COVID recovered patients. And then we used a long COVID or past patients here. And we took these individuals and looked at their Mensas and looked at all the viruses. And what you can see here, there's more red than there is in the COVID recovered to identify these viruses. This was like our discovery platform trying to look through linear peptides of 149,000 different peptides to all the known human viruses, except for SARS-CoV-2, because they hadn't made that platform at the time that we were doing this. And you can see here that EBV, other viruses, CMV, the herpes viruses were picked up when we were looking at this. So we added those to our long COVID or post-acute sequelae assays. So this is what the data looked like when we looked at healthy controls. You can see here that a lot of people had positivity to serum and CMV, about 60%, and HSV. There was also some positivity, but the bottom line is that they were not, these are all before COVID. So these individuals are all before COVID. So here's what it looks like in the serum. And there was a little bit of reactivity to some of the EBV in the Mensa, for example, and we would probably see that in some patients who respond. So the question is, we've had a lot of talks about, Alzheimer's or perhaps multiple sclerosis that people have talked about with EBV, potentially causing reactivation, maybe with aging. If you have lots of reactivation, this is not a good thing to have. CMV, we know. So the question is, that's what it looked like in healthy people before the pandemic. After the pandemic, we recruited 23 patients who had fully recovered, and you can see here that they kind of look like most of them are positive in the serum. So, about 90 to 95% of them are positive for EBV, and you can see that about more than 60% were positive here for CMV, but it's not all of them. In HSV, you see similar results. So the bottom line is, in the Mensa assays, that's what's wrong with you today. There were only a few people, and that could be reactivation of EBV and CMV. This person may have had a herpes response, like a cold sore or something else. This wasn't always HSV2, so I'm sorry for this typo. This is a protein that could be recognized most herpes viruses, and you can see here that there's others for the Mensa. But when you look at the long COVID patients, and this is basically what we had with 60 patients, you can see they look identical to the patients who had fully recovered. But what we're starting to see is what's wrong with you today. There was 40% So let me see. Those are all the differences that we saw in the serum. But with what we saw in SARS-CoV-2, you could see that basically 40% were positive, about 40%, 37% were positive for EBV compared to the COVID recovered groups. CMV, it was 23% versus 4%. HSV was 15% versus 4%. And you can see that if we looked at all the herpes viruses, it was about 45% versus 17% for long COVID versus COVID recovered. And then when we looked at everybody in general, there's about 60% versus 17% in the COVID recovered. So long COVID, we could identify a lot of potential viral triggers that were triggered by these antigens that persisted in this. And we've learned a lot about those that could persist in the platelets and megakaryocytes, perhaps in the marrow. There's just so many reservoirs, and in the gut that we've all heard about today. We would love to do some of these assays together with the others as well. Then there's no differences that we see in the serum. What was really exciting was our collaboration with PolyBio. We were able to work with Max Kwan at the Venter Institute. This is Beverly and Peng that you heard from earlier today, and Max Kwan. And we were able to basically look at, we didn't have enough patients to do machine learning for our symptom scores, but using their whole group of, I think it was almost 1,800 patients, no, maybe 1,500 patients. I may have gotten that wrong, but the bottom line is they were able to do machine learning to do symptom scores. So we were able to layer our data on top and look at Mensa as a biomarker. And you can see that there was a pretty good correlation associated with that that was statistically significant. So then we had been working with PolyBio on another project, a separate project. They came to us and said, can you do Mensas in this other project that David is running from Mount Sinai, looking at these HIV repurposed drugs versus placebo in this group. And so basically, I just want to give you a brief update of what these samples showed in the Mensa study. So basically, in this study, what we did was we were getting baseline 90 days and 180 days after treatment. And when we did that, this is just half of the samples. We have 80 samples collected so far. At the baseline, we've analyzed 25, and 25, we see about 25%. We actually did others because we didn't know if they were newer strains of the virus, such as Omicron or Delta. So we wanted to make sure we kept it in line with that as well. There weren't as many CMV or EBV patients as we had seen the first time, or HSV, but you can see this is basically what it looks like in the plasma or the serum. We had a few patients at 90 days. Here they are. It's just not enough. I just want to tell you that we are moving forward. We analyzed 8 of them, and then these were 2 samples at 180 days. So just to let you know, more to come. I can't wait to finish this whole data set, but we were just running them. I don't know if I have time, but I just wanted to say that we were also using this Mensa assay from our data that we have with Lyme disease that we've done. We've made a multiplex assay. Everything key is like basically making sure you have all the antigens for multiplexing for Lyme disease. And this is what we've done, some early antigens, late, and then the C6 and the PepC10 peptides that are used currently for diagnosis. We enrolled, I can't remember, 30 some patients from Maine Medical with acute Lyme, and we followed them sequentially. This is from the lab that we had in Atlanta before the pandemic. And whole blood samples were collected at these multiple time points. And then what we did was, I just want to give you an example, a smattering of what this data looks like. It was just early in terms of some of them had an infection with resolution, some of them had an infection and they had sequelae, and we had blood samples from them. So this is what the data looks like from the person who had the infection. So you can see here they had the tick bite, they had a rash, they got treated, and then you can see here by day 16 they stopped the treatment with doxycycline, and then basically several days later they continued to have joint pain, muscle aches, and everything else. This is what I'm sorry, that they were completely resolved. They did not have any problems. This person had a Mensa response early on, they got treated, and the Mensa went away. Serum titers persist. So that's basically what I wanted to show. If you look at this patient who had a sequelae, basically they got a tick bite, got treated, still had fevers, chills, missed a couple doses, but ended their treatment with, they missed a draw, but they ended their treatment with doxycycline on day 18, but they continued to have fatigue and joint pain and persisted up to day 98. And you can see here that the Mensas are positive, whereas the serum also was positive in these individuals. So this was on a different assay that we were doing, but I just wanted to show you a smattering of another chronic disease that we think that we could potentially look at, so that may be useful for acute Lyme, for diagnosis of people who've had an infection previously, because they may have serum titers and the only diagnosis is with serum. Sorry for the typo for aid. That we could actually be helpful in diagnosing sequelae of Lyme, and that this may be useful for secondary Lyme infections. So not acute, sequelae. So we think Mensa could be an important snapshot to tell you what drivers there may be for long COVID from from the host immune response actively at that moment, from viral proteins that are present in your body at that time. For example, in long COVID, we've seen the early study that I showed you where we saw the responses to herpes viruses, and then the recent study that we've done with PolyBio and this past year, and looking at those, we haven't finished. So I just want to say that's just preliminary data. Don't want to make any conclusions from that just yet, but to show you that we're actively looking at it, and that it could potentially be an aid to diagnostics, and that perhaps we can make a Mensa test that is not just for long COVID, which is our original interest, but we can couple that with chronic Lyme. So we can do that together when people come in, and now that, COVID infection, everyone has had a COVID infection multiple times. So with that, I'll stop. I think I went a little bit over so that you can be released from your chains. So I want to thank, there's a huge number of people that I want to thank, the people in the lab who've been working on this assay, Natalie, especially Jack Deas at Beacon DX with Robin, as well as we want to really thank PolyBio. And we've been working with Michael Peluso at UCSF, trying to look at even if we can do this from the frozen sample. So I think we're close to 18 patients, fresh and frozen comparison. So seeing if we can do Mensa, because you really need fresh samples. So we really appreciate that collaboration, and especially the collaboration with David. And we really want, and Max, that has helped her, this data really come forward with the symptom scores. And Amy, of course, with PolyBio, we really thank her for all her support. So with that, I'll stop.
Of course. Wow, it would be huge to run the Mensa more on the chronic Lyme patients. I'm glad that you talked about that part. All right, then I'll introduce our next speaker, who's Marta Mirabent, and she's a postdoctoral researcher in Nadia Rowan's lab at the University of California, San Francisco. She'll be talking about SARS-CoV-2 persistence and T-cell activity in the female reproductive tract.
Well, thank you, Amy, for inviting us to talk about our recent study on the SARS-CoV-2 persistence and T-cell activity in the female reproductive tract. As it has already been broadly introduced, but just to mention that the LINK cohort was launched in 2020, aiming to try to provide research to understand a little bit better the long COVID pathology. And there's many different studies that have been looking at many different aspects. Some of them have been already mentioned today. I just wanted to point out that some of the studies have focused on the immunity on long COVID and have observed some immune dysregulation that might associate with long COVID. Here I'm highlighting one study that showed that there's some immune perturbations on certain populations in long COVID compared to regular patients. And not only that, if we look within the long COVID patients that in the LINK cohort, we see that they are overrepresented by female, and that has already been mentioned today in another cohort. But importantly, within those women, most of them are premenopausal, suggesting that there might be some implications of the sex hormones with long COVID pathology. So therefore, we ask, how might hormones affect the SARS-CoV-2 infection? Well, there's already some hypotheses that suggest that there might be some sex bias in SARS-CoV-2 infection. First of all, the ACE2 receptor, which is the receptor that SARS-CoV-2 uses to infect the cells, is regulated by sex hormones. But not only that, it has already been reported that females exhibit stronger innate responses. For example, they have higher interferon alpha responses in PDCs. But not only that, they also have greater antibody responses against viral infections, but also vaccination. And if we are talking about sex hormones, it's logical to think about the female reproductive tract, which is the most hormonally responsive tissue. And not only that, it is also a tolerogenic mucosal site. And Tim has done a brilliant presentation, actually showing us some updated information on the gut and the SARS-CoV-2 persistent in these tolerogenic mucosal sites. Not only that, unpublished studies in the lab have shown that there's higher frequency of SARS-CoV-2 specific responses in the endometrium 5 months post-infection, and that these responses are more than 10-fold higher than what we would see in blood. So there's a distinct immune response in the female reproductive tract. And as I already mentioned, there's higher prevalence of long COVID in women. And therefore, for all these reasons, we were very interested in understanding what was happening in the female reproductive tract during the long COVID. For that, we, thanks to the funding of PolyBio, we designed a study that focused on women and the female reproductive tract, and we aimed to answer 3 main questions. The first one was, is there persistent SARS-CoV-2 reservoir in these tissues? The second question was, what's the nature of the immune dysregulation in these tissues? And the third one was to assess whether there's specific SARS-CoV-2 responses in these tissues. And for that, we designed a study that would collect 6 different tissues, 3 regions from the gut, ileum, rectum, sigmoid rectum, and colon, and 3 different regions of the female reproductive tract, being in the endometrium, endocervix, and ectocervix. And we would also collect blood and nasal swabs. And we were targeting 5 long COVID patients and 5 controls that would help us understand what the differences in both the persistence but also the immune dysregulation there. And today I'm going to show you some data on both the first 2 questions, the presence of persistent SARS-CoV-2 and the immune nature of these tissues. And first of all, to answer these questions, we wanted to leverage the most advanced single-cell technology that 10x Genomics has released. This is a probe-based technology that therefore it uses probes to amplify the transcriptional to do the transcriptional profiling, and we combine that with a panel of more than 300 antibodies targeting membrane and intracellular. So therefore, we also have the protein expression on these cells, and that would help us understand the nature of the immune dysregulation. But we needed to complement this method to be able to detect the SARS-CoV-2 cells. For that, we designed 25 probes targeting different regions of the SARS-CoV-2 genome. And we also included within the CytSeq panel, the antibody panel, we included an antibody targeting the nucleocapsid protein. And because that's a new method, we call it COVID-Prex. So first of all, we wanted to validate how good was this method detecting the SARS-CoV-2 infected cells. For that, we use an in vitro infected model. Here I'm showing a UMAP of these controls. UMAP is a visualization that helps us understand how close cells are based on the transcriptional phenotype. On the left side, you have the UMAP of the uninfected cells. And on the right side, you have the UMAP of the in vitro infected cells. And the first thing that I want to point out is that all these red dots are cells that were positive for SARS-CoV, for any of the probes that we designed for the COVID RNA. So the first conclusion was that this method was sensitive enough to detect those cells, but also it was specific because we didn't saw any positive cells on the uninfected. So there was no background on this method. And the second thing that we were a little struck about is that, as you can see, there's an aisle of the infected cells, suggesting that the SARS-CoV-2 infection induces some transcriptional remodeling on those cells. And as I mentioned, we were targeting different regions of the SARS-CoV-2 genome. What I'm showing here is the signal on the different regions. As you can see, there's very light signal on some of the regions, but all of the regions were positive for. We could detect signal on all the different regions, but being nucleocapsid and spike, the regions that were giving us the highest signal. And not only that, I also mentioned that we included a nucleocapsid antibody. So here I'm showing a dot plot comparing the expression of the RNA on the y-axis and the expression of the protein, the nucleocapsid protein, on the x-axis. And as you can see, the uninfected control was clean for both of them. But on the in vitro infected sample, we were capable of detecting a very high signal both on the protein side and on the RNA side. Therefore, the conclusion of this first validation was that COVID-Prex is able to detect SARS-CoV-2 infected cells expressing both viral RNA and protein at the single cell level. And therefore, once the method was validated, we moved to apply this to our samples, our tissue sample cohort, to answer both the presence of the SARS-CoV-2 reservoir, but also the nature of the immune dysregulation. And I'm going to show you some data of the first donor that we've run. So here I'm showing again the HuMap of the different tissues of this donor. The red dots represent the COVID cells that were positive for RNA. And as you can see on the upper row, you have the three different regions of the gut, being ileum, colon, and sigmoid rectum in this order. And you can see that we were able to detect positive cells in all of the three different regions, being the colon the one that exhibit the highest amount of COVID RNA positive cells. And on the lower row, you have the three different regions of the female reproductive tract, being endometrium, endocervix, and ectocervix in this order. And also, again, we were able to detect COVID RNA positive cells in all of the three different tissues. And we further performed some cell annotation to try to investigate the phenotype and the cell class of these positive cells. And what we could see is that those COVID RNA positive cells distributed preferentially within the epithelial compartment. So what we can conclude for this second part is that we found evidence of SARS-CoV-2 infected cells both in the gut and the female reproductive tract on this donor that after more than 4 years after the initial infection. Although one of the caveats of this is that it is a blind study, so we don't know whether there was reinfections. What we know is that there's a negative PCR at the time that we performed the procedure. And the future directions of this study is that once we have in vitro validated and also validated this method in one of the donors, we are going to perform the same method. We have already collected an additional donor, and we have 2 other donors scheduled before the end of the year, and we're going to push to try to finalize the collection of the 5 long COVID and 5 control donors. And we are also collecting, so some of the tissues that we receive, we are saving them, we are preparing them and saving them for spatial imaging. Therefore, we would be able to have a very deep phenotyping of those cells, but also we would be able to locate those cells within the spatial context and try to understand if they have any relationship with the architecture of the tissue. And we are also separately moving forward with some in-depth analysis of the SARS-CoV-2 specific cell responses. And with that, I would like to acknowledge everyone involved in this project, especially my mentor, Nadia Mackenzie Roberts, that has been also highly involved in this project, all the UCSF Link team that has been extremely helpful on the recruitment of these patients and the funding, PolyBio funding, because it was great to be able to perform this study and the participants, because that's not an easy thing to ask for a participant. And with that, I'll be happy to answer any questions on the chat.
Wow, thanks, Marta. That's really just exciting early data. So thank you. Our next speaker is Mark Painter. He's a postdoctoral researcher in John Ware's lab at the University of Pennsylvania, and he'll be talking about T cells as biosensors of viral persistence in long COVID.
Does this look okay, Amy?
Yes.
Awesome. Okay, thank you, Amy, and thank you, everyone, for the wonderful presentations today. It's a pleasure to be here to give an update on our work using T cells as viral antigen biosensors in the context of long COVID. So we've had plenty of introduction to long COVID today, and what we really want to test is whether there are persistent viral antigens, either from SARS-CoV-2 or from other viruses like the herpes viruses, which we've heard quite a bit about, that are actively stimulating immune responses in people with these persisting symptoms after SARS-CoV-2 infection, very similar to what we just heard about using the Mensa assay. And so the concept is to use the T cells that are inside the patient as an in vivo biosensor of these viral antigens. And the way this conceptually works is that everybody has a diverse repertoire of T cells that are constantly surveying virtually all of the somatic cells in the body. Each of those T cells has a single T cell receptor that is highly specific for peptide, viral, in this case, viral peptide antigens being presented by MHC on the cell surface of an infected cell. The vast majority of those T cells do not recognize any given viral peptide, but those rare cells that do recognize a viral peptide, if it's detected by engaging peptide MHC on a virally infected cell, that specific T cell will become activated. And through decades of research, we know a lot about the processes downstream of T cell activation and the changes in gene expression and protein expression that can be detected in the lab. And so the concept is that these cells are surveying the tissues. If there are SARS-CoV-2 or herpes virus antigens present anywhere in the body, T cells will find that antigen, they'll become activated, and at least some portion of those T cells will circulate in the blood, still bearing these hallmarks of activation in their gene expression and their protein expression. And for this to work, we need to be able to fish out those rare virus-specific T cells from the blood. So the vast majority of the cells in the blood are not sensing any given virus, but if we can pull out those rare virus-specific cells, we can then ask whether they show any signs of having recently been stimulated by viral antigens in vivo. And so for that, we built a multiplexed panel of custom HLA class 1 tetramers for CD8 T cells. So these are HLA class 1 molecules loaded with peptides from spike, EBV lytic antigens, EBV latent antigens, flu antigens, VZV, CMV, or non-spike SARS-CoV-2 antigens. And then all of these tetramers are assembled on a unique combination of two fluorophores that we can then detect by flow cytometry. And what this data looks like is this, where the vast majority of the CD8 T cells in the blood will not bind to any of these viral peptide MHC tetramers, but rare populations will. This is data concatenated from many donors so that you can see a large number of cells. But you can see that these SARS-CoV-2 spike-specific cells can be sensitively pulled out from all of the other viral specificities, all from a single sample in a single well. And so we can really deeply then characterize, we can run millions of cells at a time on the flow cytometer and look for these rare virus-specific cells that are often as 1 in 10,000 CD8 T cells in the blood, find those cells and then see how they're reacting in vivo in the patient without any additional stimulation in vitro. So these cells, if they were activated in vivo, we'll be able to see that they were activated in our assays, and if they weren't, we aren't stimulating them in any way. So in order for this to work, we need to have samples, and we've been very lucky and blessed to have great collaborators, both here at Penn with Nuala Meyer and Ben Abramoff, at Yale through Akiko Yosaki, and then the UCSF team, who we've heard from quite a bit today. with Michael Peluso, Steve Deeks, and Tim Henrich, who have helped us accumulate a large number of samples from acute SARS-CoV-2 infection or vaccination, people who fully recovered after a SARS-CoV-2 infection, over 100 samples from over 50 participants, and then people with long COVID, over 340 samples from, or 340 samples from over 200 participants. And we're casting a very wide net here. So our long COVID cohorts include people all the way from, relatively mild to very severe symptoms, no specific symptom types, but at least one symptom persisting at least 4 months after infection. So we can take the CD8 T cells from all of these samples, this is hundreds of samples, and we can look at their expression of classic T cell markers, and we can use high dimensional clustering to separate these cells into some known cell types, where you can see like activated cells over here in pink, a lot of effector cells down here in the bottom, memory cells at the top, naive T cells that have never been stimulated here on the left. And if we look at bulk CD8 T cells, so just all of the T cells in the blood, from samples from acute infection with SARS-CoV-2, acute after mRNA vaccination, people who fully recovered, and long COVID patients, a few things jump out. One is that there's a very small but detectable signal of just broad T cell activation during acute infection. It's really not there in vaccination. And then the other thing that really jumps out is that if you just look at the total T cell populations, they look extremely similar between people who recover and people with long COVID. And if you look at the breakdown here, you can again see there's this very small signature of activation during acute infection, with some of these activated clusters being detected. But if you just look at total T cells in people who recover and people with long COVID, they look remarkably similar. And if you look specifically for activation of those T cells, you see no signature that there's elevated T cell activation in people with long COVID compared to people who recover. And I think broadly, this is, good news. It's encouraging that there's not a profound T cell defect or rewiring of the T cell compartment in people with long COVID. But rather, we're looking for something more subtle. As we know, most T cell responses are antigen specific and thus are operating on a very small percentage of the total T cell repertoire. And so can our tools to pull out these virus specific cells provide greater sensitivity by allowing us to detect only the virus specific cells? So I'm going to start looking at SARS-CoV-2 spike specific T cells. So we can find these T cells using our tetramer reagents. And here's just a participant where these cells are not activated. So this is kind of your classic person in the absence of viral antigens. These cells get activated and then they return to a resting state where they're not expressing these activation markers like PD-1 and CD39. But if we look in a different participant, they may actually have fewer spike-specific cells overall, and the total abundance of the response is, I think, poorly predictive here because of the tools we're using. But if you look at those cells, 60% of them in this person with long COVID are activated and remain activated for a year after that initial infection with repeat sampling. And so this is really striking as an example. And if we look across the whole cohort at the activation of spike-specific T cells, of course, during acute infection, spike-specific T cells are highly activated. And after mRNA vaccination with spike, you get very high activation of spike-specific T cells. In people who recover, activation is relatively low. And in most people with long COVID, we actually see similarly no evidence that SARS-CoV-2 spike is, spike antigens are persistently stimulating these T cells. But in a subset of people with long COVID, we see this persistent, elevated activation of spike-specific cells. It doesn't look exactly like acute infection. So it's not that there's like so much spike antigen everywhere that it's overwhelming these T cells and activating every single one to a high level, but rather that it seems like rare, or some, percentage, half of the T cells sometime within the last few weeks have encountered that antigen somewhere in the body. So it's suggestive, I think consistent with much of what's been discussed, that the reservoir of this virus might be relatively small, and yet it's there, and T cells are able to sense it, become activated, and show those signs of activation in the blood. And if we just look over time, we see this is on the x-axis, the activation of spike-specific cells at 4 months, and on the y-axis at 12 months. And you can see in people who recover on the left, activation is consistently low at both time points, 4 and 12 months post-infection. And then in people with long COVID, you see a large group that look just like the people who recover. I would say, at least with this T cell assay, no evidence of spike antigen driving an immune response. But then you see these other people, there's a strong correlation where you have persistent activation over time. And the exact level of activation may fluctuate a little bit, but they're elevated both at 4 and 12 months. And if anything, activation is increasing the further these people get from infection. So in summary, we see that SARS-CoV-2 persistence appears to drive sustained T cell activation in a subset of long COVID patients. And I just want to say that David Rieg in our lab has developed tetramers for CD4 T cells recognizing spike and has made very similar observations in CD4 T cells as well. We're not only looking at spike, of course, so we've also looked at EBV. We've heard quite a bit about EBV already. But what we see really strikingly is that during acute infection with SARS-CoV-2, it's very common to get activation of EBV-specific T cells. And these are specifically the lytic antigens that are expressed when EBV is actively replicating. This does not happen with vaccination. In people who fully recover, this activation tends to return to a relatively low level. But again, in long COVID, there's a subset of people that seem to have this persistent high activation of EBV. And we're starting to look into now using longitudinal sampling, whether this is really like that gets activated during acute infection and then never returns to baseline, which is our hypothesis here. And importantly, this is virus specific, so we don't see the same thing for CMV. We do see some evidence in a smaller proportion of people that you can get activation of CMV specific cells during acute infection with SARS-CoV-2, but there's no indication that activation of CMV-specific T cells, at least, is enriched in long COVID in our assays. And you see the exact same thing with flu, that you can probably get some bystander cytokine-driven activation of flu-specific T cells during acute infection, but there's no evidence that flu-specific T cells are elevated or activated in long COVID. So it suggests that it's really a virus-driven phenomenon. In like the last minute, I just want to talk about some of our single cell sequencing data now, where we've basically to look deeper at some of these cells than what we can look at by just flow cytometry, look at all of the genes that are being expressed by these virus-specific T cells from a number of long COVID, acute infection, and convalescent samples. We're able to identify classic gene expression clusters of CD8 T cells, including activated clusters here. And within our 10x single cell sequencing data, using oligonucleotide bar-coded tetramers, we're able to very sensitively identify virus-specific T cells within our sequencing data. We have thousands, tens of thousands of virus-specific T cells across all of these samples that we can study. We can see that the virus that a T cell recognizes has a large influence on the differentiation of those T cells and the types of genes they express. But what's really exciting is if we look at flu-specific T cells from people who recover during acute SARS-CoV-2 infection or long COVID, they all look very similar. They express very similar genes because this is not a flu-driven phenomenon. But if we look at SARS-CoV-2 spike-specific T cells, they look like flu-specific cells in people who fully recover because they're both respiratory viruses. Of course, during acute infection, those cells are activated, and so they express different genes. But in long COVID patients, we also see that those cells, in many cases, look like acutely infected cells from acutely infected individuals or acquire different phenotypes compared to what we see in people who fully recover. If we use the genes that are upregulated in the samples from the acute infection compared to people who fully recover. So this is really a signature of we know the antigen is present during acute infection. We assume it's likely absent in most people who recover fully. What gene expression changes do we see in spike-specific T cells in that setting? We can derive a signature of T cells that have recently been exposed to the spike antigen, and we can look for that signature in people with long COVID. And if we split the T cells from long COVID into people who showed evidence of activation in our flow cytometry data, so that subset with elevated spike activation versus the people who looked more like the convalescent controls, what you see is here in acute infection, you have a high expression of this gene signature of recent antigen encounter. In the people who recover, that's very low. And you can then see two different subsets of long COVID patients with spike-specific T cells that either show signs of having recently encountered spike somewhere in the body or those that have not. And specifically, we can see that these spike-specific T cells in long COVID, unlike the ones from people who fully recover, which cluster close together with flu-specific T cells, they're over here with the acutely activated spike-specific T cells, expressing a lot of genes that may be associated with T cell exhaustion, definitely genes of activation. I'm not going to go through all of this, but and clear signatures of effector biology and a lack of a classic memory signature like LEF1, CCR7, cell, TCF7. So what we see in summary is that bulk T cell populations are really not substantially altered in long COVID patients. Virus-specific T cells isolated from the blood, though, can be used as sensitive antigen biosensors in vivo to get a clearer indication of more nuanced biology than what you see looking at the total T cells. We see evidence of persistent viral antigen sensing in about 40, even almost half of our long COVID patients that we've tested, some with SARS-CoV-2, some with EBV, and a little bit of overlap in those two groups. And spike-specific T cells from some long COVID patients have activated effector features and lack memory features even months or years after infection, which I think is consistent with the SARS-CoV-2 persistence hypothesis in at least a subset of people. So I want to thank John Wary, my mentor, and Naomi Duick and David Rieg, who have been enormously helpful in performing these experiments. All of our collaborators here at Penn, Yale, and at UCSF, who I already mentioned, and of course, all the study participants and funding from PolyBio for supporting this project. So thank you.
Awesome. Thanks, Mark. That was great. Okay. All right. I'll introduce our next speaker, who is Marcelo Fiere. He is an associate professor in genomic medicine and infectious disease at the J. Craig Venter Institute, and he'll be talking about proteomics revealing immune markers in long COVID, vagal nerve stimulation clinical trial.
This is providing some updates in our multiple proteomics projects. We have done acute phase, long COVID projects in a cross-sectional manner, but this one is very interesting because it is an involvement of a clinical trial and checking saliva before and after vagal nerve stimulation. So the VNS clinical trial was developed and created in Mount Sinai, and David Petrino and Proal and many others are involved in that design. And my lab was responsible to evaluating the saliva samples and doing shotgun proteomics. So I'll provide some update by my interns that worked in this project during the summer. In my laboratory, we are very interested in the oral cavity as a major organ for the body health. There are things that can be detected in the oral cavity that can be a proxy to body's health, to the body health, but sometimes it's just related to the oral health, but sometimes can be used as a non-invasive mechanism. For example, it utilizes saliva. Because of that potential, we use also proteomics, which is a sequencing of proteins. And in that case, we're interested to understand what's unique of saliva, what's common to blood, what's unique to salivary glands versus the whole saliva, the passive drool. In addition, we're very interested in tissue biology. Either tissue or biofluids provides message, and there are signs within tissues that can lead us to understand disease or several stages of disease, mild, moderate, severe, but can also, hopefully in the future, And I saw some speakers earlier talking about the non-invasive needs, right? And to actually predict. So one modality is very important, but clearly after watching all the presentation, we have to think about multiple modalities within one model to really understand more long COVID. So for people, the audience that are not familiar with proteomics, it's a study of the proteins and the sequences and the functions. We try to understand what are the actual proteins present, and we do that via mass spectrometry. My laboratory is responsible and leads this human salivary proteome, which is an online and open source database for anyone, patients, clinicians, scientists can utilize, evaluate data that's uploaded there. And this chart here is not just a diagram, is in fact the actual number of proteins we have identified so far. So 3,209 saliva proteins were found in whole saliva, and some of those are common to blood. So we are understanding what's just in saliva and what's common to blood, but we also think of all the parts of the body and understand this by shotgun sequencing. So we use this portal not just to deposit things, but to evaluate differential expression analysis to identify key proteins in their abundance and biological functions. The Mount Sinai clinical trial basically recruited long COVID subjects and created a trial with sham and vagus nerve stimulation for treating long COVID subjects. And they collected multiple samples, including saliva before and after. So the idea is that there's inclusion criteria, some of the inclusion criteria for this project, but the idea is, can we make a difference in the patient's lives? And can we do something? And by doing that, can we find molecular cues that are responsible for those improvements? So the vagus nerve stimulation is very important because deliver electrical impulses to the vagus nerve. It's FDA approved for multiple other conditions and has some emerging uses, including anti-inflammatory in Alzheimer's and other conditions. So it was a no-brainer to think to use this for long COVID, which is multifactorial. So the recruitment were 50 long COVID patients, recruiting saliva samples, treatment with vagus nerve stimulation, placebo, and we have samples from visit 1 and visit 5. Visit 1 is the baseline, and visit 5 is the post-treatment. And this is our demographics of our samples. And our goals is really to understand something that's less invasive than collecting bone marrow samples and blood, and hopefully finding markers that sustain. So will be the data from visit 1 and visit 5. And we will do comparisons within treatment and between the treatment. So within the treatment, we have visit active treatment. We're doing visit 5, which is the post-treatment, the last one, right? So if patients improve, so that versus the baseline visit 1. And the active is actually this treatment, and then sham, it's that they do not get the full treatment. There might be a placebo effect. So there's visit 5 versus over visit 1. And also in between treatment components where patients are compared between active versus sham. The hypothesis here is that vagus nerve stimulation alters expression of salivary proteins associated with inflammation in long COVID. So here we see the sham, a volcano plot. So what you see on the right side is upregulation, where you see on the left side is downregulation. But I want to point out to the audience here the amount of upregulated when you don't have an active treatment, meaning a lot of salivary inflammatory proteins are super upregulated. So we have 139 upregulation in sham and 32. But then there is a shift. Once you have active treatment, there's a full shift in terms of the amount. And amount is important, but we also have to see what type of molecules are being up and down regulated. Clearly, we see that shift between sham and active. And this is very cool because we are using saliva, we're using shotgun proteomics to evaluate a response to something that was done systemically. So we are finding significant results. Now what our team is doing is understanding what are the functions of these proteins. How do they relate? I saw a speaker talk about network analysis, and that's something where we want to keep going. But the idea that the downregulated during active treatment demonstrated some inflammatory molecules, such as neutrophil mediators and some other immune cells mediators, S100 family of proteins. So there's an anti-inflammatory component happen in the VNS trial. There are other comparisons that we did. It may be too much to speak about all the comparisons, but again, if we do just baseline versus sham, we clearly see that there is more upregulation. But the minute we do the active versus over sham in visit 5, we start to see the downregulation. So there's a trend here in controlling the inflammatory molecules, which in another moment we can talk about specifically their functions. So some top significant molecules are listed here, top 15 upregulation and top 15 downregulation. We're very surprised how some inflammatory molecules were downregulated in the active treatment versus in the sham treatment, which has upregulation of caspase and some other molecules that are quite inflammatory, and meaning if the patient didn't have the VNS, then that inflammation continued or exacerbated. So here is the visit 5 active versus sham, and demonstrated some results that are really interesting, and we are still learning about the actual functions of each of those. But we are interested in neuroinflammation, interested in general inflammation, and we are dissecting this top upregulated and downregulated molecules. It's very rare to do any proteomics that you find significant difference in saliva pre and post treatment. So we know that some molecules that are involved here, they are regulating antiviral functions, interferon response, and also control innate immune cells such as neutrophils and macrophages. And there is some importance to the molecules that we're finding here in CD8 T cells as well. So we know that some of these molecules impact neutrophil extracellular traps, and such as S100 molecule that was downregulated after the treatment. And these traps control multiple chronic diseases, including cancer, lupus, and create vascular damage. So in conclusion, for our 10 minutes talk here, saliva proteomics provides a non-invasive method to monitor biological change, and we can see that it works really well for the VNS and long COVID clinical trial. We do have to investigate further how this top-regulated, up-regulated, and top-down-regulated are actually working together in providing signals that are meaningful for what we think are the markers of improvement or the markers of disease continuing in the sham. So we believe our hypothesis at this moment is supported, where the vagus nerve stimulation alters the expression of saliva proteins associated with inflammation and neuroinflammation in long COVID. So we're very thrilled about this direction of using saliva in clinical trials. I'd like to acknowledge Kashifa, who was the summer intern that took this project, and many other team members, Salim and Sarah, Brett, and many other team members that did the protocols for saliva extraction. We do a very different protocol, and we do the shotgun proteomics, and the funding, PolyBio and NIDCR, JCVI, and some of it from Prebys Foundation that allowed us to recruit interns during the summer. So thank you.
Thanks, Marcelo. It's a great example of how measuring different parameters in trial participants allows you to much more interpret the results with much more depth.
Exactly.
Great.
Thank you, PolyBio, for all the work that you guys are doing. Bye.
Of course. Yes, bye. All right. The next speaker is Max Kuhn, and he is a associate professor at the J. Craig Benner Institute. He'll be talking about long COVID endotype identification.
Thank you, Amy. Can you hear me?
Yes.
So yeah, I have presented this topic for previous 3 times. So today I'm going to focus on longitudinal and meta-analysis. So I think previously we focused on individual sites to identify the endotypes, but now it's time for me to summarize those results across sites. And for people who didn't know why we are doing this, that was because, I mean, the symptoms actually are persist, and also they have really, varying kind of complexity and across, different organ systems and time. So we are not the only group who is trying to solve this problem, but I think previous approaches did not really use machine learning very effectively, or they actually are focusing on EHR, which are difficult to be compared between different sites. So we are trying to see whether we can focus on questionnaires to identify some patterns, like to see, how the symptoms actually can be categorized into different endotypes. And the inputs are just like presence or absence of each individual symptom. And then we try to convert this into the metrics to represent the patient by symptom situation. And then we will summarize the data in a way for downstream analysis. So this table summarizes the data we have analyzed during the past, I would say, 18, 24 months. And I really appreciate everybody's support for providing me those data sets. And so there are 4 sites from UCSF. The link cohort is longitudinal. I think the keyword is longitudinal. And also from Sinai, so the keyword is the working clinic. So the cohort is, I would say, it could contain huge heterogeneity. And but Emory people provided me that really exciting MASA assay results, so that I can actually correlate those MASA results with the endotypes we found and the severity group we found. And Cardiff is the only case control study. So that means we actually have labels to tell us which patient is long COVID or not. Convalescent controls in long COVID. So in total, we actually analyzed about 1,600 subjects, and most of them are female. Yet that's very commonly seen in the long COVID cohort. In the method, I have introduced it for a few times before, but I want to remind people, so this is a pretty carefully designed pipeline. So it's, from the method perspective, it's probably not new, but from the pipeline perspective, it did lots of work to fine-tune each step and to automate the whole process to make sure it's actually effective for symptom analysis. In the 2 major steps in the pipeline, the first is the called topic modeling. So compare with those PCA or UMAP methods. So topic modeling is really designed for downstream cluster analysis. So it doesn't really compress your information into only 2 or 3 features for visualization, such as PCA and UMAP. It's actually designed for to reduce the dimensionality into, say, around 20, so that you can actually apply clustering methods to identify the clusters without being affected by the curse of dimensionality. And then second step is that we evaluate all the clustering methods and identify the optimal parameters to do this clustering analysis, to identify the symptom clusters. So you can see to the right side, so we first convert and transform the feature space using topic modeling, and then we apply data clustering to identify symptom signatures and symptom clusters. Based on the symptom signatures, symptom clusters, we derive the organ systems involved to define the endotypes, and we also predict the severity score for each individual cluster. Okay, so the first output, as I mentioned, so are those symptom clusters. I think compared with previous approach, we are able to identify the signature symptoms in a data-driven way. And then we actually plot those symptom signatures using the star chart, so we can see whether those patient profiles are consistent. So as I mentioned before, so if you don't do this process, so every patient is basically unique, because you look at all the characteristics, so everybody is unique. However, if we are able to identify a correct subset of symptoms, which are the signature, and then we will be able to see consistent profile of every individual patient in that group. So that's what you can see in the star charts below. So now every patient becomes similar to each other in this subspace. And we correlate these findings with some preconditions to explain why this patient reported these symptoms, and they match pretty well. And the second output is the endotype, which is defined by the organ system involved. So there's already some well-established mapping between symptoms and the organ systems. For example, if you if you have some like a constipation, that's probably related to your GI systems. So that's how we kind of map the current, say, 35 to 45 symptoms to 9 organ systems. And then based on the 9 organ systems, we can automatically merge the cluster into endotypes. And the table to the right side shows the mapping. So from those 16 clusters, we identify 9 endotypes, and what are those signatures in the organ system involved in those endotypes. And finally, so based on the scores of the topic modeling, so we are able to sum all the scores across the symptoms in the same cluster to produce a cluster-specific severity score. So compared with EQ5D, EQ5 score, so I think this severity score is not only quantitative, but also allows to link to the organ system as well as the single symptoms. So this will allow us to understand the severity better. And you're seeing one example only to see, so when we visualize our cluster on the UMAP, so those results map to the UMAP islands pretty well, and those very severe clusters will be very distant from the other clusters. And this actually match very well to our machine learning predicted severity scores. So you can clearly see there are 3 different severity levels, and they're pretty well segregated. And okay, but that's not the focus of today's presentation. So I'm trying to show you. So now we have a model to actually to profile the symptom changes. So the low-hanging fruit is like we are able to see, okay, what are the frequency of those kind of symptoms being reported along the time. So here we just show individual symptoms one by one to see how those symptoms change across the time. So we can see some symptoms actually got recover pretty well. So they kind of disappear after say a certain time period. For example, the trouble with smell, you can see they kind of disappear after 1 year. But there are some symptoms they are persistent. They got kind of reduced after a certain period. That's the category 2. And we can see difference between male versus female too. And then finally, those are very difficult symptoms. They not only got reduced, but they actually got even more severe after a certain time of period. And so those symptoms would be, I would say, really the focus for, from clinical treatment perspective. And so these are really informative, but they are not very useful. The useful thing is, so the machine learning now allow us to model all the things using the same cluster structure so that we can actually compare those symptom clusters across time. So you are seeing 3 UMAPs here. The first one is actually from the acute phase, which is before day 30. And the second one is early post-acute, so between day 30 to day 90. And the third one showing the after 90, so which are long COVID. So since they are organized by the machine learning topic modeling approaches, so we actually can compare how those symptoms evolve along the time. So the acute cluster is actually to the, I would say, the upper right side. And after 30 days, so you can see most patients actually began to have those kind of symptoms to the bottom side as here. And then finally, after 90 days, so those clusters got evolved to the left side, which are severe. So this, I would say, this black arrow shows how those symptoms got evolved from the acute to the long COVID. Let me remind you, so every cluster, we have the severity score. We also have the organ systems identified, so which allows to probably have some high level understanding of those etiology. And say, cluster 2 is a typical acute cluster, which is moderate, and it's related to temperature regulation and pulmonary issues. And after 30 days, I think the symptoms get a little bit mild, and you begin to see the musculoskeletal problems. But after 90 days, we are seeing this heterogeneity. We are seeing 4 severe clusters. And they actually get neurological systems involved, hormone functions on issues and sensory change induced. So this basically shows us how those symptoms go evolve along the time. And so to validate this kind of approach, we kind of try to map the machine learning predictor to the EQ5D, because we know EQ5D kind of reflects your overall health status. So we see a pretty good negative correlation, which means, so the more severe, the lower the EQ5D score is. So this is probably one of the most important evidence to show those scores pretty make sense. And based on the clusters and anotype, we are able to see which clusters actually will recover and which cluster will get worse. So I use the red rectangle to show this cluster actually will get worse symptoms. And the blue one shows, okay, this cluster will recover pretty well. If we don't divide this into clusters, we are not able to see this kind of cluster-specific recovery and change. And so before I leave, so I want to show you, so we actually can compare the results now across different cohorts. So we identified those 10 global endotypes by looking at the common symptoms shared by those patients. And there are many things we see from this, but I want to point to you. So one thing we are seeing is that now we are able to organize the organ systems. So based on this heat map shown here, so we are seeing cardiovascular is a pretty unique in distance in organ system. So people tend to report it, for example, neurological and sensory change symptoms together. So you always see those symptoms go reported on the same set of surveys, but cardiovascular is separate from the others. And then you also see the GI is also related to pulmonary. So they also got closer to each other in that dendrogram. If you look at the left side, so that dendrogram. So this will provide some information to tell you how you can actually group your patients into different, experiment groups or trials. And I think the bottom side shows you, so we actually can see the composition of those endotypes in male versus female. So we cannot compare female versus male directly because that depends on the study design, but we can see within female and within male. So what's the composition, and to show, okay, what's the situation regarding those endotypes in male versus female. And finally, so we can see this global score for measuring the severity across sites. So which means, so we are able to compare severity across different sites. So we are seeing this as a evidence-based definition of severity based on symptoms. So you can see here, so UCSF people, they actually have relatively milder symptoms compared with the treatment sites at Sinai as well as the Cardiff. And this also got confirmed by statistics. So the global severity score from the UCSF cohort actually lower than the other two cohorts. So actually, I would say those results map to each other pretty well. Okay, summary. So we have kind of conducted this machine learning analysis across 4 sites, and we are trying to do not only the cross-sectional, but also longitudinal analysis. We will try to summarize the results across to see whether we can identify the same thing across all the cohorts. But most importantly, so what I want to do is what we wanted to predict the future, predict the endotype of a new patient, and to predict the symptom change after a certain time period. So that will be my next step. And so everybody listed here is essential to this project. It's really a collaborative project. I appreciate everyone so much. And I think, yeah, that's all for today.
Awesome. Thanks so much, Max. Okay, our next speaker is Mario Murakami. He's a postdoctoral researcher in Mike Van Elzakker's lab at Harvard Medical School, and he'll be talking about vagus nerve sensitivity as a common infection-associated chronic disease mechanism.
Hi, everybody. My name is Mario Murakami, and I'm a postdoc in Mike's laboratory. It's a pleasure to be here and present my project. So I'm going to start with some premises. The vagus nerve is the gigantic nerve that innervates the major organs in the body and connects those informations from those organs to the brain. It can send some signals and information from those organs to the brain. And the way that it does, it has a hub in the dorsal brainstem and a nuclei called the nucleus solitary tract. This nucleus solitary tract is surrounded by other nuclei, and those nuclei are responsible for what we call the sickness behavior response. It produces the symptoms of pain, nausea, autonomic signaling, and fatigue. And our goal here in this project is to prove that persistent activation of the vagus nerve can cause sensitization, and this is one of the drivers of neuroinflammation. This is a continuation of my past presentation in other symposium. So we know about these 2 conditions, the very end and the very beginning. We know that those 2, myalgic encephalomyelitis and long COVID, has similar symptoms. So the end of the story, the consequences, those 2 have like similar clinical criteria of diagnosis: brain fog, pain, fatigue, autonomic dysfunction. And we know also the beginning of the story. We know that they are triggered by an infection, initial infection. We have some different pathogens to myalgic encephalomyelitis and COVID for long COVID. But what we don't know is the middle. It is the mechanisms, what those infections is causing to create those symptoms. We have just gigantic puzzle, and we have some glimpse of some pieces. Some pieces that we know is that we have neuroinflammation in chronic fatigue. We have neuroinflammation in long COVID, and this paper was created, this project was Mike's project. And also we know that there is some disruption in the blood-brain barrier. But the whole picture is still a little uncertain. Talking about just a little bit of blood-brain barrier, this is just blood-brain barrier is very complex. I'm gonna just give a glimpse. It is this physical vascular barrier that separates the body from the brain, but and it filters most of the metabolites that comes from the body. Now, one way that our body can communicate with the brain is through the vagus nerve that can bypass the blood-brain barrier and connect direct to the dorsal brainstem. So the way that it does is, as I explained, it has branches all over our body, and it can get information from those organs and send to the nucleus of solitary tract. The nucleus of solitary tract is gonna communicate all those insights and communicate to the brain. In a healthy control, in a healthy individual, exercise, intense light can stimulate this vagus nerve and send some signals. So now our brain understand what's happening with our body and can feel a different feeling. But what happens when a person has a chronic condition like long COVID is that this chronic reservoir or other type of input chronically activates the vagus nerve in a way that sensitize it. And when a normal input like this light or exercise or even stand up can cause major feelings, major symptoms actually. And those people chronically feel the symptoms, the brain fog, the pain, the fatigue. The way that it happens, what he hypothesized that it constantly activates the nucleus of solitary tract and all the nuclei that surrounds it, those blue nuclei that represents the nuclei that causes the sickness behavior response. So our hypothesis is that vagus nerve sensitization drives a dysfunctional brain and dorsal brainstem, the region where the nucleus of solitary tract is located. And if you prove it correct, those are evidence that there is some neuroimmune and metabolic dysfunction in the region of nucleus of solitary tract. The way that we did to prove that is perform this 3-model brain image. We acquired PET scan with this tracer that Mike explained already, the PBR28, that is a tracer of microclot activation that represents neuroinflammation. If we find increase, that is that neuroinflammation in this region. We also acquired functional MRI with a task. The MCIT, and Mike already explained too, is a concentration task. So those regions are actually dysfunction in function when the person is concentrated. So increase of both signal means dysfunction on those regions. And MRS, that is a neuroimaging that gets concentration of metabolites. And the way that we are going to see here is decrease of this metabolite, total N-acetyl-aspartate, that represents neuronal integrity marker. So if we find those 3 changes, it proves the evidence of disruption of neuroimmune and metabolic dysfunction in the nucleus of solitary tract. And the study design that we did that is we recruited 61 subjects, 14 healthy controls, 23 long COVID, and 24 ME/CFS. We collected data in this 3-model, multi-model MR machines in Martino Center, and we processed using those cutting-edge packages and pipelines. We processed the MCIT, the fMRI, the PET, MRS. We co-register all those images together, and then we perform the statistical analysis. And what we found is that, yes, there is some neuroinflammation. If you see in the circle, those are regions where has more activation of PET signal in the long COVID than non healthy control. So there is neuroinflammation in this dorsal brainstem, and also there is some dysfunction in the functional fMRI using this task. So dysfunction in the concentration in this region in long COVID compared with the healthy controls. We also found similar results with ME/CFS. The same region in the dorsal brainstem, we have more uptake of the TSPO that represents neuroinflammation in the dorsal brainstem, the region where the nucleus solitary tract is located. And we also found regions of dysfunction in the MCIT and the functional fMRI. All those regions we're talking about are those ones inside those red circles. And in the 3rd image, we also found decrease of that metabolite that represents neuronal integrity in this region of interest, that is the dorsal brainstem. So dysfunction of the brainstem in the region of the nucleus solitary tract. As an overview, this is the region that I'm talking about a lot, the nucleus of solitary tract located in the dorsal brainstem. And I'm gonna zoom the images that the results that I showed. There is neuroinflammation in the brainstem of dorsal long COVID, there is dysfunction of the fMRI, the test of concentration in the long COVID. There is neuroinflammation in the dorsal brainstem and the MCFS, and there is dysfunction in the fMRI in the MCFS, and also dysfunction in the metabolites that represents neuronal integrity in the MRS. So this proves part of this big puzzle that we are trying to solve. One of the pieces is dysfunction in the brainstem, in the solitary tract, that we think is because of this sensitization of the vagus nerve caused by these chronic conditions. And as a takeaway, I want to just emphasize the vagus nerve is this long nerve that connects all the information that we have from the body to the brain, bypassing the blood-brain barrier. The dorsal brainstem is the region where all those branches have and connect together in the nucleus of solitary tract. And we did find some consistent results with neuroinflammation in this region. We found neuroinflammation in the dorsal brainstem. We found dysfunction in the MCT, in the function fMRI, and also decrease of total NAA, that is dysfunction in the integrity of neuronal integrity in the dorsal brainstem. So as future directions, we want to increase the cohort. We have just 14 healthy controls, so we want to increase the number of healthy controls to improve the statistical power. We also have some results to be analyzed from the blood study. So we're going to add immunological markers to see some correlations, increase of cytokines correlating to those neuroinflammation markers in the neuroimaging. And we also want to finish the interventional study that we're doing, the vagus nerve stimulation, and see if this is increasing those cases also compared to controls. I want to say thank you to Mike, my PI. He's awesome, and the whole laboratory, Dina, Evelyn, and Seiji. I want to say thank you to Martinez and G8 and Harvard Medical School to make everything possible, and of course, PolyBio to funding our project. Thank you very much, and thank you everybody for listening.
Thanks, Mario. Great job. Okay, our next speaker is Zian Sang. He's a professor of medicine in the Division of Cardiology at the University of California, San Francisco, and he's talking about the COVID postmortem systematic investigation of sudden cardiac death study.
Hi, everyone. It's a long day, so thank you for sticking with the end of the day. I may be the only cardiologist, or maybe one of a few, and you'll see why. I'm going to introduce our study just by way of background. I'm not sure if everyone has been introduced with our particular study, but our study design, I think, is key to the advantages of what we have and filling in some of the gaps and some of the outstanding studies you've already heard. So recently, the AHA again affirmed that heart disease remains the leading cause of death in the country. And in fact, of heart diseases, sudden cardiac death itself is the #1 killer, remains the #1 killer, more than all the top 5 causes of death combined. And what's difficult about studying this condition is that it's out of hospital. And most of our detailed clinical studies have been in people who survive and come to the hospital to get catheterizations, echocardiograms, blood studies, etc. And we're missing the vast majority of these cases, which occur out of the hospital and never make it to the hospital. And the advantage of our study is that I've partnered with the medical examiner who receives all these out-of-hospital deaths, because they all have to be reported to the medical examiner. And the limitation in general of medical examiners' offices is that their charge is to investigate medical legal deaths, forensic deaths, not natural deaths. And I've lucked into an amazing partnership with Dr. Moffitt, the medical examiner in San Francisco, who shared a unique curiosity about these out-of-hospital sudden deaths, and was willing to partner with me to investigate all these out-of-hospital sudden deaths to define what the true underlying causes were. So it's a very efficient method, because I work with just one person to capture every single cardiac arrest in the city limits, in the county limits. And we've been doing this since 2011. So just very briefly, for those of you who haven't visited our beautiful city, it's about a little under 1 million population. During business hours, it swells to a little over 1.5 million. It's very racially diverse, so our findings can be applicable to pretty much any community in the United States, if not the world. We have 10 hospitals, 3 ambulance companies, and again, by state law, every one of these have to be reported to the medical examiner. So working with their office, I get to capture all these cases. And so again, here's Dr. Moffat. We published this, the original study that described our methodology a few years ago. Now we've since built this and continue to build this over the last 12, 13 years. Very busy slide, but I just wanted to point out, we start with every single death in the county. We look at all the cardiac arrests that didn't make it to the hospital. And then Dr. Moffat approaches the family and asks if they're okay with an autopsy. And as you see here, 97% autopsy rate, because most families want to know exactly what killed their loved one suddenly. So we have essentially complete capture of all these cases in our community. And so the punchline from our paper, that initial paper, is that the way we've defined sudden cardiac death is really not cardiac at all half the time. And in causes in green here are all the non-cardiac causes, and in red are the true cardiac causes. So again, only half the time is it truly cardiac. And so we've been able to define, with this data set, make discoveries in many different fields, such as occult overdose, contributing approximately 20% of underlying causes of sudden death, HIV itself as a strong risk factor for sudden cardiac death, device malfunctions such as pacemakers and defibrillators, and defining a new condition called sudden neurologic death. And so why am I telling you all this? I have an amazing team that reaches out to all of these family members and asks for consent for tissues. And so we built an amazing biobank of tissues. The medical examiner, Dr. Moffat, has collected samples from essentially every organ at the time of sudden death, so we can look into mechanisms. My particular interest, of course, is in arrhythmic and lethal arrhythmia mechanisms. So we've collected heart samples. We've also collected brain samples and gut samples, blood samples, etc. And we also know very precisely the underlying causes of sudden death in all of these cases. And so I partnered with Dr. Henrich, whose presentation you heard earlier, and we've taken advantage of our study design because we've been going since 2011. And so we have essentially 9 years of cases pre-2020, and that's, I think, a potential limitation of many other studies. They don't have historical controls before COVID came on the scene in 2020, and then we have now 5 years of cases since 2020. And our goal is to find the causes of long COVID. We're going to examine tissues across all these organ systems. And very importantly, these are ambulatory persons who were well up until the time they died suddenly. And that's exactly the population in whom long COVID affects. It's people who are ambulatory. They're not in the hospital. They're not sick. They're not confounded by immune responses that might happen simply because of the acute illness and the hospitalization. And I'll show you very important data to prove that point in a second. So we can examine viral persistence, immune dysregulation, interactions with chronic diseases like diabetes, endothelial dysfunction, metabolic dysfunction, and cell-cell crosstalk. So again, very importantly, in my field, we've been studying sudden cardiac arrest survivors, which is only 5% of the entire pie. And so our study has illuminated what we never get to see, which you never see in the hospital because they never make it there, sudden cardiac deaths. And very similarly here, many of our elegant autopsy studies based upon autopsy specimens have been done in people sick with COVID. But that's not the population that's ambulatory with long COVID, and so our study is able to illuminate that part of the pie. So as part of my study, we collect trauma deaths and as population controls. For my purposes, population controls with prevalent heart disease. But in this example I'm showing you, some of the trauma victims died instantly at the scene, and some survive for a few days and get transported to the ICU, where they ultimately die of their traumatic injuries. And so here is bulk transcriptomics from left ventricle in trauma victims who died instantly at the scene and those who made it to the hospital. And you can see here a huge disparity in the immune response simply because they were hospitalized in the ICU. And so it's a cautionary tale in interpreting inflammation responses from any hospital autopsy specimens. And so, again, here's our study design. And the medical examiner, has collected for us brain, gut, heart, liver. We have both fresh tissues and FFPE sections. We have what we call autopsy in a slide, very efficiently in one single slide. And then later, we can both study across organs, but within the same person and also in the same organ across different people, and looking at all these different responses. And so, so far we have 42 cases pre-2020 processed for a total of 208 bile specimens from all those different organs I show here. And then since 2020, we have 65 cases processed with also approximately 2,200 biopsy specimens from all the different organs throughout the body. And so with triplicates, now we have a total of 1,200 unique biospecimens that we've been working our way through with this support. And so I think I showed this before last time, but just again, this is in a person who died suddenly, who had recovered COVID and died suddenly of a cardiac cause. And we see SARS-CoV spike RNA in a neural tissue. So this is really sort of direct human tissue confirmation of some of these indirect radiologic studies that you've been able, that you may have heard earlier. Again, very important radiologic studies, but it's not looking directly at the tissue like we're able to do here. So we've seen now SARS-CoV spike RNA directly in neural tissue of people who've recovered from COVID. And so this sort of really validates some of the hypotheses that there's persistent SARS-CoV in the brain and inflammation that happens there. We're going to examine some of those spatial transcriptomics in process right now. But very importantly, what I want to point out is that because we have pre-2020, we have the ability to look at what is the virome, what is the infectious disease sort of milieu pre-2020. And in fact, we've seen EBV, for example, detected in the brain as well, and HIV in some of our our cases in San Francisco. Of course, we have approximately 4 to 5% prevalence of HIV. So we detected HIV in the brain, detected EBV in the brain, and so we're also then, and here's some of the examples here, high levels of HIV and EBV pre-2020. Again, pre-2020. And why is that an advantage? Because then we can look at how does that virome change, and what are the interactions between SARS-CoV and some of these other prevalent virome findings that we have pre-2020 since the pandemic started. And so right now, I skipped over this. This is progress we've made in cardiopulmonary tissues. So we've now processed 54 sudden death victims, myocardium, post-2020, tested by PCR. We've done RNA-scoped metagenomic sequencing and nCounter in process. We found one victim already with very high levels of SARS-CoV in the left ventricle with no evidence of myocarditis, but evidence of high levels of SARS-CoV in the heart itself. And also, again, in the brain and in the lung. This person died suddenly, didn't complain of frank viral illness, didn't seek care for COVID-19, and was SARS-CoV PCR positive at the time from the nasopharyngeal issue. So it was in the process of having an acute illness, but asymptomatic acute illness, actually. And again, an asymptomatic person like this would never have been captured, would not have come to any seek care, and would not have had the ability to study such a condition, such a person. And this is the type of person that maybe later on develops long COVID. And so we detected, again, high levels of SARS-CoV proteins in the myocardium and lung tissue. And so now we're currently in the process of doing metagenomic sequencing in all tissues and all time points. Again, we can detect RNA transcripts from all eukaryotic viruses in addition to SARS-CoV-2. We've already identified EBV in pre and post-2020 cases. And again, we can evaluate interactions between other viruses and SARS-CoV-2. It's a much higher sensitivity than the nCounter assay that we've been using. And so I'll stop there. Acknowledgements again to Dr. Moffitt. all the sudden death victims and their families in San Francisco County who made the ultimate sacrifice for our study. And also a big thanks to the PolyBio Foundation. As we have seen our government doesn't always function very well. And those of us who rely on government funding, we're thankful for alternative sources like PolyBio Foundation's generous support. So thank you for your attention.
Thanks, Ian. Really great overview. Okay, now I will introduce our last speaker today, who will then be Michael Peluso, who we've already met. He's an assistant professor in the Division of HIV, Infectious Diseases and Global Medicine at UCSF, and he'll be talking about supporting the immune response in long COVID, the INTERRUPT-LC trial, and clinical trial plans generally.
Yeah. Hey again. Thanks for sticking with this. It's been a really great meeting. This will not be a dense scientific talk, so I'm just going to tell you what our plans are for clinical trials for 2026. So, as has been discussed by several other speakers today, that we're at a phase in understanding long COVID biology where we have so many different leads. Some of those leads have become more prominent over the last 5 years. Other leads have sort of become, less prominent. And what our group really feels that we have to be doing to get answers for the millions of people with this condition is to be chasing down all of those leads. And in order to do that, it's not possible to simultaneously launch dozens and dozens of 1,000-person efficacy trials aiming to immediately get indications at the FDA. But it is possible to do a lot more experimental medicine. And what we mean when we talk about experimental medicine are basically trials in which we identify promising leads and target those pathways in a controlled way to understand whether we're able to alter the biology of those pathways, and if so, what happens to people clinically, symptomatically. And so, our MO here in San Francisco is that while we're involved in some of the larger network studies that, are really seeking efficacy outcomes, we do a lot of proof of concept work to really try to drive the agenda forward, very intense measurements. And we think that this is a really important component of making progress in this field, but it's one of those components that has been the hardest to support, especially at the level of the government and in programs like Recover. So I'm going to talk about 2 of our planned trials for 2026. The first is going to be INTERRUPT-LC, which we actually talked about a year ago on this call. And so the concept for this study is based in part on observations that Tim presented in his talk earlier today about the tissue-based immune perturbations that we're finding in people with long COVID compared to people who have fully recovered. And, we often talk about how people with long COVID have more inflammation. But as Tim was sort of alluding to, we think that inflammation is actually really driven by immune system dysfunction that is allowing things like viral persistence to occur, which is then in turn causing inflammation. And so what Tim has found in work from our tissue program in LINK is that we can identify things that are upregulated, and that's been the focus of much of the research over the last few years. But we can also identify factors that are downregulated. And some of these factors are related to coordination between different arms of the immune system and the antiviral immune response. So in collaboration with ImmunityBio and in collaboration with PolyBio, we are working on INTERRUPT-LC, which is a trial of Anktiva, this molecule called N803, which is an IL-15 agonist. IL-15 is a cytokine that we all make that controls a variety of downstream sort of signaling functions in the immune system. And the way that I think about it is that it really controls coordination between the innate and the adaptive immune systems. So, the innate immune system is things like the initial antiviral response, NK cells, et cetera. And then the adaptive immune system or, T cells and B cells and things like that. This drug is approved in a different formulation for certain types of cancer. And actually, we're interested in this drug because, as you know, those of us at UCSF have a background in HIV research, and we actually are also using this drug as a tool in mechanistic trials of HIV. So we have a lot of experience with this drug in people and in people with infections. And in HIV, we believe this drug has an effect on the HIV reservoir, on the ability of the immune system to see the HIV reservoir. And IL-15 signaling in general is thought to be very prominent in people who are able to control HIV without meds. So anyway, in this trial, we are hypothesizing that improving coordination between T cells and natural killer cells will actually improve the clearance of residual virus and maybe result in clinical improvement. So this is designed as a single arm open label study. And that's because it's very hard to blind studies of this drug. 2 doses of the drug. We're going to enroll 20 people, do baseline measurements, clinical assessments, and blood and tissue collections. We're hoping that the majority of people will do gut biopsies and lymph node FNAs. And then we will dose people with their first dose. It's administered subcutaneously at day 0. We have these little arrows represent intense follow-up in between. Then we'll give people a second dose a couple of weeks after that, intensely follow them, and then follow up at 30 days, 45 days, and 60 days. This is really a safety, tolerability, and biomarker study, but we're going to kind of do the full suite of link analyses that we do and that many have presented over the course of the day, and bank samples for downstream work. And it's been a long road to get here, but we actually officially opened this week and screened our first participant today. So if all goes according to plan, we'll begin dosing before Thanksgiving. So in the last few minutes here, I just want to make some comments about another study that we're hoping to launch in 2026. David Petrino presented a slide that showed this paper that a large group of us in PolyBio worked on earlier this year, talking about targeting the reservoir. We don't know the form in which the virus persists. There's data that has been presented on this call that this could be defunct RNA that isn't actively doing anything, but maybe is causing an immune response. There's data that has been presented that is consistent with a non-productive infection where virus is being transcribed and protein is being translated, but there might not be new infections of new target cells. And there's data, including double-stranded RNA data from multiple groups now, suggesting that there could be active replication. In this paper, we argued that really in order to figure this out, we're going to need to move beyond single therapy trials and do combination therapy trials. And the field is only starting to sort of grapple with this. But in this paper, we said specifically that we felt that combining direct-acting antivirals with monoclonals could simultaneously stop intracellular replication, enhance clearance of infected cells, and neutralize extracellular virus and protein. And we argued that this approach needs to be tested directly. So we've been working over the course of the last few months to design one of these experimental medicine, probe clinical trials that's called COMBAT-LC, which would be the first combination trial targeting specifically the SARS-CoV-2 reservoir in long COVID. This trial will use a combination of an antiviral Paxlovid, which I'll talk about in a second, and a monoclonal Pemgarda. Paxlovid studies so far have been negative, but we know that some people seem to benefit from antiviral treatment. It's possible that that's a placebo effect, but it's also possible that it's an on-target effect that we're actually trying to get. And I don't have time to go into the limitations of those other studies, but knowing whether people are, have a reservoir up front is really important. There has also been a lot of discussion over the last 6 months in the scientific literature, in the popular media, and on social media about PEMGARDA, including this paper that just was posted online a couple of weeks ago, suggesting that some people with long COVID seem to have a significant improvement with this monoclonal antibody. It's important to know that this monoclonal is not FDA approved. It is authorized, and so it cannot be prescribed legally off-label. And so this really needs to be done in the context of a monitored clinical trial. The cases that have been reported are people who met clinical criteria because they were immunocompromised. So this is an investigator-initiated study that we proposed to Invivyd, which is the company that makes PEMGARDA, and they, expressed willingness to donate the drug for the trial. The primary outcome is going to be safety and tolerability. There's going to be a variety of secondary outcomes. This is designed as a 2-center study here at UCSF and also at UCLA. Priscilla Hsu, who's the chief of cardiology now at UCLA, and I have been working to design it together. And it's really one of these sort of pure proof of concept studies. So what we'll propose to do in this trial is screen people in, do baseline assessments, and then we're actually going to stratify people at randomization based on whether they have antigen positivity in their blood. So this will be the first trial that is trying to use that as a methodology. 25 people will get PEMGARDA, 25 people will get placebo. And then once the monoclonal has been infused, or the placebo has been infused, we will give everybody in the trial 2 weeks of Paxlovid as antiviral treatment, and then do follow-up assessments over the subsequent months. We're also hoping to build in an open label crossover in this study, where the people who previously got placebo will then get an infusion of PEMGARDA at the end of the study and be followed again prospectively. And the reason that we're doing that is that there's a lot of power in that design because we can make a randomized comparison in terms of the biological outcomes in the main part of the study, but then we can actually do a comparison of people who got placebo plus the antiviral versus the same people then getting PEMGARDA. And that, for the biomarker work, is very enticing to be able to do that. So it's been a bit of a process. We submitted the investigator-initiated trial application earlier this year. It was approved. I now have an IND at the FDA to proceed with the study, and we recently got IRB approval. We're waiting on some final internal logistical approvals. We're currently looking to get the study fully funded so that we can launch early in 2026. But I think this trial, from our perspective, is really something that's very important to move forward the agenda. So I will end there. I want to specifically acknowledge ImmunityBio for their support of the Interrupt LC trial and PolyBio for their support of that. Invivyd for being willing to donate drug for Combat LC. And yeah, we'll see what happens in 2026. Thank you.
Great. Okay. It's a cool trial. I'm really excited about both. And I think activating the immune system in long COVID and going in that direction with Interrupt LC is a paradigm shift. Very few trials have been done to target persistent virus that support the immune response so clearly. So it's really cool. And with that, we will end the symposium. Thank you to all the speakers who did a great job and everyone who's working so hard on the research. It's coming together so well, and it's really amazing to see everyone's findings and studies connecting. We also thank all of our supporters and all of our donors and everyone who helps PolyBio exist. And we look forward to seeing everyone at the next symposium. So take care.